At the outset, I request everybody to please rise for the university song.
नित्यं हि विद्याचिन्तनं नित्यं च विद्यामन्थनं नित्यं हि विद्यासर्जनं नित्यं च विद्याजीवनम्
जनमन जागर करित निरंतर विद्यापीठाले जनमन जागर करित निरंतर विद्यापीठाले
संत गाडगे बाबा तुमचे स्वप्न पूर्ण झाले संत गाडगे बाबा तुमचे स्वप्न पूर्ण झाले
मुनीन आता खोळे घडानी ज्ञानाचं विज्ञान कळाले
पर्णकुटीतील सखी भेदानी आवाजे अमुक मिळाले
दशसूत्राचे अक्षर अक्षर वेद नवा बोले
संत गाडगे बाबा तुमचे स्वप्न पूर्ण झाले संत गाडगे बाबा तुमचे स्वप्न पूर्ण झाले किती रंधरे किती गाजरे किती आजळे डोळे पांगळे
कर्मयोग निष्ठा महाचरुण कवेत घेता बांधव सगळे भूतदयेच्या ओलाव्याने माणूस मोहरले संत गाडगे बाबा तुमचे स्वप्न पूर्ण झाले संत गाडगे बाबा तुमचे स्वप्न पूर्ण झाले धर्म जातीच्या पलीकडे माणूस केवळ माणूस असतो भेदभ्रमाचे बंध तोडून मानवतेचे पूजन करतो पिढ्यापिढ्यांचे जीवन दर्शन विश्वात्मक झाले संत गाडगे बाबा तुमचे स्वप्न पूर्ण झाले संत गाडगे बाबा तुमचे स्वप्न पूर्ण झाले जनमन जागर करित निरंतर विद्यापीठाले संत गाडगे बाबा तुमचे स्वप्न पूर्ण झाले तुमचे स्वप्न पूर्ण झाले तुमचे स्वप्न पूर्ण झाले
Thank you, everyone. Very good morning to all present over here. I take this privilege to warmly welcome you all for this 46th All India Botanical Conference of the Indian Botanical Society and International Symposium on Synergy in Plant Science and Sustainable Future 2023, organized by the Department of Botany, Sant Gadge Baba, Amravati University, Amravati. Very special thanks to you all for your endeared presence. My name is Dr. Neeraj Ghanwate, Associate Professor in the Department of Microbiology from this university, and I am delighted to be your anchor for this inaugural function. Before we begin, ladies and gentlemen, I would like to take a few moments in introducing to you our esteemed panel on the dais. For today's inauguration, we are blessed with the presence of Dr. P.A. Vadegaonkar, Honorable Pro Vice Chancellor and Head Department of Biotechnology, Sant Gadge Baba, Amravati University. He will be presiding today's inaugural event. Our chief guest, Dr. Atul Vaidya, Director, NIRI, Nagpur. We are blessed with the presence of President, Indian Botanical Society, Professor N.K. Dubey, the Secretary, Indian Botanical Society, Professor Seshu Lavania, Treasurer, Indian Botanical Society, Professor Alok Srivastav, and Editor-in-Chief, Indian Botanical Society Journal, Professor Y. Vimla, and our organizing secretary for this conference and head Department of Botany, Sant Gadge Baba, Amravati University, Professor Varsha Nathar. We are honored to have all of you with us today as we embark on this incredible journey of knowledge, innovation, and collaboration. This conference serves as a platform for experts, researchers, and professionals to discuss breakthroughs, share experiences, and foster meaningful connections. Let us begin today's auspicious event with the praise of the most merciful and the most benevolent. We will have our customary lamp lighting ceremony for seeking the choicest blessing of God and pay tribute to a great social reformer, Sant Gadge Baba, who told people the importance of proper education, self-reliance, and hygiene. I request all the dignitaries on the dais to please come forward for the auspicious inauguration.
स्वागतम करते हैं आज हम आपका स्वागतम करते हैं आज हम आपका स्वागतम
स्वागतम स्वागतम स्वागतम स्वागतम स्वागतम स्वागतम स्वागतम स्वागतम करते हैं आज हम आपका स्वागतम करते हैं आज हम आपका स्वागतम 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 स्वागतम
खिला सूखा चमन कलिया बागों की भी
दुख की बेला गई और विपदा की भी
हट गई मुश्किलें जो भी राहों में थी
बात जो थी कठिन आसान हो गई आपके साथ से जीत जाएंगे हम आपके साथ से जीत जाएंगे हम करते हैं आपका स्वागतम स्वागतम करते हैं आपका स्वागतम स्वागतम
बंद द्वारे सभी वो भी खुलने लगी
भूली राहें भी राहों से मिलने लगी
दूर थी मंजिलें साथ रहने लगी
बेला खुशियों की जीवन में आने लगी
आपके राह
में
में
चलेंगे हम आपके राह पे चलेंगे हम करते हैं आपका स्वागतम स्वागतम करते हैं आपका स्वागतम स्वागतम
करते हैं आज हम
आपका स्वागतम करते हैं आज हम आपका स्वागतम 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 स्वागतम
थैंक यू
Thank you, everyone, and thank you for the beautiful song. As a token of our heartfelt appreciation and gratitude for their invaluable time and support, we would like to welcome our guests on the dais by offering mementos. May I request Dr. Varsha Nathar to kindly come forward and welcome the chairperson of today's endeavor, Dr. P.A. Vadegaonkar, sir, Honorable Pro Vice Chancellor, Sant Gadge Baba, Amravati University, Amravati. I also request her to please greet our today's chief guest, Dr. Atul Vaidya, Director, NEERI, Nagpur. She will also greet the President, Indian Botanical Society, Professor N.K. Dubey. I also request her to please welcome the Treasurer, Indian Botanical Society, Professor Alok Srivastav. She will also greet the Editor-in-Chief, Indian Botanical Society, Journal, Dr. Y. Vimla. And I also request her to please greet Professor Shashu Lavania. Thank you so much, ma'am. To accomplish great things, we must not only act, but also dream. Not only plan, but also believe. I would specially like to thank and welcome Dr. Varsha Nathar, who has been a key person in helping throughout to design and take this conference to the state that it has been reality of the day today. Thank you so much, ma'am. Off the dais, I especially welcome all the present and former HODs, senior botanists, senior professors, the participants, faculty members, students of various departments, ladies and gentlemen. Before we proceed, let us take a moment to express our deepest gratitude to all the organizers, sponsors, volunteers and contributors who have made this event possible. Their dedication and hard work are commendable, and we are truly grateful for their unwavering support. Now, I would like to call Dr. Varsha Nathar, Organizing Secretary of this conference, for the welcome address.
A very pleasant morning to all of you. Dignitaries on the dais, Honorable Chairman, Professor Prasad Vadegaonkar sir, Pro Vice Chancellor and Head Department of Biotechnology of this university, our respected Chief Guest, Dr. Atul Vaidya, who is the Director of NEERI, Nagpur, President of IBS, Professor N.K. Dubey, who is from Banaras Hindu University, Varanasi, the Secretary of IBS, Professor Sheshu Lavania, who is from Lucknow, Treasurer of IBS, Professor Alok Srivastav, Editor-in-Chief of IBS, Professor Y. Vimla, of the dais, all the respected ECM members of IBS, all the senior botanists, all the invitees, all the heads of the departments of the university, all the delegates, the university authorities, the officers, ladies and gentlemen. It is indeed a great pleasure for me to offer you all a very warm welcome for this 46th All India Botanical Conference of the Indian Botanical Society and the International Symposium on Synergy in Plant Sciences and Sustainable Future. Really, we are very happy to have a galaxy of botanist fraternity today in our auditorium, and who have come all the way from a very long distances to attend this conference, which is 46th Botanical Conference, to say with how the journey began. The Department of Botany has already organized several conferences before at national and international level. We have also organized conferences with IAT, Indian Aerobiological Society. Only we were lacking with IBS. So we have given the priority that this year we should go with IBS. And it so happened, when the idea struck up, I just had a verse with Professor Chari sir, Professor S.R. Yadav sir, how to proceed. We are interested. And as they have said, we have given the letter to the Secretary of IBS, Professor Sheshu Lavania ma'am, and after that the process began. But while organizing this conference, it was really a very Herculean task to say with, because we are only 3 faculty members. And to organize a conference is not an easy task. But still, I would say, it is all the support of my students, as well as it is the blessings, it is the cooperation, it is the support of all the people like you. So I would like to thank all of you, that because of you all, we could see this picture today. So thank you all of you. Then the source of inspiration for this conference was our then and late Vice Chancellor, Dr. Dilip Malkede. When I moved the intent letter, I had kept 2 options, 2023 and 2024. But he called me, he said, Madam, why 24? Why not 23? And he pursued for that, and accordingly, it came up. And so, I should remember him also on this day. I thank all the Executive Council members of the Indian Botanical Society, especially for granting permission to conduct this 46th All India Botanical Conference in our university. So, really thank to all the Indian Botanical Society Executive Council members. I feel greatly honored to welcome all the delegates, all the fellow botanists who have come all the way from different long places to attend this conference. The another thing is that now how to go for the theme of the conference. So with the objective, the focal theme of the conference is Synergy in Plant Science and Sustainable Future. We all know that plants are inevitable for the welfare of human beings and progressive development of the society. Plants are always in interaction with nature, and they establish a sort of connection with entire network of natural resources. The plant science itself is the best example of synergistic approach, as well as the branches are all complementary and important for the progress and innovations in academics as well as in research. Plant science is directly connected and important for the ecosystem services and maintaining balance in the surrounding environment, so also in harnessing the natural resources overall development in society, and that to the civilized society. Sustainable development, as you all know, is related to conservation of biodiversity and bioresources. It is a prime concern to ensure the conducive future of our next generations. And synergy in plant science is a keystone component for sustainable future. I hope that with these ideas, we have designed the theme of the conference. And I wish that this conference will really be a fruitful, it will be an exciting one, it will be an encouraging one also, because it will coming up with an excitable outputs, also with discussions and interactions. And I wish you all a very happy stay, and I wish you all the best of luck, all the participants. With these words, I take my leave. Thank you.
Thank you so much, ma'am. The Indian Botanical Society is one of the national learned society for the botanists of India, and it was founded in 1920. Now I call upon Professor N.K. Dubey, BHU Varanasi, and the President IBS for welcome address.
Good morning to one and all present in the hall. Sabko namaskar. Distinguished dignitaries sitting on the dais, senior botanists. Some of them I realize they are my academic guardians, very senior botanists. Members of Executive Committee of Indian Botanical Society, young botanists, ladies and gentlemen. Today we all are gathered here to celebrate 46th annual meeting of Indian Botanical Society. It is indeed an honor to me to be here on the dais amongst the noted dignitaries. I have attended many annual meetings of Indian Botanical Society. I was member of Indian Botanical Society, life member I became when I started PhD under Professor S.N. Dikshit, and have missed only a few. On behalf of Indian Botanical Society and my own personal behalf, I welcome you all and express my gratitude to our chief guest, Professor Atul Vaidyaji, for coming here and gracing the occasion as chief guest. During the last meeting of Executive Council at Lucknow University, we were discussing the venue of coming next Indian Botanical Society meeting for this year. There were two proposals. One was from Jammu University and another one from Amravati University. Some of our senior botanists, Madam, mentioned their names, who were knowing Amravati University, highly appreciated Amravati University, talked about Professor Varsa on the mobile during the meeting. At that time, what happens, we members of Indian Botanical Society, hearing the appreciation of this department, this university, our ears were satisfied. But our eyes were not satisfied. Vice Chancellor Sahab, दूरे पिश्रुत्वा भवदीय कीर्ति कर्णो च तृप्तो नहीं चक्षुषि में तयो विवादम परिहर्तु कामा समागतो हम तव दर्शनाय। उस मीटिंग में आपके यूनिवर्सिटी के कीर्ति को सुन करके हम लोगों के कान सेटिस्फाई कर गए, लेकिन आंख नहीं सेटिस्फाई की। देयर वाज सम प्रॉब्लम बिटवीन ईयर एंड आई, दोनों में विवाद हो गया। तो उस प्रॉब्लम को सॉल्व करने के लिए आप लोगों ने बड़ा अच्छा किया, यहाँ हम लोगों को बुलाया। थैंक यू वेरी मच। You know, the truth must be told. I am thrilled to meet the organizing committee members of botany department at this university, who have, you see, went to airport late in night to fetch us. Friends, the 3 days gathering of Indian Botanical Society, we shall discuss on different botanical aspects and shall formulate and recommend some policies for future research. I express my deep regards to senior botanists. Their presence is inspiration for us. Young botanists from different parts of the country have come here. It gives me great hope and pleasure to see their shining faces and immense potential in them. The main motto of the meeting is to encourage the young botanists. Gathering of senior botanists with the young botanists reminds me here, Prachiti Prachi Kamel Sundar. I wish the annual meeting and the conference a great success. Again, good morning to everyone.
Thank you so much, sir. Indeed, we are happy to welcome you, and I am sure that our beautiful and lush green campus will also satisfy your eyes. Thank you so much. To know more about the Indian Botanical Society, I request Professor Sheshu Lavania, Secretary IBS, to please come forward.
Thank you, Nirja. Good morning to all of you. On behalf of the Indian Botanical Society and on my own, I extend a very warm welcome to the Pro-Vice Chancellor of this university, Professor P.A. Vadgaonkar, for so kindly accepting our invitation to inaugurate 46th All India Botanical Conference, accepting our invitation to inaugurate 46th All India Botanical Conference of the Indian Botanical Society. Sir, we value your commitments in supporting the cause of botanical sciences. It's my proud privilege to welcome Chief Guest Dr. Atul Vaidya, Director NERI, all dignitaries, guests, sorry for the interruption. It's my proud privilege to welcome Chief Guest Dr. Atul Vaidya, Director NERI, all dignitaries, guests, scientists, scholars, young participants present in the August gathering, having joined here from different parts of the country and also from Sri Lanka to treasure this moment. I extend a very warm welcome to all the invited speakers, senior botanists, medal awardees, chairpersons of different sessions for their gracious presence. I would like to highlight specifically for the knowledge of the younger generation who have joined recently as life members or the annual members about the journey of Indian Botanical Society. A journal named as the Journal of Botany, edited by Mr. P.F. Faison of the Presidency College, Madras, started its publication in September 1919. With required logistics and financial support from Mr. L.J. Sedgwick and Mr. T.R.D. Bell. Professor M.O.P. Iyengar of the Presidency College, Madras, soon joined the journal as its assistant editor in October 1921. Beginning with Volume 3, Number 4, April 1923, the journal was renamed as the Journal of Indian Botanical Society. Professor Birbal Sahani of Lucknow University took over as editor in 1927. The Indian Botanical Society owes its inception in a resolution passed by the Botany section of the Indian Science Congress on January the 7th, 1920 during its Nagpur session. A follow-up as an organizational committee chaired by Dr. Winfield Jejan of Ewing Christian College, Allahabad, Dr. Paul Brule of University College of Science, Calcutta, Dr. Rai Bahadur Rangachari of Agricultural College, Coimbatore, Professor Shivram Kashyap of the Government College, Lahore, Professor Birbal Sahani, then he was in Banaras Hindu University, and Dr. W. Burns, College of Agriculture, Pune, as its member, was constituted to formalize the society. In October 1920, the letters were sent to as many botanists to become the charter members, and in view of overwhelming response, the organizing committee could hold the election of the office bearers by the mid of November, and the society was formally declared organized on December the 6th, 1920. Starting with a modest beginning, having founding support of the botanical stalwarts of their time, the society continues to grow, and now we have over 2,580 life members, and this year we could add 150 annual members. I think it's a good number for the annual members also, because they are mostly the students. For 57 years, the society has held its annual sessions along with the sidelines of Indian Science Congress, with growing enthusiasm in the botanical fraternity towards the society, and to ensure proactive participation of budding botanists, the society made a new beginning in the year 1978 and decided to independently hold the Indian Annual Botanical Conference across the length and breadth of the country. The credit of this goes to late Professor Y.S. Murthy, who shaped and nurtured the activities of the Indian Botanical Society and made a robust beginning to organize the first All India Botanical Conference at Meerut in 1978. The present conference is 46th All India Botanical Conference in the series. The Society awards various medals every year. The Lifetime Achievement Award, instituted in 2009 through the contributions made by the family members of Professor Vishwambhar Puri, this award is to be given to a distinguished botanist who has rendered exceptional contribution to any branch of plant science and or services to the Indian Botanical Society. This year, the medal is being awarded to Professor S.K. Sapori, Fellow of the all three National Academies, Science Academies of India, and TWAS, Distinguished Fellow and Senior Emeritus Scientist, ICGEB. Professor Birbal Sahani Medal, instituted in 1956 through the contributions made by Professor T.S. Sadasivan to commemorate the memory of Professor Birbal Sahani, his revered teacher, and one of the founding members of the Indian Botanical Society. This medal is awarded every year to a distinguished Indian botanist for her contributions and devotions to the cause of Indian botany. This year, the medal is being awarded to Professor Amita Pal, Fellow Nasi, Kolkata. Professor Panchanan Maheshwari Medal, instituted in 1977 through the contributions made by Dr. K. Subramaniam, ex-director of the Botanical Survey of India, to commemorate the memory of Professor Panchanan Maheshwari, his revered guru, in recognition of his valuable contributions to the botanical sciences and in appreciation of his services to the society. This medal is being awarded every year to a distinguished Indian botanist for original, sustained, and significant contributions in the advancement of botanical knowledge. This year, the medal is being awarded to Professor R.D. Tripathi, Fellow Nasi, CSIR and BRI, Lucknow. Professor Vishwambhar Puri Medal, instituted in 1977 through the contributions made by Professor V. Puri Commemoration Organizing Committee for his well-known services to the society and the subject botany. This medal is awarded each year to a distinguished Indian botanist for her significant contributions to the advancement of botany, alternately for significant contributions to the morphology of angiosperms. This year, the medal is being awarded to Dr. A. Mao, Director, Botanical Survey of India, Kolkata. Professor Y.S. Murthy Medal, the medal is instituted in 1985 by the proceeds and endowment by Professor Y.S. Murthy Commemoration Organizing Committee. This medal is awarded each year to a distinguished young botanist or a fellow, a life member of the society of not more than 40 years of the age, for outstanding contributions in any branch of botany. This year, the medal is being awarded to Dr. Neha Pandey, Assistant Professor, CMP PG College, Prayagraj. Further, 2 new medal awards have been instituted in 2019 on the recommendation of the Executive Council and the General Body of the Society. Professor H.Y. Mohan Ram Memorial Award for Excellence in Teaching and Advancement of Plant Sciences. The recipient of this award this year is Professor A.K. Bhatnagar, New Delhi. Professor P.C. Trivedi Medal for Editorial Excellence is being awarded for the publications. The recipient for this award this year is Professor Ashwini Kumar of Jaipur. Further, the Society has 4 lecture awards to commemorate the memory of distinguished botanists. These lectures are to be delivered in alternative years: Professor G. Panigrahi and Professor L.B. Kajale Memorial Lecture Awards, Professor Umakant Sinha and Professor A.K. Sharma Memorial Lecture Awards. The memorial lecture in honor of Professor A.K. Sharma shall be delivered this year by Professor Anita Mukherjee from Calcutta University, Kolkata. Professor L.B. Kajale Memorial Lecture Award shall be delivered by Professor P.C. Trivedi, our ex-president, medal awardee, and Vice Chancellor of the 5 state universities, and he's a very big CV, of Jaipur. In addition, a memorial lecture in honor of Professor S.N. Chaturvedi was instituted in 2018, which is to be delivered at a place of choice of the awardee. Professor S.N. Chaturvedi Commemoration Memorial Lecture was delivered this year at MJP Rohilkhand University by Professor D.K. Maheshwari on October the 28th, 2013. Foundation Day Lecture Award. This lecture award was instituted in the centenary year by the Executive Council of the Society. Foundation Day Lecture of 2022 was delivered by Dr. D.K. Upreti, FNA of CSIR NVRI at Calicut University, Calicut. Competitive awards. In order to encourage young botanists of different age groups, the Society has provisions of various competitive awards. 1. Young Botanist Award, open to 35-year age category. To encourage young and budding botanists, the Society has Young Botanist Award for the life members below the age of 35 years. The award includes Dr. Bahadur Singh Gold Medal for the best paper presented under Young Botanist Award category. And here we always have a separate competitive session for the selection of the award. Women Botanist Award, open for 50-year category, age group, instituted from the contributions made by Professor Muniyamma in 2011. This award is given for the best paper presented by a lady botanist during the conference, and a separate competitive paper presentation session is arranged for the purpose. Poster award contest. There shall be 2 medal best poster award contests. 1, Professor K.S. Bilgrami Gold Medal, instituted in 2004 by the contribution made by the students of Professor Bilgrami. This medal is awarded to a young life member below the age of 35 years for the best poster presented during the conference. All sections included. We have a separate session for it. Professor S.N. Dikshit Gold Medal, authors below 50 years of age, instituted in 2012 by the students of Professor S.N. Dikshit. This medal is awarded to a life member below the age of 50 years for the best paper presented during the conference. Here also we have another set of poster presentation contest. Medals by application restricted to specific age group. Women Scientist Medal, instituted in 2006 by the Society. This medal is awarded to a distinguished lady botanist below the 50 years age for her outstanding research contributions in plant sciences. This year, the recipient is Dr. Jyoti Mathur from Banasthali. Professor Y.S. Murthy Memorial Gold Medal, instituted in 2012 by the late Professor C.M. Govil and Professor Y. Vimala and her family members in 2012. This medal is awarded to an outstanding plant morphologist or taxonomist up to the age of 50 years for her research contributions in any group of plant kingdom. This year, the recipient is Dr. Ritesh K. Chaudhary, Agarkar Institute, Pune. Professor S.V.S. Chauhan Gold Medal is instituted in 2013 by the students of Professor S.V.S. Chauhan. This medal is awarded to a life member below the age of 25 years. For the best oral paper presentation during the conference, all sections included. That means the youngest will get. To encourage young students up to the age of 30 years, medals, merit certificates are given in each section in honor of senior botanist if recommended by the experts. Rolling memorial lectures. 3 rolling memorial lectures have been instituted by the respective students and well-wishers in honor of Professor Y.S. Murthy, Professor Atul R. Mehta, and Professor C.M. Govil, Merit. These lectures are arranged in different universities across India. Every year, distinguished botanists are invited to deliver memorial lectures on the invitation of the host university. This year, Professor Y.S. Murthy Memorial Lecture was organized by Deen Dayal Upadhyaya University, Gorakhpur. Professor V.N. Pandey was the nodal person. Professor A.R. Mehta Memorial Lecture was organized by Professor Meenakshi Vaidya at Mumbai. Professor C.M. Govil Memorial Lecture was organized by Even Kishan College, and Dr. Sonali Chaturvedi was the nodal person. The society also felicitates all botanists who have attained the age of 60, 70, 80 and 90 years during this year. We have sent the invitations to all such botanists. Such invitees who are present here shall be felicitated in the inaugural function. Before I close, I may like to express my deep sense of gratitude to Professor Vadgaonkar, Pro-Vice Chancellor, authorities of this university, Professor Varsha Nathar, Organizing Secretary, her dynamic team I have seen since second, because I have arrived here on second evening, and for their support, continuous support, facilitating to hold this 46th All India Botanical Conference here in this university, Baba Gadge University. Let us hope that the deliberation at this conference would be purposeful and academic sessions would be stimulating. I believe that the conference would enlighten our young scientists to orient their future researchers in the service of our nation and humanity. Jai Hind.
Thank you, madam. We are very happy to host you. Being told you are appreciated is one of the simplest and most uplifting things you can hear. Awards or nominations serve as a positive drive to do better work, and it is something given to a recipient as a token of recognition of excellence in a certain field. Each year, IBS honors distinguished botanists from India for his or her contributions and devotion to the advancement of botany by conferring various awards, as just mentioned and informed by Professor Lavania. I call upon Professor Seshu Lavania to please announce this year's awards and recognition, which she has already done. She will be reading the citations for each award. I request all the dignitaries to please come forward to give away the awards. The very first award to be given is the Lifetime Achievement Award. And may I call upon Professor S.K. Sopori for the Lifetime Achievement Award on the stage.
Professor Sudhir K. Supori is not here. He is in USA. So I would read his citation, which we will be, I suppose, will be presented up here also. Fine. The Lifetime Achievement Award 2023 of the Indian Botanical Society is awarded to Professor S.K. Sopori, FNA, FASC, FNASC, FNAS, FTWAS, former Professor and Vice Chancellor, Jawaharlal Nehru University, and Group Leader and Director, International Center for Genetic Engineering and Biotechnology, ICGEB, New Delhi. During the 46th All India Botanical Conference held at Sant Gadge Baba Amravati University, Amravati from 4 to 6 November 2023, for his seminal contributions in understanding the genetic regulation of abiotic stresses and developmental physiology of plants. An elected fellow of all the 4 major science academies of India and of the World Academy of Science, Professor Sopori worked initially on the production of haploid plants via anther culture and has worked extensively to understand plant responses to light and stress conditions and identification of associated gene regulation and pathways. His recent work has shown role of melatonin as also prion-like proteins in stress memory responses. He has edited 13 books and published over 270 scientific papers in international journals and 60 book chapters. Amongst his many awards, he has been conferred National Civilian Award, Padma Shri, S.S. Bhatnagar Prize of Council of Scientific and Industrial Research, Birbal Sahani Medal of the Indian Botanical Society, Lifetime Achievement Award of the Indian Science Congress Association, PHSS Foundation of Biotechnology Society of India. He was the first Indian to receive corresponding membership of the American Society of Plant Biologists. He has been conferred DSc degree honorary from Banaras Hindu, honoris causa from Banaras Hindu University Varanasi and Rani Durgavati University Jabalpur. He was the president of Indian Society of Plant Physiology and vice president of Indian National Science Academy and National Academy of Sciences India. Currently, he is the president of Biotech Research Society of India and emeritus senior scientist at ICGEB, New Delhi. Thank you.
The next award is Professor Y.H. Mohan Ram Best Teacher Award. Is to be given to a distinguished teacher for excellence in teaching and advancement of plant sciences. I request Professor A.K. Bhatnagar to please come on the stage to receive this award.
Thank you, Nirja. Professor H.Y. Mohan Ram Award for Excellence in Teaching and Advancement of Plant Sciences 2023 of the Indian Botanical Society is awarded to Professor A.K. Bhatnagar, Fellow Nasi, former professor and head of the Department of Botany, University of Delhi, for his significant contributions to teaching and research, integrating traditional botanical disciplines with modern tools and societal needs. His scientific work is principally focused on embryology in relation to taxonomy, reproductive biology of rare threatened tree species, and on effect of diverse factors such as microalgal and mycorrhizal associations on some important crops and their products. He has published over 80 research papers in eminent international journals and another 60 articles in magazines and newspapers, besides co-authoring 1 and co-editing 4 books. Sir, please, your citation is coming, please. An elected fellow of the National Academy of Sciences, India 2019, Dr. Bhatnagar has been the president of Indian Botanical Society 2011-12 and the recipient of its commemoration Professor V. Puri Medal 2005. He is the president of the Orchid Society of India and the Society for Conservation and Resource Development of the Medicinal Plants, a former editor-in-chief of Phytomorphology and the Botanica. He is now editing International Journal Medicinal Plants since 2011. He has been member of advisory committee of many educational, research and regulatory bodies and has served as chairman of Ecosystems Research Committee and AICP on Taxonomy and of MOEFCC. Congratulations.
The next award is Professor P.C. Trivedi Medal for Editorial Excellence, which goes to Professor Ashwani Kumar. I please request Professor Ashwani Kumar to come on stage to receive this award.
Professor P.C. Trivedi Medal Award for Editorial Excellence 2023 of the Indian Botanical Society is awarded to Professor Ashwani Kumar, Alexander von Humboldt Fellow, Germany, former professor and head of Botany Department and Biotechnology, Director Life Sciences, Director Library and Information Sciences, University of Rajasthan, Jaipur, for his notable contributions as an author of textbook Plant Cell and the Tissue Culture, a Tool in Biotechnology, published by Springer in 2020, that has run into 2 editions, strategically designed to cater the need of advanced students and teachers. This book is clearly structured with 14 illustrated chapters and practical examples for easy understanding and direct implementation. Besides, he has 27 other edited volumes to his credit, published by international publishers, 5 in the past 5 years, covering topical issues related to biofuels, environmental biotechnology, environmental bioremediation, fern biotechnology, and genome editing. A brilliant scholar and gold medalist in his academic pursuits, he sidelined the lucrative administrative job of IPS, instead preferred preferred teaching and research inspired by his botanist father. He has immensely contributed in the services of botanical sciences, having published over 370 research papers, executed R&D projects funded by all major funding agencies of Government of India, mentored 39 students for PhD, occupying high positions in universities, including central universities. He has profusely participated in international scientific meetings as an invitee and to chair academic session, visited advanced labs as a recipient of Alexander von Humboldt Fellowship, Germany, visiting professorship twice to Japan Society for Promotion of Sciences, that is JSPS, INSA DFG Fellowship, Germany, British Council Program to the UK. He has been actively associated with IBS and was a recipient of Professor V. Puri Commemoration Medal 2008 and adored the office of President of the Society in 2019. Congratulations, sir.
The next award is the Women Scientist Award, is given to a distinguished lady botanist. May I call upon Dr. Jyoti Mathur to receive this award.
Women Scientist Medal of the Indian Botanical Society is awarded to Dr. Jyoti Mathur, Banasthali Vidyapeeth, for significant contributions in the area of phytoremediation for removing heavy metals and hydrocarbons from the industrially polluted soil using hyperaccumulator plants.
The next is Professor Y.S. Murthy Memorial Gold Medal, which is awarded each year to a distinguished young botanist. May I call upon Dr. Ritesh Kumar Chaudhary for this award.
Professor Y.S. Murthy Memorial Gold Medal of the Indian Botanical Society is awarded to Dr. Ritesh Kumar Chaudhary, Agarkar Research Institute, Pune, for his significant contributions in the field of angiosperm plant taxonomy. Congratulations.
Congratulations to all the awardees and thank you for gracing us with your presence. We are privileged to have such distinguished personalities among us who have significantly contributed to their respective fields and continue to inspire countless individuals worldwide. The Indian Botanical Society life members completing 5 years of membership are chosen by the society as fellow botanical society by calling applications, and they are selected by the committee on merit basis. I will call the names of the FBS to please, they will come on the stage to receive the FBS certificate. First of all, I call upon Professor Birendra Kumar from CSIR CMAP Lucknow to please come on the stage and receive the FBS certificate. Next, I would call upon Dr. Digambar Ramrao More from Narayan Rao Wagmare Mahavidyalay, Balapur, Hingoli, to please come on the stage to receive the certificate. May I call Dr. Juhi Dhillon from CCS University, Meerut, to please come on the stage to receive the FBS certificate.
Yes, ma'am.
Dr. Juhi Dhillon from CCS University, Meerut. Next, I would like to call Dr. Lakshman Nagar from CCS University, Meerut, to please come on the stage. Dr. Lakshman Nagar. Next, may I call upon Dr. Mahesh Vinayak Kaware from Dhote Bandhu Science College, Gondia, to please come on the stage. Dr. Mahesh Vinayak Kaware. Next, I would like to call Dr. Mohammad Shakil Ashraf from Bandipur, Jammu and Kashmir, to please come on the stage to receive the FBS certificate. Next, Dr. Nasir Aziz Anjum from Aligarh Muslim University, Aligarh, may please come on the stage to receive the certificate. Next, please, Dr. Nani Rajan from MV College, Buxar, please come on the stage. Dr. Nani Rajan. Next, may I call upon Dr. Neha Pandey from CMP Degree College, Prayagraj. Dr. Neha Pandey. Next, may I call Professor Shravan Shrivastava from Shri LBS PG College, Gonda. Dr. Vijay Vishnu Vagh from CSIR NBRI Lucknow. Please come on the stage to receive the certificate. Dr. Vijay Vishnu Vagh. Next, may I call Professor Vinod Shimpare from the New College, Kolhapur.
Congratulations, sir.
Thank you.
Professor Vinod Shimpare. Congratulations to all the FPS members. Effective senior leaders not only manage their respective teams but are responsible for creating a strategic direction for the organization, promoting a positive work environment. So we are going to felicitate the senior members of the society. So these people can manage changes efficiently and inspire other members to achieve their objectives. So let us felicitate the senior members of IBS for their unwavering contribution to IBS. I would like to call Dr. A.K. Pandey for completion of 70 years of active life and support to the Indian Botanical Society.
Thank you.
I would like to mention one more thing. Professor Pandey is the one person who is attending all the IBS since 1978. All, not even single.
Thank you, sir. Dr. A.K. Sharma, I think he is not there. Dr. D.A. Patil. I call Dr. D.A. Patil for completion of 70 years of active life and support to IBS.
मैंने कहा सबको मालूम होना चाहिए कि इतने IBS के करीब हैं. Somebody must not have एक बार मिस हुआ होगा, but not yours. Yes.
नमस्कार मैडम.
नमस्कार सर.
The next name is Dr. D.K. Maheshwari. I think he is not there. Dr. R.D. Tripathi. Please come on the stage. The next names Dr. R.K. Kohli and Dr. Tribhuvan Singh. They are not here. Next, I would like to call Professor Brindra Kumar for completion of 60 years of active life and support to the Indian Botanical Society. Professor Brindra Kumar.
Birendra Kumar ji, where are you? Dr. Birendra Kumar, CSR CMAP. Forgotten that he has completed 6 years of age. Please, sir. 60 साल वालों को भी हम लोग सम्मानित करते हैं। कोई बात नहीं, हम लोग साल से चलते हैं, डेढ़ से नहीं चलते। वो रिटायरमेंट वालों के लिए ठीक है, हम लोगों के लिए नहीं। 60वां साल चल रहा है, अभी तो पूरे होने में टाइम है। We only go by the year, not by the date. वो कभी नहीं होगी।
The next names Dr. Alok Kumar Krishna and Dr. Mukesh Kumar. They are not present here today. May I call Dr. Vinay Raure to please come on the stage.
Welcome, sir, in our cattle.
Doctors, 4 are there. Only 1 is there. Next name, Dr. Susie Albert. She is not there. Dr. Ashok Chauhan. Yes. On behalf. Then Dr. Suhana Binis and Dr. Sumitra Chaudhary were the next name, but as informed, they are not present. Now it is rather amazing that, may I request the dignitaries to please take their seats. They have been standing from long. And you might have seen, amazingly, none of these senior members, they seem to be 70 or above 60. And of course, they are an inspiration to all of us to remain as active and young they are. Thank you all, the respected senior members of the society. Now let us move towards the release of the conference abstract book. I request the dignitaries on the dais to release the abstract book and announce the inauguration of this conference. Thank you so much, everyone. The Department of Botany, Santa Gadge Baba Amravati University, has completed 33 years successfully with excellence in academics and research. The faculty of this department worked under various capacities with able leaderships of its former heads, senior professors, and those associated with this department. Let us pay due respect to their dedication and fatherly support to this department by felicitating them. May I call upon Dr. G.V. Patil sir, former Vice Chancellor of this university, to please come on the stage and accept our felicitation. We are so happy to see him. He's been ever inspiring, and you can see he still is equally dedicated to this university. May I request Professor S.R. Yadav sir, former professor and head, Department of Botany, Shivaji University, Kolhapur, to please come on the stage. These seniors members have always been associated with this department. May I request Professor K.H. Makde sir, former Professor and Head Botany Department, RTM Nagpur University, to please come on the stage and accept our felicitation and respect. Thank you so much, sir. I request Professor U.S. Chaudhary, sir, former Professor and Head Department of Botany, Santa Gadge Baba Amravati University. Okay. I request Dr. S.R. Manik, sir, former Professor and Head Botany Department, Santa Gadge Baba Amravati University, to please come on the stage.
This is such a dangerous one. हमको कहना चाहिए, इसको so slow बनाना चाहिए. This is very dangerous. Everybody falls. हर किसी को दिक्कत है. Steps दिखाएंगे. चतुर्वेदी है. Yes, she's there. She's there. She's also. Ashok Chaman is not there. Tidke sir and Chaturvedi madam. Should I call the names yet? Oh, no problem. Ashok Chaman sir is not there.
Thank you so much, sir. May I request Professor J.A. Tidke, sir, former Pro-Vice Chancellor of this university, Professor and Head Department of Botany, Santa Gadge Baba Amravati University, to please come on the stage and accept our felicitation. Thank you so much, sir. May I request Professor Alka Chaturvedi, madam, former Professor and Head Botany Department, RTM Nagpur University, to please come on the stage. Thank you so much, ma'am. Professor Ashok Chauhan, sir, former professor and head botany department from Baba Saheb Ambedkar Marathwada University, Aurangabad was the next name, but I think he's not there. Thank you all the respected members. I request the dignitaries to please take their seat on the dais. It's always so comfortable to have all these senior professors around us. It actually gives us that parental support in all our endeavors. Thank you so much, all of you. As we move forward, let's acknowledge that this conference is not just about listening to great ideas, but also about engaging in sessions. We have an exciting lineup of panel discussions, workshops, presentations in all these days, allowing you to delve deeper into specific areas of interest and interact with experts from various domains. Ladies and gentlemen, it is now time for the chief guest address, a moment that promises to set the tone for the conference. Our chief guest is a visionary in his field, renowned for his work on solid waste management and profound insights. Please welcome Dr. Atul Vaidya sir, Director, NEERI, Nagpur.
Still it is good morning, not at good afternoon. So good morning to all. At the outset, I first sincerely apologize for the delay for my arrival here from Nagpur, which was due to unexpected traffic conditions while getting out of the Nagpur and more than expected conditions at the entry of the Amravati. So these conditions were beyond my control, and I'm sorry the program got delayed probably because of my late arrival, and I sincerely regret for that. It's a great privilege to be here for this particular international symposium on synergy in plant science and sustainable future. And Amravati University is the place that is still, I'm more happy about it. I have been associated with this university since almost more than 20 years in different capacities. I have been here as a board of studies, or I have been here as an external examiner or in the selection committee panel, and I was also I was member of academic council of this prestigious university, and therefore when this was requested me to be here as a chief guest, I could not say no, obviously. And then one more thing which I would like to add to what Professor Dubeji has said. He had some discord between eyes and ears. I hope that is cleared, sir, after visiting Amravati, because this is the land of Gadge Baba and Sant Tukadoji Maharaj, where discord between the societies have been removed. So it was a very small discord, and I am sure it must have been removed after visiting the Amravati. And it's a very nice campus, nice city, and we are, as a Vidarbite, we are proud of Amravati as well as Nagpur. Now coming back to the conference, there are three important words which I can find here as the first and foremost is plant science, for which all of us are gathered here as a society. Botany is a science of, as I had learned, of course, I was a good botany student till 12th. After that, I opted for engineering. It is a plant science, or the science of plants, so many things there. But it is not, that is the 12th science definition, and we were related to taxonomy of botany or plants, and some cross sections of leaves and all those things, which are, botany was not limited to that, obviously, we realized it later on. But nevertheless, it is a very important topic as far as environment is concerned. I have been associated with environmental science and technology research as NEERI scientist and now as a director of NEERI. since almost more than 30 years. And we find that application of plant science is gaining importance. It was there, and probably NIRI had done pioneering work, and probably that is the reason we have, as a CSR, we have two important labs practically related to plant sciences. One is SIMAP Lucknow, from where my colleague has been felicitated, and another laboratory, that is National Botanical Research Institute. Again, one colleague from that institute was also felicitated. We have a dedicated lab like that, and NIRI being the seat of environmental sciences and technology, plant science, we have a separate department in NIRI also. So we do realize the importance of plant science. Environment, plant science, we talk in terms of green belt development, right in the beginning of this after say last 3 decades, when industrial development is there, development of green belt around the industry. This you must have heard, all of us have heard. And green belt is nothing but you must apply all your practical knowledge of the plant science or botany. You cannot have anything and everything in any plant there. It should be depending, you have to study the soil in and around, you have to study the climate, you have to study the industry type, and then you have to select the species which are to be planted in the green belt, so that the very purpose of green belt, to abate the pollution, is served. It cannot be a common thing, and therefore always we think that plant science is very important as far as green belt is concerned. But concept was not limited to that. Then we started talking of remediation of the land with the plant, and that itself, phytoremediation itself, is a big area in the environment which is there and which is still picking up. And there are many areas where phytoremediation finds its application. And again, here also you need lot of expertise in plant science. It is not that Atul Vaidya as a director NIRI will go and say you plant this and job is over. It's not like that. You need a very good plant scientist, you need a very good soil scientist, and they will decide what kind of plantation would come there for the purpose of phytoremediation, what are the species which are tolerant to the pollutants which are present in the soil, and then only, and with the systematic planning of plantation. Also, it cannot be just go plant and forget, it cannot be like that. So plant scientists would see to it that the properly plantation is done, it is executed, and it flourishes there, and it takes care of the phytoremediation, the very purpose of the plantation. So it's a big science in itself, where plant science or botany is of utmost importance. One more application which we find nowadays is the reclamation of degraded lands. It's not a contaminated sites. There is a difference between degraded land and contaminated site. And reclamation of degraded lands is another area where plant science has huge potential, huge. NIRI is experimenting on bamboo, you must be aware of that. We have practically tried bamboo plantation on degraded land, we tried bamboo plantation on waste dumps, otherwise nothing is growing there. We were successful in growing the bamboo plantation in and around, say, thermal power plants, on fly ash dumps, on many such dumps which cannot be removed, but at least they can be remediated with the phytoremediation. And I am very much sure that this is the area of plant scientists, and all NIRI, plant scientists from NIRI has done excellent job. One more example I would like to put here, that in 1996 to 2000, around that period, there was a mine spoil dump of MOIL, Manganese Ore India Limited, near Nagpur, near Ramtek rather, and that was a completely barren kind of hillock, where overburden dump from the mines were dumped, and now you will find that it is a lush green forest on that particular hillock. It's a huge area, and that was actually done with the help of plant biologists or plant scientists from our institute, with the help of MOIL, and it has set a very good example of reclamation of the spoiled dumps. And now it's a perfect natural ecology, it's a natural forest there. You will find all species and flora, fauna, everything everywhere in that particular forest. What I mean to say is that plant science and environment go hand in hand. Coming back to the sustainable future, now the current trend, as I would like to say a few words about that. There are two concepts, the two ideologies in science. If you go to the philosophy of the science, if you go into the physics or the chemistry or even the botany or any plant sciences, human being to consider observer as not as a part of the system and then developing a science. That is a classical physics or classical science. And considering observer also as a part of the system and his observation making the impact on the environment or on the science and the observations is another thought which probably followed in the quantum physics. Forgetting the nitty-gritties of all these basic things, the concept of human being standing alone outside the domain of flora, fauna and natural things has gone. We are in the era of one health. That means flora, fauna, human beings and all this earth, nature, we are one, and health of one will impact health of another. So it is not only my health at the premier because I am human being, I am the best race, so-called thinking animal. It is not so. We have to take care of flora, fauna and all other things in case you want to survive. And for sustainable future, this is a kind of mantra you can say, it's a one health. I cannot neglect any single component of the environment, whether it is living or non-living, because ultimately it will impact my own health. And therefore is the keyword. Unless you have a good coordination, good harmony between these 3-4 things, you cannot attain synergy. Whatever classical definition of synergy I have learned is that when two systems work together and achieve the better efficiency than the collective efficiency is called synergy. And if this synergy is to be achieved, you need a better harmony between different disciplines of the sciences, different disciplines of the environment, and for which such conferences like plant science and sustainable future, plant science and environment, or maybe animal science and I am not restricting myself to any particular science. Basically, we need to look for the synergies between different scientific disciplines in order to achieve one health or the, you can say, sustainable environment or sustainability goals. And therefore, I always think that although for specific purpose of career, specific purpose of our study, we have divided ourselves between different disciplines of science, but at the end of the day, if you look at the ultimate goal of the science and technology, we have to work together irrespective of what discipline you are working in, and therefore it calls for a very good harmony, very good cooperation between different disciplines, and therefore automatically you will achieve the synergy which is expected out of that. Sustainable future is mostly related to carbon footprinting, global warming, climate change, and carbon dioxide is one of the major issues which we are dealing with. Unfortunately, nature and natural systems and laws are like that. When I use energy for any purpose, I create either carbon dioxide or I create some lower form of energy, and that entropy will keep on increasing. That I always say, and therefore we cannot go against these natural laws. Purpose is whatever carbon dioxide is released, whether we can really capture that and whether a appropriate sink is available for that. And as all of us know, trees best thing. Now trees means green kind of flora. Not only trees, shrubs, even algae. Wherever chloroplast is there, it is a good sink for the carbon dioxide. And what human development has done, that we are cutting the forests and we are increasing the use of energy. We are releasing more and more CO2, and less we are actually reducing the sink. This is kind of putting axe on your own foot or own feet. For the luxury purpose, for comforts, for consumerism, you are just cutting the forest. You are reducing the green cover, you are reducing the sink. Basically what you are reducing green cover is reducing the sink for CO2, as if sustainability is considered. And therefore sustainability or sustainable future is directly linked to the plant growth, use of CO2 for nice purpose, for constructive purpose, reducing the energy requirements, and all these things are basically related to the theme of this particular conference. Therefore, I say that unless there is a synergy between all these sciences, not only plant science, we cannot achieve sustainable future. दुबे जी ने एक श्लोक कहा था, so what is sustainability? That it is actually taught by trees. एक हमें श्लोक याद आता है हमारे 10th क्लास का कि छाया यच्छन्ति पान्थाय तिष्ठति स्वयं आतपे. दूसरे को छाया देके खुद धूप में तपते हैं. ये ट्रीज के लिए ये वर्णन था. सस्टेनेबिलिटी का इससे ज़्यादा बड़ा उदाहरण कहीं नहीं है. Trees will capture CO2 and you will keep on releasing CO2. Still, tree will capture CO2 in modern context. And therefore, we have to look for sustainable future. We have to look for plant sciences, and we have to look for the synergy between all disciplines. And as a director of Environmental Research Institute, I know very well that what is the multidisciplinary programs and what are the importance of each and every discipline to achieve the goals of sustainability. I therefore congratulate Botanical Society, I congratulate Amravati University for organizing such an important conference, international symposium to touch the current issues of sustainability. I also congratulate all the seniors, all the stalwarts of this science, plant science, who have been felicitated here. Young scientists who have received the citations, scientists of my age who have received citations, and all young generation which should receive lot of citations, not only from this society, from environment as such, and from all departments of sciences. With this, I stop here. I congratulate you again. I wish all the best for this conference. Thank you very much.
Thank you, sir. Very truly said. There is no environment without plants. They do go hand in hand and exist in synergy. And plant sciences, not only plant sciences, but even the other sciences, help to keep them in harmony for a sustainable future. Thank you so much for your enlightening speech and for appreciating us. Your words have surely sparked our imagination and ignited a passion for exploring new frontiers. I take the privilege to welcome the chairperson of today's event, Dr. P.A. Vadegaonkar sir, Honorable Pro Vice Chancellor and Head Biotech Department, Sant Gadge Baba, Amravati University, for his presidential address.
गुरु गोविंद दो खड़े काके लागू पाए, बलिहारी गुरु आपकी गोविंद दियो बताए. Thank you, ma'am, for giving me an opportunity to take this opportunity and आप सबको प्रणाम करने का जो मुझे मौका मिला है, उसके लिए मैं मैडम का और प्रशांत का बहुत बहुत आभारी हूँ. डॉक्टर अतुल वैद्य, चीफ गेस्ट and Director Neeri, Professor Dubey, all office bearers of IBS, Professor G.V. Patil, former Vice Chancellor of this university, Professor Tirke, former Pro-Vice Chancellor of this university, all distinguished invitees and student friends. आप सबका संत गाड़गे बाबा अमरावती विद्यापीठ के परिसर में बहुत बहुत स्वागत है। दुबे सर ने जैसे कहा, मैं उसको आगे बढ़ाते हुए यह कहना चाहूँगा, दिस कैंपस इज एन एग्जाम्पल ऑफ फूड पिरामिड, बिकॉज द थीम ऑफ आवर conference is synergy in plant sciences and sustainable future. Future can be sustainable only when there is a complete food chain or food pyramid. Why I am saying we have the complete food pyramid, because the leopard is staying in this campus since last 6 years, and we are maintaining the complete pyramid. And if you are lucky, you can experience this. Otherwise, I will ask my students who are sitting there, they can show you the video made by them only. So this is the one of the best place to organize a conference when we are talking about the sustainable future. I will not take much time, but I am really grateful to you all that you have visited to this university. And personally, I will say that I have realized, otherwise I was thinking that there are only 2 or 3 years there for my retirement, but seeing this galaxy of scientists sitting here, अभी बहुत लंबी इनिंग खेलने की है आपके सबके आशीर्वाद के साथ. I am sure that I will be active in the academics for many years because of your blessings. I will not take much time, but I will just make a few statements. This type of conferences, and especially this type of conferences where 85, 86-year-old scientists are sitting here and 22-year-old budding scientists are sitting there. This is the best place for the networking. We have to spend time with each other. The students are having the best opportunity to interact with the people, make use of their experiences, and same time, इस ऑफिस में आने के बाद मुझे लगने लगा था, otherwise I was always 22, 23-year-old person when I am still with the students. So I'm sure this type of conference is making us young. So I'm sure that this conference will fulfill all the objectives, and I'm really grateful to Indian Botanical Society for organizing this international symposium in our campus. Again, I welcome you all. Thank you.
Thank you, sir, for your very kind words. Yes, as said by our Honorable Pro-Vice Chancellor, we do live with synergy and harmony with leopards in our campus. So lastly, let's remember that networking is a crucial aspect of this conference. Take the time to meet new people, exchange ideas, and forge connections that have the potential to lead to groundbreaking collaborations. Make the most of this platform to create lasting impressions and explore new horizons. We believe that this conference will not only broaden your knowledge, but also leave you inspired, motivated, and ready to contribute to a brighter future. Lastly, I call upon Dr. Prashant Gawande to please deliver the vote of thanks.
Thank you, madam. First, I pay my gratitude to the great educationalist of this region, Sant Gadge Baba, who is social reformer in the field of education as well as the cleanliness drive in Maharashtra. After his name, our university is dedicated. On behalf of organizing committee of this 46th All India Botanical Conference of the Indian Botanical Society and International Conference on Synergy in Plant Science and Sustainable Future, I, Dr. Prashant Gawande, convey my profound regard and hearty thanks to our late Professor Dr. Dilip Malkhede, who is a man behind the inception of this conference in our university. I am very much thankful to our Vice Chancellor Dr. Pramod Yevle sir, as well as I am very much thankful to our Pro Vice Chancellor and Chief Guest of today's function, Dr. Professor Prasad Walegaonkar sir. Along with this personality, one more person involved in this conference, Dr. Sanjay Dudul. He is a Dean Faculty of Science and Technology, for their constant support and possible guidance to organize such a great event in this campus. My special thanks to Professor N.K. Dubey, the President of Indian Botanical Society, for his gracious presence and rich patronage to host this event. At this juncture, I would like to thank Professor Shashu Lavania Madam, Secretary IBS, and Dr. Alok Srivastava, Treasurer and Editor-in-Chief of IBS. I am again thankful to Y. Vimla Madam for the countless help we received towards this conference. My words are not enough to express gratitude to the chief guest for this today's function, Dr. Atul Vaidya sir, Director NIRI Nagpur, for accepting our invitation and grace the function with his kind-hearted words. I will ever remain grateful to my head of the department and colleague Dr. V.N. Nathar Madam. She is organizing secretary, and also my colleague Dr. K.C. More, the joint organizing secretary, for their persistent support to organize this event. I feel proud and I am very much thankful to Professor A.K. Sapori, Professor A.K. Ashwin Kumar, Professor Jyoti Mathur, and Dr. Rizesh Kumar Chaudhary to be present here for the part and parcel of awardee ceremony. I express my deep sense of gratitude to senior members of from Botany Fraternity, Dr. G.V. Patil, our ex-Vice Chancellor, Dr. J.A. Tilke. He is our former Vice Chancellor and former Pro-Vice Chancellor of our university. Dr. S.R. Manik, former Professor and Head of our department. Dr. Alka Chaturvedi Madam, she is from Nagpur. Dr. A.K. Pandey sir, Dr. D.A. Patil sir, Dr. R.D. Tripathi, Dr. Birendra Kumar sir, Dr. Vinay Ravle, Dr. S.R. Yadav sir, and Dr. K.H. Makle sir, for making this event memorable to us I am thankful to most imperative one, our Registrar Dr. Tushar Deshmukh and Accounts and Finance Officer for administrative approval and organizing this event in this campus. They supported in all possible manner to organize this conference. I would like to thank all honorable delegates who blessed us their presence. I am also very much thankful to the Program Advisory Committee of this conference and invited speakers. Words are not enough to thank their constant guidance and support to shape up this type of event. My sincere thanks to all authorities of Sant Gadge Baba Amravati University, including Management Council, Academic Council and all academic bodies of the university. All members present here and this hall. I am very much thankful to the funding agency like DBT, ICSSR, SERB, DRDO, INSA, SERP, UGC and support from our financial partners BioEra, Labline, High Media and Shikshan Prasarak Mandal, SUM College, Ghatanji. It will have been not possible to organize this event without their support. I would like to thank our transportation and accommodation and catering team. Without they, we cannot organize this event successfully. At the end, I would like to take this opportunity to thank my nuclear counterpart of Botany Department, especially my MSc students, research scholars and all fellow teachers from our department. I again very much thankful to all delegates who comes from each and every corner of this nation. More than 345 delegates are here to attend this conference. I am very much thankful to all of you. Thank you very much.
Thank you, sir. So, once again, we extend our heartfelt gratitude to all the participants, speakers and sponsors for being part of this event. Let's embark on this remarkable journey together and make the most of the opportunities that lie ahead. Enjoy the conference. Thank you and good day. Just an announcement to make, there will be a break of just 10 minutes for tea, and the tea is arranged at the right side of the stage outside this auditorium. Please gather as soon as possible in the auditorium for the presidential address. Thank you. Thank you so much.
से रिक्वेस्ट है अंदर आ जाइए. एंड आई एम श्योर आप सबको पता है दुबे जी का लेक्चर बड़ा सिंटिलेटिंग रहता है. आप जाके मिस कर देंगे उसको. आइए अंदर. थैंक यू. आई रिक्वेस्ट दुबे सर. I request President Indian Botanical Society, Professor N.K. Dubey sir, to deliver presidential address.
नेक्स्ट के लिए और प्रीवियस
नेक्स्ट.
हाँ.
ठीक है. हमारा लेक्चर करो फर्स्ट कर दे. फर्स्ट good afternoon to everyone. Now we should start this session. We are late. Respected senior botanists and ladies and gentlemen who are present in this hall, I am feeling honored and privileged to have given the opportunity by the members of Executive Council of Indian Botanical Society to address on this occasion as President. I am thankful to them. Really, last year I got a mail, surprising mail, and soon I got a phone call from Dr. Lavania ji. Actually, from core of my heart, I feel I am a small person, but the members of Executive Council, they given the responsibility of this President of Indian Botanical Society, and many eminent botanists they have been given earlier. So I am really thankful to members of Executive Council of Indian Botanical Society. Friends, it is a big day for me, and I remember my teachers, Professor K.S. Bhargava, who has been also past president of Indian Botanical Society, and Professor S.N. Dikshit ji, and all my PhD students who helped in my academic career. I shall try to finish my lecture in time, maybe 4 or 5 minutes. Friends, this topic, a usual topic, which I have selected to deliver here is Indian ethno-medicinal plants, sustainable approach towards health security. This is my topic, and in this lecture I shall try to deliver before you, think locally, act globally, with reference to ethno-botanical plants of India towards miraculous sustainable approach for health security and green technology in agriculture. This is the era of modern system of medicine, allopathic system of medicine, and in this era I am going to deliver the old system of medicine, ethno-medicine. The thrust of my lecture will be why in this modern system of medicine, what is the perspectives of this ethno-medicine. Then if today also even there is recognition of medicinal plants, ethno-medicinal plants, what is the position of our country in the field of medicinal plants? If there are so many wonderful life-saving medicinal plants are found in this country, so what is our position in global herbal market? If this is not appropriate, what are the bottlenecks? What are the options before us? On these aspects I shall try to deliver before you. Friends, United Nations General Assembly in the year 2015 has designated 17 goals which are known as Sustainable Development Goals. And these goals are to be completed by 2030. Among these goals, goal 3 is to ensure healthy life. Not only healthy life, we are healthy, but also promote well-being, खुशहाल रहें. This is the UC objective of goal 3. I shall try to present before you how the ethno-medicine, medicinal plants may be and important components in fulfilling goal 3 of United Sustainable Development Goal, providing safe, effective, quality and affordable essential medicines. Friends, even today, 80% population of the world, they are behind the use of herbal products, either in cosmetics or for cure of different diseases. And this percentage is not only in the biodiversity-rich countries, even the European countries which have less number of biodiversity, they also use herbal products for different purposes. Friends, in Ayurveda, there is a slogan, similar cures to similar. If the plant part is like heart-shaped, leaf is heart-shaped, that part will be used for cure of heart diseases. If it is kidney-shaped, reniform, that will be used in cure of kidney diseases. Based on these different old botanical gardens, the CMAP is as example, they are creating Mano garden. Mano, and for the cure of different diseases of the organs, there is a plant, Mano garden. And because of this, 80% population of the world, they are behind use of herbal products. When we grow old, जब हम बुढ़े हो जाते हैं, तो we face different types of age-related diseases. Among the important age-related diseases is कि हम भूलने लगते हैं. We face dementia, we face Alzheimer's disease, and for during old age, our body is not able to tolerate the side effect of different types of allopathic medicines. The safer option is the use of herbal products. Products of Vinca rosea, Risalpine, Vibin, Vinking, or Curcumin, or the safer one is Jatamansi root along with Medyabati in milk. Friends, when antibiotics were discovered, what happened? They cured the diseases within limited period. And Paul Ehrlich told that these are magic bullets, a magic bullet है जो टाइफाइड हर्बल प्रोडक्ट से 42 दिन में क्योर होता था, 5 दिन में क्योर हो जाता था. What happened for that antibiotic? कि the discoverer was felicitated by Nobel Prize, ये उसको दिया गया. आज तक किसी भी आयुर्वेदिक प्रोडक्ट में ये सब नहीं मिला है. भारत रत्न नहीं मिला है, जबकि हम कितने साल से यूज करते चले आ रहे हैं. नोबेल प्राइज उनको मिला जो डिस्कवर थे एंटीबायोटिक के. लेकिन क्या हुआ कि द माइक्रो ऑर्गेनिज्म इजिली डेवलप रेजिस्टेंट स्ट्रेन, जैसे कोरोना कह रहा है, एंड दैट डिस्कवर्ड एंटीबायोटिक, जिसको इतना हम लोग रिकॉग्नाइज किए, दैट बिकम्स आउट ऑफ डेट. Then again antibiotic, again you see the Nobel laureate that got the you see Nobel Prize, that person got the Nobel Prize. So this development of resistance against the antibiotic, because the antibiotics are having single mode of action based on single product, and the herbal products are decoction of many components act synergistically. Against them it is very difficult to you see induce resistance to pepper, to develop resistance against the herbal products. So this development of resistance against the antibiotic easily is another reason that 80% population of the world, they are behind the herbal products. What happens in the modern pharmacopoeia? There are many drugs which are plant-based drugs. The willow tree, Salix alba, was used by Native Americans long ago, since long ago. Their company visited Native America in the 1899, and based on the ethnomedicinal property of this Salix alba bark, कि वो दर्द को कम करते हैं, they formulated a drug, aspirin, anti-inflammatory drug, aspirin. This aspirin is nothing but a synthesized analog of salicin, which is found in the bark of Salix alba. हमारे कहने का मतलब यह है कि there are many plant-based products which are integral part of allopathic system of medicine. This is one example. Another example is Papaver somniferum, young fruits of Papaver somniferum that is used medicinally. And the first of all, the technician, assistant of pharmaceutics in 1803, he got the medicinal property of the plant कि उसको जब सुलाया जाता है, खिलाया जाता है तो आदमी सोने लगता है. तो उससे उसने निकाला एक ड्रग जो कि सिडेटिव इन नेचर है, और ग्रीक मिथोलॉजी में सोने वाले देवता, ड्रीम लाने वाले देवता, ही इज नोन एज मार्फियस. तो इसके नाम पर उस ड्रग का नाम मार्फिन पड़ा, विच इज यूज्ड टू काम द क्राइंग चिल्ड्रन. बच्चे जब ट्रेन में रोने लगे, उसको लो डोज पर खिला दिया जाता है, बच्चे सो जाते हैं. एज एनेस्थीसिया एट हायर डोजेज इट इज हार्मफुल. एंड दिस Papaver somniferum was used also in export, history of export import. In the 18th century, European countries, they demanded Chinese goods. चाय उनको ज्यादा चाहिए था. चाय के बदले में वो यूरोप से सिल्वर चाइना को देते थे. बाद में यूरोप का सिल्वर खत्म हो गया, कैसे उसके साथ चाइना के साथ वो टी का ट्रेड करें. इन दी
year around 18th century, East India Company 1773 that conquered Bengal. बंगाल को उसने जीता। बंगाल में ज्यादा पेपर सोमेनिफेरम होता है और इस पेपर सोमेनिफेरम से उन लोगों ने अपना एक्सपोर्ट इंपोर्ट का बिजनेस चाइना से किया। Another plant that is member of Amaryllidaceae, Galanthus, two species of Galanthus, they give us galantamine. This is plant-based products and it is still choline esterase inhibitor, dementia में use होता है। So I wanted to say that there are many plant-based products which are part of allopathic system of medicine, and because of this 80% population of the world they are behind the use of herbal products. कैसे हमको पता चला? बहुत पहले जब साइंस नहीं था कि किसी पौधे में मेडिसिनल प्रॉपर्टी है। क्या होता था कि पहले आदमी जंगल में रहता था, जानवरों के एक्टिविटी को, बिहेवियर को ऑब्जर्व करता था। हिमालय के तराई बेल्ट में लोग देखते थे कि सांप, बड़ा सांप और छोटे नेवला में जब लड़ाई होती है, ज्यादातर नेवला जीत जाता है 1 घंटे के लड़ाई में। क्यों जीत जाता है? कि हमारे ऋषि महात्मा, पर्टिकुलरली इंडिया, उन लोगों ने देखा कि बिफोर द स्टार्ट ऑफ द फाइट नेवला गोज, मैंगूज गोज, ही डिग्स द रूट ऑफ ए प्लांट, एक पौधे के रूट को खाता है और वो तब सांप से घंटों लड़ता है। वो अपने ब्लड प्रेशर को कंट्रोल करता है, लो ब्लड प्रेशर, हाई ब्लड प्रेशर को कंट्रोल करता है। सांप कंट्रोल नहीं कर पाता है। वी केम टू नो अबाउट ए प्लांट दैट वी कॉल सर्पगंधा और राउल्फिया सर्पेंटाइना। When there is pain in the stomach of chimpanzee, चिंपैंजी के पेट पे, गोरिल्ला के पेट में जब पेन होता है तो नार्फ्लक्स नहीं खाता था, किसी पौधे के पत्ती को खाता था, जिंजबरेसी के पौधों के पत्ती को खाता था। वी केम टू नो अबाउट मेडिसिनल प्रॉपर्टी ऑफ मेंबर्स ऑफ जिंजबरेसी। तो दिस जू फार्मेकोलॉजी हैज हेल्प्ड अस, एनिमल सेल्फ मेडिकेशन long ago has helped us in discovery of different types of medicinal plants. भारत पाकिस्तान में बंटवारा हुआ, हम लोग बात करते हैं, दुखी होते हैं कि हमारा अच्छा सा कश्मीर का एक टुकड़ा पाकिस्तान के पास चला गया, लेकिन एक और जगह है जिसको हम लोग गिलगिट कहते हैं, और वो गिलगिट क्या है कि मैदान से एकदम कटा हुआ है, पहाड़ी इलाका है, वहां के लोग मैदान के सुख सुविधा को नहीं पा पाते हैं, लेकिन गिलगिट वहां खाना बनाने के लिए ईंधन गैस का सप्लाई नहीं हो सकता है। लेकिन गिलगिट के लोगों की विशेषता क्या है कि द पीपुल ऑफ गिलगिट दे आर मोस्ट ब्यूटीफुल इन द वर्ल्ड, दुनिया में सबसे ज्यादा सुंदर होते हैं, और उनकी जो आयु होती है, लॉन्जिविटी होती है, दैट इज मोर देन नॉर्मल, मोर देन 100 ईयर, 125 साल तक वे लोग जिंदा रहते हैं। मदर इज ऑफ 92 ईयर, डॉटर इज ऑफ 75 ईयर, फादर इज ऑफ 95 ईयर, सन इज ऑफ 75 ईयर। ये है, देखिए इतने साल तक क्यों उनकी लॉन्जिविटी ज्यादा होती है, क्यों इतने सुंदर होते हैं, क्योंकि वो पकाया हुआ खाना नहीं खाते हैं। दे डिपेंड ऑन अनकुक्ड फूड, दे डिपेंड ऑन एप्रिकॉट, फ्रेश चेरीज, ग्रेप्स, पीचेज, इसको वे खाते हैं। हुंजा वैली के, खत्म हो गया, लगा दो, इसको वे खाते हैं, एंड दिस इज द रीजन फॉर देयर लॉन्जिविटी। वहां पर जो हुंजा रिवर है, एल्कलाइन पीएच पर कैंसर जल्दी नहीं होता है, इसी से जापान और कोरिया में कैंसर कम होता है। द वाटर ऑफ हुंजा रिवर इज अल्कलाइन, ये देखो नेचर उनको दिया है, एल्कलाइन वाटर ऑफ कैंजन वाटर इसको कहते हैं, कैंजन इज द रिवर ऑफ कोरिया। तो जो अल्कलाइन वाटर है, एंड दे गेट फ्रेशी फूड, दे डिपेंड ऑन नेचर, मेडिसिनल प्रॉपर्टी ऑफ नेचर, एंड दैट प्रोवाइड्स द मोस्ट ब्यूटीफुल पर्सन और हाई लॉन्जिविटी। This is, you see, nature based on the importance of medicinal plants. Two important treatises they were documented from our country, Charak Samhita and Susru Samhita. Susru Samhita was written on the bank of river Ganga at Kashi. बनारस के बैंक में गंगा के किनारे सुश्रुत संहिता लिखा गया। एंड इन दोज डेज सुश्रुत यूज 21 टाइप ऑफ माइनर इंस्ट्रूमेंट्स फॉर द ऑपरेशन ऑफ आई, उसको फादर ऑफ सर्जरी कहा जाता है। भारत विश्व गुरु था। क्यों विश्व गुरु था भारत? इसीलिए था कि हमने आयुर्वेद को डॉक्यूमेंट किया था, और इसके कारण अशोक के बौद्ध धर्म से चारों तरफ फैला। सुकरात जब भारत पर आक्रमण करने, सिकंदर जब भारत पर आक्रमण करने आ रहा था, उसने अपने गुरु सुकरात से पूछा, इंडिया इस सोने का चिड़िया, जहां डाल डाल पर सोने की चिड़िया करती है बसेरा, मेनी सेकेंडरी मेटाबोलाइट्स इन द डाल ऑफ इंडियन प्लांट्स। तो उसने कहा कि कितना टन सोना लेकर के मैं आऊंगा गुरुजी। सुकरात कहा, आओ सोना वोना मत लेकर के आना, तुम क्या कहोगे? कि कहा सिकंदर सुकरात से, क्या तुझको लाऊंगा विजय डंका बजाकर हिंद से जब लौट आऊंगा। कहा सुकरात, ए राजन, विजय करके तू जब आना तो मेरे वास्ते भारत से एक ज्ञानी गुरु लाना। आयुर्वेदाचार्य को चाहता था। दिस इज यू सी इंपॉर्टेंस ऑफ इंडियन मेडिसिनल प्लांट्स। आज हम एक पौधे के पत्ती पर टुडे वी डू रिसर्च, एक्टिव प्रिंसिपल आइसोलेट एंड थेसिस इज सबमिटेड। आवर ग्रेट सेंस 10 रूट्स ऑफ 10 ट्रीज, 10 रूट, 10 पौधों के रूट के मेडिसिनल प्रॉपर्टी को जाने, 10 मूला रिस्ट बनाए, एंड दैट इज नॉट इफेक्टिव अगेंस्ट 1 डिजीज, that is effective against 16 disease, and no resistance of microorganism reported against 10 मूला रिस्ट। Ayurveda, before Ayurveda we have been using tulsi का काढ़ा against microbial infestation. No microbial resistance has been reported against tulsi, decoction of tulsi का काढ़ा। So this is the importance of herbal products. It is very easy to formulate antibiotics, that is single medicine active principle based products. Our herbal products, च्यवनप्राश, that is formulated based on 42 components, 42 component को मिला करके। इस how difficult it is, and this is still used। च्यवन ऋषि को के लिए सोनी कुमारों ने बनाया था to against the stress, cold stress for our soldiers in Ladakh। So these are, you see, importance of herbal medicine, and 80% population of the world, they are behind the use of herbal products। कोई एंटीबायोटिक नहीं है कि दूसरे एंटीबायोटिक के एक्टिविटी को बढ़ा दे, कुछ नहीं है। बट इन आवर हर्बल प्रोडक्ट्स देयर आर प्रोडक्ट्स which enhance the activity of antibiotics, even antibiotics, and such products are known as bioenhancer. Like in the kali mirch there is a piperine, and this piperine has been reported 60% to enhance the activity of rifamycin, and this is reported, first report credit goes to India by Dr. C.K. et al., डायरेक्टर, एक्स डायरेक्टर, रीजनल रिसर्च लेबोरेटरी जम्मू। कितने पौधे हैं वंडरफुल अपने कंट्री में, देखिए कि दिस इज द प्लांट विच इज फाउंड इन द यूसी ए हॉट स्पॉट दैट वेस्टर्न घाट में पाया जाता है। बड़े बाटनी सर्वे करने पौधों का गए वहां पर, सब पुष्पांगदन। एक बार पहाड़ पर चढ़े, थक गए, आकर के गेस्ट हाउस में एसी रूम में बैठ गए। उन्होंने देखा कि दी लोकल ट्राइब्स जब चाहते हैं पहाड़ पर चढ़ जाते हैं, उतर जाते हैं, वो थकते नहीं है। उन्होंने उससे पूछा कि आप व्हाई यू आर नॉट टायर्ड, आई एम टायर्ड क्लाइंबिंग ऑन द हिल्स। उन सबों ने कहा कि वी यूज द ट्यूबरस रूट्स ऑफ दिस प्लांट, दिस इज आरोग्य पच्चा इन लोकल लैंग्वेज। He worked on this plant, identified the plant as Trichoepis zillainicus, and he formulated, got American patent in 2004. That is आरोग्य पच्चा। मतलब क्या है कि हमारा लड़का नीट की तैयारी कर रहा है, 8 बजे सो जा रहा है, इसको ये खिला दो 12 बजे तक पढ़ेगा, थकेगा नहीं। तो दिस इज द इंपॉर्टेंस ऑफ द प्लांट्स यू सी, एंड सच टाइप ऑफ वंडरफुल प्लांट्स, वंडरफुल फॉर्मुलेशंस नेचर हैज गिवन। 80% पॉपुलेशंस आर हैविंग बिलीफ इन हर्बल प्रोडक्ट्स। जब जब पेंडेमिक आता है हमारे मेडिसिनल प्लांट्स का इंपॉर्टेंस बढ़ जाता है, कुछ रहता नहीं है। कोरोना के समय साइंस डेवलप कर गया है, दूसरे फेज में अंत में वैक्सीन आया। उसके पहले हम लोग किस पर डिपेंड किए? पौधों पर डिपेंड किए। जब जब पेंडेमिक आया है तो क्या है, नेचर में बाहर से देखने से नहीं पता चलता है। द सेकेंडरी मेटाबोलाइट द प्लांट कंटेंस, जैसे कि याज्ञवल्क्य जी ने कहा है, आत्मा बा अरे दिष्टव्य श्रोतव्यो मंतव्यो निष्दास्विष्टव्य हो, माने कि आत्मा ही असली चीज है, उसको मनन करना चाहिए। द सेकेंडरी मेटाबोलाइट present in the plant, that is important, and in tropical flora that contains, because of the stress, many valuable secondary metabolites which are used in cure of plants। जब जब पेंडेमिक आया है 1374 से 1351 तक, बट बल्बोनिक प्लेग आता था, गांव के लोग खत्म हो जाते थे, कोई मेडिसिन नहीं था। दैट इज फर्स्ट पेंडेमिक जो कि रिपोर्ट हुआ है। और देखो ये सब लोग मरते चले जा रहे थे। उसी समय क्या था, गांव में कुछ बदमाश आदमी रहते हैं, ट्रेन का एक्सीडेंट हुआ, कुछ लोग मदद करेंगे पांडे जी, कुछ लोग घड़ी छीन लेंगे, ये होता है। तो उस प्लेग के समय क्या हुआ कि लोग मर रहे थे, लेकिन 4 लोगों का गैंग था, वो घर का सामान लूट लेते थे, जो मर जाते, उनको प्लेग नहीं होता था। क्यों प्लेग नहीं होता था? ये हमने जो लिखा है आप नेट पर डाल करके देख सकते हैं। दिस इज साइंटिफिक स्टोरी। 4 थीव्स विनेगर, वो क्या करते थे कि ये जो पौधे लिखे हुए हैं एंगेलिका, ये सब 4 पौधे, उनको क्रश करके सिरका में डाल करके अपने शरीर पर लगा लेते थे। दे आर फ्री फ्रॉम प्लेग। उसको लगाने के बाद उनको प्लेग नहीं होता था और घर के सामान का लूटपाट करते थे। 4 थीव विनेगर। बाद में देखा गया साइंटिफिकली कि दीज प्लांट्स हैव एंटीवायरल एक्टिविटी एंड फ्लू रिपेलेंट एक्टिविटी। ये है आफ्टर डॉक्यूमेंटेशन दीज मेडिसिनल प्लांट्स, इथनो मेडिसिनल प्लांट्स हैव बीन फाउंड साइंटिफिकली ट्रू। इसी तरह से स्मॉल पॉक्स that came and this insectivorous plant Serratia purpurea that was used against this pandemic, and later on when bioprospection was made this was found correct, it was having antiviral activity। हम कई बार सुनाए हैं, आप लोग नहीं है, उसको थोड़े में हम सुना दें कि पेरू में 1971 में मलेरिया आया था। गांव के लोग मलेरिया से मरते चले जा रहे थे। उसी पेरू में उसी किसी गांव में एक बुढ़ा आदमी था, वो सोचा कि सारा डॉलर हमारे ऊपर लगा देंगे। लड़के वो जंगल में गया मरने के लिए, लेकिन मर नहीं रहा था, कुछ खोज होने वाली थी। उसको बहुत प्यास लगी। 10, 12, 15 दिन बाद, 15 दिन बीत गया था। लड़के उसका सब बाल ओल करके क्रिया कर दिए थे, डॉलर वगैरह सब अपने नाम से करा लिए थे। वो घूम रहा था चारों तरफ। जब बाद में उसको प्यास लगी, उसने सोचा कि मलेरिया से तो बाद में मरेंगे, पानी से पहले मर जाएंगे। He saw a pond and he went to that pond. Bitterness was in the water of the plant, बदबू आ रहा था, but he was bound to drink the water of that pond। उस पौधे के पानी, उस पोखरे के पानी को उसने पिया, थकान लगा, सो गया। 3-4 बार पिया, मलेरिया गायब। मरने निकला था वो जिंदा बच गया, गांव लौट करके गया। जिनको मलेरिया हुआ था they went there and drank the water of that pond। ज्यादातर लोगों का मलेरिया खत्म हो जा रहा था। हाँ, तो देखो लोगों ने, अच्छा पेरू के राजा के वाइफ को भी मलेरिया हुआ था, उनका भी बड़ी तबीयत खराब था, उनको भी उस पोखरे के पानी को पिलाया गया, उनका भी मलेरिया खत्म हो गया। पेरू का राजा वाज वेरी प्लीज्ड और उसने सारे साइंटिस्ट को कहा, फिजिक्स, केमिस्ट्री, जूलॉजी, बॉटनी, ज्योतिष, कि बताओ इस पोखरे में क्या है जो कि मलेरिया को खत्म कर दे रहा है। एंड देयर वाज ए बॉटनिस्ट, उसने देखा पांडे जी जैसे कि दिस इज ए प्लांट, दिस इज ए न्यू प्लांट एंड द लीव्स and leaves of this plant is falling on this pokhara, or because of this there is bitterness, he discovered an anti-malarial plant, you see। And that was later you see from Rubiaceae family active principle was discovered। तो अगेंस्ट पेंडेमिक 1920 में 20 वाला साल बड़ा खतरनाक होता है। 2020, 1920, 1820, 1700 में पेंडेमिक वाला पीरियड होता है। 1920 में पेंडेमिक आया एंड द ओनियन वाज इफेक्टिव टू सेव द लाइव्स ऑफ डिफरेंट पीपल्स इन आवर कोरोना। सच टाइप ऑफ स्पाइसेस, 5 स्पाइसेस दैट बूस्ट आवर इम्यून सिस्टम, उस पर हम डिपेंड किए। अदरक, हल्दी, इन पर हम डिपेंड किए। एंड दिस प्लांट्स Actinospora cardifolia। तो फ्रेंड्स, दिस इज द इंपॉर्टेंस ऑफ द प्लांट्स, दिस इज द इंपॉर्टेंस ऑफ द बायोडाइवर्सिटी। है 2 तरह का रिसर्च होता है, इसको भी हम कई जगह बताए हैं। वन रिसर्च इज कि एनके दुबे का एच इंडेक्स कितना है, साइटेशन कितना है, ये केवल एनके दुबे जानते हैं और कोई नहीं जानता है। एक ये रिसर्च हुआ पेपर बेस्ड रिसर्च, जैसे कि पेपर स्पेसीज हम कहते हैं। दूसरा रिसर्च होता है चंद्रमा का खोज, मंगल ग्रह का खोज। एक ही साथ इसरो 100 सैटेलाइट को लॉन्च कर रहा है। सीबीएसई बोर्ड के लड़के सब देखेंगे। इनका That type of research is known as adventures in science, उसको कहते हैं। And people think in space technology or bioinformatics, they only provide adventures in science. In our plant also, there are many researches which are not less than Chandrama का खोज and मंगल ग्रह का खोज, like discovery of anti-malarial plant Cinchona officinalis, रबर जिसने कि हमको कल्चर होने को बताया, एज ए डायरेक्टिन, एज ए डायरेक्टा इंडिका, बायोडीजल प्लांट फ्रॉम जस्टोफा एल्बा। तो मेनी यू सी एडवेंचर्स हैव बीन रिपोर्टेड फ्रॉम आवर प्लांट साइंस आल्सो। पहले क्या था, आदि मानव था जमीन पर सोता था, कीड़े काटते थे उसको आया कि हम थोड़ा जमीन से ऊपर सो जाए, आज हम लोग गद्दा पर सोते हैं। तो कैसे गद्दा कंसेप्ट उसके दिमाग में आया कि उसने एक्सपीरियंस के आधार पर जमीन पर रखा, मिट्टी रखा। पहले मिट्टी से कीड़ा चढ़ते थे, मॉइस्चर कंटेंट कम हो जाता था, कीड़ा मर जाते थे। फिर उसने क्या किया, एक्सपीरियंस के आधार पर कैंफर रखा, कैंफर के पत्ती को रखना शुरू किया, कीड़े नहीं आते थे। तो इस तरह से हमारे पास क्या आया, बेड बनाने का जो आया था सिस्टम, इस तरह से शुरू हुआ कि हम पहले कैंफर के पत्ती पर सोते थे। बाद में क्या हुआ, वी saw the importance of camphor and camphor is used against the pillow, तकिया के नीचे कैंफर को रख देते हैं as a sleep inducer since ages, and that helps in curing the diseases like pneumonia, जिनको नींद नहीं आती है, and other disorders, and it has insecticidal activity। खटमल जिस खाट में हो वहां पर कैंफर रख देते हैं अगेंस्ट बेड बाइट, और तेज पत्ता लीफ अगेंस्ट द पिलो। दी ट्रेडिशनल प्लांट्स को, इथनो मेडिसिन को हम यूज करते हैं, एंड दे हैव बीन फाउंड टू हैव डिफरेंट टाइप्स ऑफ इंसेक्टिसाइडल एक्टिविटी। फ्रेंड्स, पहले क्या था, जब हम चावल गेहूं का खेती करना नहीं जानते थे, कुछ खाते तो थे पेट में, तो क्या खाते थे, क्या था, न्यूट्रास्यूटिकल का कंसेप्ट। वर्ड अब आया है, कंसेप्ट बहुत पहले से था कि इंडियन रीजन के लोग जो थे वो बथुआ की स्पेसीज चिनोपोडियम क्यूनोआ, उसके सीड को खाते थे, चावल के जगह पर उसके सीड को खाते थे। बाद में पता चला कि इट कंटेन्स मेनी लाइसिन, एसेंशियल अमीनो एसिड्स एंड मेथियोनिन, ऑल दीज, कैल्शियम, ये इसका मेडिसिनल प्रॉपर्टी हुआ। एंड लुकिंग इनटू द इंपॉर्टेंस ऑफ मेडिसिनल प्रॉपर्टी ऑफ दिस प्लांट, 7000 ईयर इसको हम यूज करते थे। नाउ इट इज यूज्ड एज सुपर फूड बाय CFTRI to combat malnutrition, and United Nations General Assembly declared the year, जैसे इस साल मिलेट ईयर है, declared the year 2013 as International Year of Quinoa। इन द सेम ईयर जब हम चाय नहीं बनाते थे, साल्विया हिस्पेनिया के लीफ को करते थे, and that contains omega-3 fatty acid which is medicinally important for our heart. So I wanted to say कि earlier days when science was not developed, based on our experience, we use different types of medicinal plants, जिसको इथोनोमेडिसिन कहते हैं, but when scientifically they are where you see analyzed, their activity was found to be true. But this, because of such importance, 80% population of the world, they are behind the use of herbal products. This was the first thrust of my lecture. The second thrust of my lecture is what is the position of India in the field of herbal products. India is a country of mega biodiversity rich countries, and amongst 36, 34 plus 2 marine, 36 hot spot of biodiversity, 4 are found in our country. And in our country you see, young botanist के लिए कहते हैं कि read Bentham and Hooker system, 10-15 मिनट में हम खत्म कर देंगे। We read, बताते हैं Bentham and Hooker system of classification. Bentham and Hooker belong to Royal Botanic Garden Kew. They thought to produce a system of, Hooker came to India. Why Hooker came to India? इंडिया के पौधों को क्यों सर्वे किया, चाइना चला जाता, बिकॉज इन द वर्ल्ड देयर आर 8 continents and 16 biogeographical zones, and in our country all the 16 biogeographical zones and 8 continents are found. So our country, the plants, they attracted many foreign scientists to work on this plant, and many you see plant based products, 10, 8 and 9 are found in our country. This is the important, 44%, 50% plants of India, कहीं नहीं सिनेरियो है, they are of medicinal property. अपने कंट्री का हम मेडिसिनल प्लांट को देखते हैं, बासमती राइस को खाते हैं, the underutilized plants of our country, जिसको हम नहीं जल्दी खाते हैं, they are not less than that धान बासमती rice. In basmati rice, this is you see kala joha rice found in northeast India. In basmati rice, 2 1 pyrrolene. This is the aromatic component. This is also found in kala johar rice, black rice जो नॉर्थ ईस्टर्न रीजन में है. But in basmati rice, these two compounds, omega-6 fatty acid and omega-3 fatty acids are not found. In kala johar rice जो नॉर्थ ईस्ट रीजन में है, जो अंडर यूटिलाइज्ड प्लांट है, the omega-6 fatty acid, omega-3 fatty acid, they are used for low blood pressure and high blood pressure. They are found in appropriate ratio. So I wanted to say, in our country, nature has given even the underutilized plants, they are also having medicinal property. इतनी खुशबू थी. असमीज रामायण में लिखा हुआ है कि कुंभकर्ण को जब इमरजेंसी में जगाना होता था तो रावण यही काला जोहा राइस उसको खिलाता था. तो you see, this is the importance of the plants of our country. Many spices are found. इनके लिए कई country लड़ी, fought together for the Indian spices. You see, they created their colony. So you see, this is the importance of plants in our country. Why India is having rich biodiversity? Earlier the whole globe was united at 2 points. One was Gondwana connection, another one was Laurasia connection. Later on, because of the activity of the earth, ये दोनों पॉइंट टूटे, 8 कॉन्टिनेंट बने. एंड दोज कंट्रीज, दोज प्लेसेज विच बिलोंग टू गोंडवाना कनेक्शन एक्सेप्ट चाइना, दे आर बायोडाइवर्सिटी रिच कंट्री. एंड इंडिया इज ऑल्सो बायोडाइवर्सिटी. देखिए जुड़ जाएगा, दोनों जुड़ जाएगा, अफ्रीका एंड साउथ अमेरिका. तो इंडिया बिलोंग्स टू गोंडवाना कनेक्शन. इट्स ओन द ओरिजिन टुक प्लेस फ्रॉम मेडागास्कर, दैट इज गोंडवाना कनेक्शन. एंड दिस गोंडवाना कनेक्शन, ये दोनों पौधे या तो मेडागास्कर में है नहीं तो इंडिया में है. दैट कनेक्ट्स, कनेक्टिंग लिंक. तो वी बिलोंग टू गोंडवाना कनेक्शन एंड वी आर बायोडाइवर्सिटी रिच कंट्री. ज्यादा हम नहीं बताएंगे. दीज आर सम इंपॉर्टेंट मेडिसिनल प्लांट्स विच आर फाउंड इन आवर कंट्री. दिस इज यू सी ए bio enhancer plant guruch, menispermaceae family, Tinospora cardifolia, and you see this is known as amrita. This is you see Carla fabriata, used fat burner, मोटापे को कम करता है, the formulation reducta. Such valuable plants are found. This you see aloe vera, you know very well. विदैनिया सोमनीफेरा, नागौर रीजन में शेखावत जी आए हैं, सर है त्रिवेदी साहब. जो कीमोटाइप, इलाइट कीमोटाइप पाया जाता है, दैट हैज एक्सेलेंट एक्टिविटी. मेथी नागौर का खाने में डालिए, मीट आसानी से गल जाएगा. तो यू सी दीज आर नोन एज इलाइट कीमोटाइप. राजस्थान का क्लाइमेट ऐसा है. तो दिस इज विधेनिया सोमनीफेरा. और दिस राउल्फिया मोर देन 21 एल्कलाइड्स आइसोलेटेड फ्रॉम राउल्फिया सर्पेंटाइना. गांधी जी जब अनशन कर रहे थे 1942 में, अंग्रेज उनका अनशन तोड़वाना चाहते थे, वो तोड़ नहीं रहे थे, तो उनको इसी महाराष्ट्र के जेल में जबरदस्ती बंद किए सब. वो जेल में अपने आत्मकथा में उन्होंने लिखा है अनशन तोड़ देंगे. गांधी जी वाज ए मैन ऑफ मेडिसिनल प्लांट्स. अपने धोती में राउल्फिया सर्पेंटाइना को बांध करके ले गए थे. दतवन करते थे, दातुन करते थे, राउल्फिया सर्पेंटाइना को खा लेते थे. अंग्रेज कितना भी प्रताड़ित करें उनके ऊपर, ट्रांकुलाइजर एक्टिविटी इसको कहते हैं. तो दिस प्लांट हैज ए ट्रांकुलाइजर एक्टिविटी. राउल्फिया सर्पेंटाइना, सब सराका अशोका. सारा वाज द वाइफ ऑफ लिनियस. लिनियस के वाइफ का नाम सारा था. शी वाज वेरी ब्यूटीफुल. और उसके नाम के आधार पर सारा का नाम पड़ा. एंड व्हेन इट ब्लूम्स इट इज वेरी ब्यूटीफुल. एंड फॉर द क्योर ऑफ गायनोकोलॉजिकल प्रॉब्लम्स ऑफ लेडी, दिस प्लांट इज स्टिल वेरी इंपॉर्टेंट. अशोका रिस्ट. This is a millet you know very well. So you see this is the importance of the Indian plants. This is you see opportunity for us. But you see there are some problems. क्या है कि today the biotechnology rich countries, America and Japan, they have less number of plants. What they do, they survey the plants of biodiversity rich country, but biotechnologically poor country वो आसानी से पेटेंट नहीं करा सकते हैं, और उनके पौधों को चोरी छिपे वो पेटेंट करा लेते हैं. एंड दिस इज नोन एज जेनेटिक डकैती, जीन रॉबिंग, बायोपायरेसी. इंडिया में भी देर आर मेनी प्लांट्स विच आर मेंशन्ड इन डिफरेंट टाइप्स ऑफ लैंग्वेजेस. तेलुगु, तमिल में, इंग्लिश में नहीं है. दे हैव मेडिसिनल प्रॉपर्टी एंड दीज प्लांट्स आर फाउंड इन नेबरिंग कंट्री आल्सो. तो देयर यू सी फियर ऑफ बायोपायरेसी अपने कंट्री में सबसे ज्यादा है. इसको हुकर ने भी लिखा है कि सिग्निफिकेंट फीचर ऑफ इंडियन फ्लोरा इज द कॉन्फ्लुएंस ऑफ फ्लोराज ऑफ सराउंडिंग कंट्री. ये उसने लिखा है. एग्जाम्पल है कैथरेंथस रोजियस, मेडागास्कर का पौधा है, विन ब्लास्टिन, विन क्रिस्टिन, एंटीयूसी, ए कैंसरस कंपाउंड्स, माइनर कंपोनेंट, दे वे फार्मुलेटेड बाय इली लिली कंपनी। पौधा मेडागास्कर का फार्मुलेट इली लिली कंपनी कर लिया। बायो इकोनॉमी की आज हम बात करते हैं, वो उसका ले रहा है, सोवरिन राइट खत्म हो गया जो 1992 में हुआ इस बायोपायरेसी के कारण। 5 times sweetener than sugar, pentadiplendra brasiana, the sweetening gene is usually formulated by European. These are examples of biopiracy. Appetite suppressor compound of this plant, Hudia gondwana, एक कालाहारी रीजन में मिलता है। एक पेटेंट, दीज आर एग्जाम्पल्स यू सी कि चोरी छिपे जो करते हैं। देर आर मेनी एग्जाम्पल्स ऑफ बायोपायरेसी। तो दिस इज ए ग्रेट थ्रेट फॉर इंडिया, दिस बायोपायरेसी। आवर गवर्नमेंट हैज रियलाइज्ड एंड टू सॉल्व दिस वी हैव टू कन्वर्ट बायोलॉजिकल वेल्थ इन द इकोनॉमिक वेल्थ। We have to bio prospect our bio resources. Either उसको हम ड्रग बना लें, मायो साहब बैठे हुए हैं, या किसी भी रूप में करें, जैसे कि असलीस मिर्चा पाया जाता है। बिटरनेस बहुत ज्यादा होती थी। when राजनाथ सिंह जी वाज डिफेंस मिनिस्टर, कश्मीर में गोली चलती थी, मानवता की बात आई, एंड दिस असलीस मिर्चा वाज यूज्ड एज ए रिपेलेंट। इसको हम लोग भूट जलो किया, इसको हम लोग यूज किए बिकॉज ऑफ बिटरनेस। तो कई तरह से हम कर सकते हैं, स्ट्रेटजी में कर सकते हैं। एंड CSIR हैज फॉर डेवलप्ड ट्रेडिशनल नॉलेज डिजिटल लाइब्रेरी इन February 9, 2009 उसने किया है। इट हैज डिजिटलाइज्ड मोस्ट ऑफ आवर ट्रेडिशनल नॉलेज amongst 5 languages so that the biopiracy gene robbing of the traditionally used plants of the India may be controlled, and that was you see that आज के जमाने में इन्वायरमेंटल इथिक्स खत्म हो रहा है। दैट इज अनदर पॉइंट। हमारा टाइम खत्म हो रहा है फ्रेंड्स। ये सस्टेनेबिलिटी का क्या मतलब होता है, देखिए कितना इंपॉर्टेंट है सर ये सस्टेनेबिलिटी। एक रावण राज्य था, एक राम राज्य था। रावण राज्य में क्या था कि सारे ग्रहों को रावण अपने पलंग में बांध करके रखा था। उसका ऑटो कंट्रोल था। जब वो कहता था हवा चलती थी, जब वो कहता था तो सब ग्रह उनके कंट्रोल में थे। हनुमान जी जब गए तो देखे उन लोगों को, बहुत दुखी हुए थे। तो धरती क्या थी सर, जिसमें धारण करने की क्षमता धरती में है, उसका सस्टेनेबिलिटी खत्म हो चुका था रावण राज्य में। वो त्राइमाम कर रही थी धरती, और देवताओं के पास गई कि अब हम अपनी ड्यूटी नहीं कर पाएंगे। सस्टेनेबिलिटी रावण राज्य में खत्म हो गया है, ग्रहों को उसने बस में कर लिया है। सारे देवता के पास गई, कोई मदद नहीं कर पा रहा था। वेश बदल करके गाय के वेश में गई, ऐसे जाता तो रावण जाने नहीं देता। सब देवता के पास वो गई, देवता लोग नहीं कर पा रहे थे। शंकर जी के पास गई, वो भी कुछ नहीं कर पा रहे थे। बाद में क्या हुआ कि देवता लोग ऑनलाइन जुटे। जो कोरोना में हम लोग ऑनलाइन पढ़ाना शुरू किए, सबसे पहले उस समय हुआ। ऑनलाइन वे लोग कनेक्ट हुए और आपस में बात किए कि रावण को कैसे मारा जाए। तो उस समय शंकर जी प्रेसिडेंट थे। ब्रह्मा जी कहे कि विष्णु लोक में क्षीर सागर में विष्णु भगवान है, वही रावण को मारेंगे। अपना पतरा देख करके वो बताए है। अब विष्णु भगवान को क्षीर सागर में सर कनेक्ट नहीं हो पा रहे थे, कनेक्शन गड़बड़ हो जा रहा था। तो शंकर जी कहे कि यहीं से प्रार्थना करो, हर व्यापक सर्वत्र समाना। और उन लोगों ने प्रार्थना किया। देखिए इसमें लिखा हुआ है, जय जय सुरनायक जन सुखदायक प्रणत पाल भगवंता गोद्विज हितकारी जय असुरारी सिंधु सुता प्रिय कंता पालन सुर धरनी। धरती के सस्टेनेबिलिटी को काम करने के लिए आप अवतार लीजिए। दिस इज यू सी इंपॉर्टेंस ऑफ सस्टेनेबिलिटी। धरती के सस्टेनेबिलिटी जो खत्म हो चुकी थी, उसको काम करने के लिए रामावतार हुआ। एंड राम राज्य व्हाट हैपेंस कि दिस सस्टेनेबिलिटी ऑफ द अर्थ दैट वाज रिस्टोर्ड। शशि संपन्न सदा रह धरणी। धरती की सस्टेनेबिलिटी रिस्टोर हो गई। देयर वाज नो वाटर पॉल्यूशन, एयर पॉल्यूशन। शीतल सुरभि पवन बह मंदा गुंजत अलिले चल मकरंदा। सरिता सकल, केवल गंगा जी ही नहीं, सरिता सकल बह बरबारी। शीतल अमर कंजन वाटर जैसे अल्कलाइन, शीतल अमल स्वाद सुखकारी। तो दिस इज सस्टेनेबिलिटी व्यू पॉइंट। दिस इज द डिफरेंस बिटवीन राम राज्य एंड रावण राज्य। एंड व्हाट हैपेंस बिकॉज ऑफ क्लाइमेट चेंज, सर लिखे है फ्लोरा, प्लांट पहले फूल जा रहा है, खत्म हो जा रहा है, फ्रूटिंग, तो ये सब चीजें आ रही हैं। and that climate change is affecting on the active principle of the plant. कम हो रहा है, ज्यादा हो रहा है. गेहूं में हम कर रहे हैं कि क्लाइमेट चेंज से टेम्परेचर बढ़ने से पैदावार कितनी घट, इसकी जरूरत है कि मेडिसिनल प्लांट में करें जिससे कि जो आयुर्वेदिक ड्रग है उस पर असर न पड़े. एंड इंडिया इज ए ट्रॉपिकल कंट्री. ड्यूरिंग हार्वेस्टेशन कलेक्शन वी कलेक्ट हर्बल ड्रग्स, बट that herbal drugs also contain different types of microorganism and that secrete mycotoxin. So this is another problem to our country, and many fungi they degrade the active principle of the herbal drugs. This is the example. So we have, you see, through different types we have to standardize our herbal drugs. And जो एलोपैथ में हो रहा है नैनो टेक्नोलॉजी, वी हैव टू यूज दिस नैनो टेक्नोलॉजी इन आवर आयुर्वेदिक प्रोडक्ट आल्सो, हर्बल प्रोडक्ट्स आल्सो. ये नैनो टेक्नोलॉजी कोई विदेश की टेक्नोलॉजी नहीं है, अपने कंट्री में बहुत पहले से यूज हो रहा है. आंख में लगाने के लिए सुरमा, जथा सुअंजल अंज दृग, साधक सिद्ध सुजान, महिमा देखत शैलबन भूषल भूर निदान. तो बहुत पहले से हम लोग नैनो टेक्नोलॉजी को यूज, सो मेनी एग्जाम्पल्स. लड़ाई में औरंगजेब के अगेंस्ट हम लोगों ने नैनो टेक्नोलॉजी को यूज किया था. तो वी हैव टू सी नो फॉर्मेशन ऑफ नैनो फॉर्मुलेशंस फॉर आवर आयुर्वेदिक प्रोडक्ट्स. एंड आई अप्रिशिएट मोदी जी, बी वोकल फॉर लोकल. आज जरूरत यह है कि हम लोग अपने बेस्ड ऑन द प्लांट प्रोडक्ट्स ऐसा ड्रग बनाएं जिसका कि ग्लोबल सिग्निफिकेंस हो, एंड दैट में बी वेरी मच यूजफुल इन आवर बायो इकोनॉमी, सेल्फ रिलियंट इंडिया, इथनो मेडिसिनल नॉलेज इन बूस्टिंग इकोनॉमी ऑफ आवर कंट्री. यू सी दिस इज इंपॉर्टेंट फॉर अस. एंड इन मेक इन इंडिया कैंपेन ऑफ मोदी जी, people think that make in India make in India campaign, creation of jobs, bioinformatics. Our, you see, agriculture and pharmaceuticals are the pillars of a country, and we can use our, you see, herbal products, an important component in make in India campaign. And मोदी जी, you see, इस बार उन्होंने कह ही दिया दशहरा के समय दिल्ली में जो सेलिब्रेट हो रहा था 2024 में, फॉलो वोकल फॉर लोकल. यही मतलब हुआ कि इथनो मेडिसिनल प्लांट्स फाउंड इन आवर इंडिया, वी शुड बी वोकल फॉर लोकल, इंक्लूडिंग मिलेट्स इन आवर डेली डाइट. देखिए, एजुकेटिंग फार्मर्स अबाउट नेचुरल फार्मिंग, बायो पेस्टिसाइड, botanical pesticides, you see. So this is the importance of our plants, this is the importance of our nature. And example is carotene for the enhancement of blood platelets in the blood. ये देखिए है, पहले हम लोग पत्तल में खाते थे, अब हम लोग स्टील की थाली में और प्लास्टिक की थाली में क्यों पत्तल में खाते थे? अल्मोड़ा में मिठाई मिलता है सिंगौरी, उसको पत्तल में ही लपेट करके बाउहीन या वैलाई के लीफ से खा लेते थे पत्तल के साथ. The पत्तल contains different types of camphorol and curacin flavonoid. ये कंसेप्ट था जो हम पत्तल में खाते थे. And that पत्तल provided during our dinner or lunch medicinal component secret होता था. ये हमारा कंसेप्ट, ओल्ड कंसेप्ट था, ओल्ड इज गोल्ड. क्या था? देखिए, आज हम एग्रीकल्चर में अफ्रीकन कंट्री यूज करते हैं, 2 पीपीटी और है शायद, है कि वहां पर स्ट्राइगा का इन्फेक्शन मेज और सारगम में बहुत ज्यादा होता है, और स्ट्राइगा का लार्वी जो होता है उसका सेल वाल जो होता है उस पर पेस्टिसाइड का असर नहीं पड़ता है. तो क्या करते हैं? 2 पौधों को खोजे, दैट इज पुश एंड पुल हाइपोथेसिस. कितना इकोनॉमिकल है कि एक पौधा स्ट्राइगा लार्वी को अट्रैक्ट करता है, दूसरा उसको रिपेल करता है. नेपियर ग्रास इज यूज्ड एज पुल क्रॉप, that enhances milk of the cattle also, and Desmodium uncinetum that is used as push crop, उसके लार्वी को. दिस पुश पुल हाइपोथेसिस, very you see economical hypothesis. Green technology in agriculture against the striga infestation of that is nematode. सर जानते हैं आप बाजरा एंड सारगम, तो ये यूज होता है. हाँ, और देखिए इंपॉर्टेंस पेड़ पौधों का क्या है, नेचर का क्या है सर, कि जहां मुर्गा बनता है डेंगू मच्छर नहीं जाता है. जहां मुर्गा काटा, बकरी के पास तो जाता है, मुर्गा जहां कटता है मच्छर उससे दूर भागता है, वहां जाता नहीं है. तो हमको करने की क्या? द मुर्गा कंटेन्स सम केमिकल्स, एरोमेटिक कंपाउंड्स, दीज आर नोन एज सेमियो केमिकल्स, बिहेवियर ऑल्टरिंग केमिकल्स उसको कहते हैं. मॉस्किटो को रिपेल करता है. ये इंपॉर्टेंस है उसका। तो जहां हम सोए हुए हैं, बड़ा सा हाल, मच्छर आ रहा है, वहीं कोने में एक मुर्गा रख दें, मच्छर आएगा नहीं और 5 बजे हमको जगाएगा भी। हाँ, ट्रेन में रखने की जरूरत नहीं है, सब लोग जाग जाएंगे। तो दिस इज यू सी कि दैट जस्ट टाइप ऑफ यू सी नेचुरल हाइपोथेसिस, वी हैव टू थिंक ऑन दिस कि the aromatic compounds, we have to work on this. Now you see my lecture is finished. गीता में श्री कृष्ण ने कहा है, जदा जदा ही धर्मस्य ग्लानिर्भवति भारत अभ्युत्थानम धर्मस्य तदात्मनम सृजाम्यहम्। जब जब बदमाश आते हैं, कंस आए चाहे रावण आए, किसी न किसी रूप में अर्जुन में अवतार लेता हूं। इसी तरह से ऊपर से धन्वंतरी कहते हैं, कैंसर अब आया है, टेक्सस बकेटा बहुत पहले से था, बिंका रोजिया बहुत पहले से था, कैरीपील अब आया है, दैट डेंगू कैरीपील, हम पपीता की बात नहीं कर रहे हैं, बहुत पहले से ये सब कंपाउंड थे। हैं तो धन्वंतरी ऊपर से कह रहे हैं, पृथ्वीवासियों घबराना मत, जब जब नई नई बीमारियों की अनुभूति हो, हमारे प्लांट बायोडाइवर्सिटी की तरफ देखना। हम ऐसे ऐसे पौधों को दिए हैं, उन नई नई बीमारियों को भी दूर करेगा। थैंक यू वेरी मच। खत्म जी।
थैंक यू सर। इतना सबको आपने पूरा रामायण महाभारत सब समझा दिया, जो नए जनरेशन को नहीं मालूम था। आई न्यू दैट इट्स गोना हैपन। और अभी आप स्टेज से ना उतरें क्योंकि बिरबल साहनी लेक्चर होना है। आप प्लीज
अच्छा अच्छा अच्छा
इसको हटा लीजिए। हाँ। इसको ले लीजिए प्लीज। नीचे से कौन है?
जरा पानी दे देना।
डॉक्टर साहब को पानी दीजिए। Now we are moving into the session 1 second lecture, that is the Birbal Sahani Medal Award lecture, which will be delivered by Professor Amita Pal. And I would request Dr. UC Lavania to kindly chair the session. Hello everyone. Before Professor Pal delivers her lecture, I would read the citation. Professor Birbal Sahni Medal 2023 of the Indian Botanical Society is awarded to Professor Amita Pal, Fellow Nasi, FASCT, formerly Professor of Plant Biology, Bose Institute, Calcutta, for her significant contributions in revealing molecular basis of. इसको आगे करिए। आपको स्लाइड दिया हुआ है ना, दूसरा स्लाइड दिया हुआ है ना, नो नो नो, जस्ट अ मिनट। Yes. And you can follow the next one. Yes. For her significant contributions in revealing molecular basis of MYMIV stress in Vijna Mungo, molecular insights of differential in vitro regeneration response of two cotyledons of Vijna radiata, molecular taxonomy of tropical bamboos, genetic enhancement of fiber quality, and biotechnology of medicinal plants. Her proactive indulgence in teaching genetics, genomics, proteomics, and biotechnology at the Bose Institute and also at Calcutta University, Nagarjuna University, and PG departments across Kolkata, and mentoring a large number of PhD scholars and postdoctoral fellows are commendable. An elected fellow of National Academy of Sciences India 2005 and West Bengal Academy of Science and Technology 2014, she received the prestigious INSA Young Scientist Award in 1979, FAO postdoctoral fellowship at Harvard University, besides host of other national and international recognitions, received Cash Award from Kothari Scientific Research Institute, Endowment Lecture Awards from West Bengal Academy of Sciences, Botanical Society of West Bengal, and Indian Botanical Society. At present, she is the president of Archana Sharma Foundation of Calcutta and editor of its scientific organ, The Nucleus Springer Nature. Thank you. Congratulations, ma'am. I would request President Dubey sir to kindly decorate her with the medal and the citation. May I request Dr. Lavania to kindly introduce the speaker.
Let me speak one or two lines. Friends, it is indeed a very great pleasure for me to say two lines about Professor Amita Pal. I have known her since 1976, when I joined the Department of Botany at University of Calcutta as a PhD student, as a teacher fellow. She was already known a distinguished scholar, a very prolific researcher, and that time it was being said that there is one person called Amita Pal, who among the scholars is familiar with most of the techniques of that time. She is my senior Didi, and I always take her advice in various ways. Presently, she is the president of Archana Sharma Foundation, Calcutta, and although she obtained her PhD with Professor A.K. Sharma, but she was more attached with Archana Ji. She always says this thing that I am more close to Archana than Sir. Although I know about Sir, I never shared with him, but he was a person who always had a very soft heart for her. Amrita, when I was there, that was a time when they started the INSA Young Science Awards, and she is perhaps one of the first 2 or 3 young INSA Young Science recipients in plant sciences when they started. So this was a very big achievement and a laurel to the Department of Botany, which has then become the Center of Advanced Study in Cell and Formation Research. She was awarded, I remember, for the work which she conducted on diascoria, on different genotypes, and trying to differentiate based on chromosome and ploidy levels the concentration of metabolites. That made the beginning of making so many studies for phytochemical of plants, giving a ploidy as a basis. Ever since, she has learned many techniques, and one of the pioneers in this country, after Professor Maheshwari's laboratory, so we started working on tissue culture at the University of Calcutta. And thereafter, she moved to Bose Institute, very prestigious institute, where she became, rose to the level of senior professor by designation, not just by seniority. And a very distinguished scholar, she was elected to the Fellowship Academy of Nasi, and she is also a fellow of West Bengal Academy of Sciences. You know that although it's the Academy of the State, but most of you know that when it comes to science, Bengal has the maximum representation in Science Academy of the country. So to be a fellow of West Bengal Academy of Sciences, and then she became the convener. Also handled the many projects for the Academy, advised many persons for the fellowships. I'm not going to speak about her scientific contributions. I know her personal abilities. She has been very kind, always very helpful, and very experienced person dealing with the scientific matters as well as editorial matters. Today we are running the general nucleus. She is our president. Although I am given the responsibility, but she is the main person, the main guiding force behind the general nucleus. And we have brought the result to a very high level. She has started many activities in the Department of Botany after retirement under the Foundation. And we are very happy that she is with us to honor the society by accepting our request to be a recipient of Bilusani Gold Medal. Didi will give a small talk, and I will invite her.
Thank you, Professor Lavanya, for your kind words. I am honored to accept this prestigious Birbal Shahani Medal Award. Perhaps this recognition is to acknowledge my dedication and passion that I have invested in the plant science research for the last about 50 years or more. I extend my heartfelt gratitude to everyone involved in the selection process of this esteemed honor. At the same time, I am grateful to the organizing secretary, Professor Varsha Nathur, and her team for this wonderful arrangement. Thank you all. So, Professor, can I have a... Professor Birbal Shahani is known as father of Indian paleontology. Actually, Professor Birbal Shahani started the paleontological or paleobotanical research in India for the first time being an Indian. Born in 1891, his lifespan was very small. He expired in 1949. During this short period, he has achieved many awards, recognitions, and many accolades. He did his DSc from the University of Cambridge, and he was a fellow of the Geological Society of Great Britain. He was the Fellow of Royal Society of London and FRS, and also he was the Founder Fellow of Indian National Science Academy, New Delhi. And I came to know that he was one of the founder members of Indian Botanical Society and registered the IBS. He was instrumental in registering this Indian Botanical Society in Lucknow in the year 1928. He did many remarkable work, which I cannot finish within this short time. So I have selected a few. He studied the anatomy and morphology of Paleozoic ferns. He reconstituted Williamsonia sewarddiana. This is in the textbook, and we have read about this before. He revised the fossil plant of Indian Gondwana Formation. I am not going into the details of anything, but Shahani was among the first to suggest a separate order of Taxalis within the conifer to contain Taxus, Torea, and Cephalotaxus. He founded a research institute in 1949, and the foundation stone was laid down by none other than the then Prime Minister Jawaharlal Nehru on the 3rd April 1949. But unfortunately, after 7 days, Professor Shahani died on 10th April 1949. Later, this institute was named as Birbal Shahani Institute of Paleobotany of Lucknow. The lives of, I read out the quotation from Henry Wadsworth Longfellow. The lives of great men remind us we can make our lives sublime and departing, leaving behind us the footprints of the sands of time. With these words, I will start my own work, our journey in bamboo research. Not the entire journey I'll mention here, a part of it which we are doing for last 2 decades. Identification to genetic enhancement of fiber resources. Actually, bamboo was previously known as the bamboo is for the poor man's timber. But it is no longer poor man's timber, it is now known as green gold. And the lignocellulosic biomass has several possibilities, and I am sure within the coming decades you will find the use of biofuels and other such very important thing. And as we have already heard from our chief guest this morning session, that bamboo is being used in their institute as for the improvement of the environment. It is a huge carbon sequestral, and because of this, it's a very fast growing tree. So this has the advantage because most of the trees are act as carbon sequestral. But this is the difference between the bamboo and other tree plant. And it is also known as, the bamboo is also known as the fiber and columns of bamboo are being used from cradle to coffin. So it's a very long use. Not only this is not only used in India, but also in other places. You know, I'm not going into the statistics of this. Oh, sorry. It's not progressing. No, just a minute. However, the major constraint of the bamboo is the identification of bamboo. It is being identified from the morphological character, mainly calm and calm-sheath characters, and these phenotypic traits are often challenged by environmental cues. Because Why we are studying from the calm and calm sheet character? Because the flowers take long time and they are monocarpic in nature. They flower once and then the entire plant dies. And for this extended sexual cycle, the flower characters are unavailable. So there is no other way but to study the morphology for the taxonomic classification or to study the genetic diversity. To overcome this problem, we have undertaken a combined approach using molecular markers along with the phenotypic traits to identify and phylogenetically characterize some of the tropical tree bamboo. Sorry. No, the previous one. In this cartoon, I am showing some of the tree bamboos, and in the left-hand corner, the first one is the tallest bamboo I have ever seen. You can see a three-storied building. It is in Kalimpong, and three-storied building, and the plant, the Dendrocalamus giganteus, is about 6 to 7 storied building height. Whereas in the right-hand corner, this is also Dendrocalamus strictus. This is another species of Dendrocalamus which is bushy in nature. And there are other characters. The next one, Bambusa striata, has striation, and the next one has the thorn, and it is very heavily branched at the lower part of the plant. And with such differential characters, next slide. Okay, I remember. So from this differential morphological traits, we have identified 15 different arm characters. And next. So we found that the diversity in calm sheet characters also. I'm not going into the details, but the different appendages like auricle, hairs, sheath, and ligule, we can differentiate them. Because of this difference, we can differentiate these calm characters and calm sheet characters. Here I have noted 17 key calm sheet characters. So 15 plus 17, 32 key morphological descriptors. With these key morphological descriptors, we have drawn the dendrogram, the morphometric dendrogram, using the neighbor joining algorithm. But we are not very happy. Don't know why I'm moving. We are not very happy with such dendrogram because you can see there is a two clade for the bambusa itself. So it was not correct. As I mentioned, that such phenotypic traits are often influenced by the environmental cues or environmental influences. You can see the diversity in the calm characters in the 4 Bambusa tulda. Now you may ask, how do you differentiate, how do you call this as Bambusa tulda, not the strata, because there is striation in the first two calm. But I'll show you in a moment how do we differentiate the species using the molecular markers. Here also similar thing, Bambusa balquar, different calm characters. So unavailability, it's moving. Don't know why I'm moving, okay. Unavailability of reproductive structure and unpredictability of vegetative traits for the identification and phylogenetic analysis demanded the application of molecular technique for high resolution and consensus. So first we started with RAPD marker. Most of you are familiar with. I'm not going into the detail, and from such profiles we have selected 244 polymorphic loci, and here we drawn the dendrogram, and we are happy with this topology because it is best fit with the Orenbarger classification, the lower right hand side, the Orenbarger classification in 2002, where Bambucini is a subtribe for these bambusa species. And here in the lower side, I have already shown the morphometric data analysis, which we are not happy with, but we are happy with the molecular data. Later, I'll show you in the next few slides with the ITS sequence. This is perhaps the best fit topology we found in the process. And here you can see in the top next, P. kurzi, pseudo Bambusa kurzi make a totally outgroup, which is very different from these bamboos. And the next group, you can see the Melucana bactifera. Melucana bactifera belongs to another subtribe, Melucani. So it is evident that they will form a different clade. So what I suggest, so we are happy with the molecular dendrogram rather than a morphometric dendrogram. Here I am showing that we have done the ITS study, that is the internal transcribed species, and internal transcribed species are 1 and 2 sequences, you know, are variable among the species, while 18 is rDNA ribosomal DNA, 5.8 is ribosomal DNA, and 26 is ribosomal DNA. They are very much conserved in most of the plant species. So it is very easy to design the primers from the conserved region and to reveal the ITS1 and 2 sequences. It can be easily done. But in case of bamboo, we couldn't use these primers just because the bamboo, most of the, I mentioned that we have used the tree bamboos, and most of the tree bamboos, they contain the endogenous fungal contamination. So if we use the primers, which are the custom made and the common primers, then the ITS1 and 2 sequence will be, from both there will be ambiguity because they may come out from fungus as well as the bamboo species we are working with. So by this method, and I cannot show you the primers because they are intellectual property under the protection of intellectual property rights. So here you can see by this method we have identified ITS1 and 2 sequences from 21 tree bamboo species, and we have deposited in NCBI. And as I have already shown, that this creates a very good topology which is best fit for and which complies with the taxonomic classification with the Orenbarger. So I conclude that dendrograms obtained from both the molecular markers are very similar to the taxonomic classification of Orenbarger, while the cluster pattern obtained from morphological markers are different. Thus, in circumstances where the full complement of morphological markers are absent or unobtainable, we have shown the validity of DNA markers, which are essentially the same to depict genetic diversity or relationship as compared to that obtained from the detailed morphological study. I said detailed because we have studied 32 key characters. I am quickly go through just to show you how they look like the structures of ITS1 and ITS2, the below ITS2, because from these ITS1 and 2, when we compare the 21 tropical bamboo species we found some conserved motifs. In case of ITS1, they are 35 nucleotides, a stretch of 35 nucleotides, and from the helix 2 of ITS1, while 50 nucleotides of helix 3 and helix 3 of ITS2, these are highly conserved. So now on, we can recommend that these can be used as barcode. So if you have a confusion, I will not mention in detail. We found the same trait and show the last one. Here we found a band in only 3 and 4 population, but none other population. And if please buy my words, that these two has high cellulose content. In other work we have shown that these two population has higher cellulose content. And here we also found in 2-3 population and band, which we have not chosen just because they are very small one, and always look for the big one. So we selected the higher one. Next, So similarly, like the previous work, we have collected from the other region also, and we have checked that it is only present in 3 and 4, and then we prepared the scar marker, and similar result has been found. Among the 20 other bamboo species, only it is present in 3 and 4 Bambusa balcoa, and here you can see the only 3-4 genotypes, the scar markers appear. Then we characterize in silico. I am going fast. That characterize in silico, and we found the homology of predicted amino acid sequence with the WRF of Bambusa balcoa with the putative rice japonica rice kinase, which is very important because in such developmental processes, most of the cases kinase plays a very good role. I'm not showing the upper, upstream work, which is possibly the primers of this WRF, which I am calling as kinase domain of Bambusa balcoa, and this full-length gene is 6848 pp long, which we retrieved by genome marking, and the cDNA of the same was retrieved by the RLM res. Next, so in the upper left hand corner, you can see this WRF has 3 exon and 2 intron, and the UTR region. I'm not going into the lower one. I'm going straight about the predicted amino acid, which was 1113 amino acid long, and the upper you can see the red highlighted region, that is the 6 LRR leucine rich repeat. I forgot to mention that in the previous, while we found that it has homology with the rice kinase, that is also leucine rich kinase, and here also we found the leucine rich region and the N terminal end, and at the C terminal end we found the kinase domain. And then this cDNA BB kinase, so far I am calling it kinase, was cloned in PET28A vector, and the E. coli were transformed with this clone vector, and then we isolated the pure protein from the cloned E. coli, and you can see the arrow, which is a purified protein and kinase protein, and the left line, it is the total protein, total soluble protein, and next to the left 2 lane 2 is the total protein in soluble fraction. And the lower left hand side, you can see the western blot of this purified protein. It is anti-His antibody, and it gives a pure band. So there is no discrepancy. That is, it is the BB kinase band. So since I know now on that it is a BB kinase, so we perform different biochemical, we studied different biochemical properties to establish that this is the kinase from the Bambusa balboa. And the first one is showing the preference of the substrate because kinase phosphorylates. So what are the substrate? Base substrate we found, the second one, casein. This first graph, second one, casein. And all these experiments were reconfirmed by the autoradiography, the lower panel. And the right hand side you can see, we checked the highest phosphorylation activity at 6 to 7.6 pH. And the lower left hand side you can see the highest phosphorylation activity with a wide range, that is 15 to 37 degrees centigrade, and the enzymes are using cofactors, and here the cation cofactors we have used because it is kinase. So from manganese and magnesium, we found that manganese gives the best result. Next. I can skip this. I can skip the left one also. That is the inhibitor effect. But the important is, I said so far I am calling this as kinase. But whether it is a serine threonine kinase or serine tyrosine kinase or threonine tyrosine kinase, that I have to prove. So we performed this experiment. This is little critical, so I am not going into the detail of it. But you can see only serine and threonine amino acid gives the band, so positive band. So we are sure now on that this is the serine threonine kinase. So BB stands for Bambusa balboa, K for kinase, and ST for serine and threonine. So we cloned the entire BBKST in a plant expression vector, that is Pcambia 1304, which has two selection markers. One is GUS, another is JFP, and we called it JS1304 because the scholar, you can see his photograph, he is Joyashish, so he put the name JS1304. And at this point of time, we had the entire transcript of BBKST, so we have cross-checked whether really it is present in Bambusa balboa or not. So with this experiment, you can see that in rhizome it is absent, but in stem it is maximum. It is also present in internodes, and it is present in low quantity in the leaves. And you can, in the lowermost panel, we are showing that it is present maximum in I10, means internode 10, I8, it is more in I8, but maximum in I10, and it started from I2, I5, etc. Again, you have to buy my words, I am not showing those experiments. What we did with confocal microscopy to show that which stage, as I mentioned, that at this stage fiber was maximum or the mature at I10, that means in internode 10, and maximum fiber generates in the internode 8. So we performed this experiment, and it goes well with our concept that at the mature, up to the mature stage, it is maximum. As I mentioned previously, that I have not shown those experiments, but the 3 and 4 population where we got this band, they have the maximum cellulose content, that I will prove again. And in the transit experiment was done with the onion pea, bombarded with this clone fragment, and in the left hand side, in the bright field, and the green one is GFP fluorescence. Next. Oh, sorry. And again, so we transform this in the tobacco plant because bamboo transformation is very difficult. We did some work in tissue culture propagation, but from callus to differentiation is a hell of a job. So you can see the blue coloration for the GUS, and one faint in the right side below is a non-transformed plant, tobacco plant. So our transformation experiment was successful, and in the upper hand side you can see the GFP fluorescence in confocal microscope, and the nucleus also stained. That means the nucleus, it is entered, our construct has entered in the nucleus as well. And in the lower panel you can see the copy number. This is a single copy, which is the ideal situation in any transformation work. Again, again in the right hand side you can see the transform plants in different stages. This is the transgenic one, means T02 we have taken the seeds and then we again generate the plant. So these are the T1 plants, and the left hand side this is the wild plant, and you can see the D, tensile strength. They are erect, but they are bent in the control or non-transformed plant, and whereas this strong and erectness is due to the tensile strength and flexibility coefficient, whereas Runkel ratio is low in all the cases, and lower the Runkel ratio is better the end product. So it is very good type of fiber formation occurs with the BBKST. And you can see in most of the cases the flexibility coefficient and cellulose ratio are high. This also denotes the best quality. Here I am showing that the untransformed one is this is the untransformed one, and the other ones are all transformed. This work has been done with Congo red under confocal laser scanning microscope, and you can see the xylary elements has been increased in girth, and as well as the every that we have measured, the every xylem vessels are bigger compared to the untransformed one. And the right hand side, these are the only bright field photograph. Here you have to only concentrate on the cellulose content. The last one, this cellulose content was higher in all cases, as was evident from the CLSM, but here we have measured biochemically. So from all this work, the role of a serine threonine protein kinase in improving fiber quality and increasing the cellulose content has been established. Such findings emphasize the significance of molecular markers and BBKST gene in advancing bamboo fiber resources for sustainable paper, pulp, textile or timber related industrial products. Thank you very much. Oh no, I have something to show you. These are the players of this work. I did nothing, but the credit goes to all these. And you can see the Dr. Nishingo De of ILS Bhubaneswar. He also collaborates in the transformation part. And Professor Shubho Chaudhuri, he is a biochemist of Bush Institute. He helped us for this biochemical analysis portion. But these are the players, and CSIR, DBT and Bush Institute financially supported us for this work. Thank you. Yeah.
So friends, in science there are two types of researches. One is vertical and the horizontal. Both have its own importance. Sometimes we try to scan the area, the flora and other things, that the work is also exhaustive. On the other hand, there is vertical research. You go from one stage to another stage and next stage. As I told you in the beginning, I have known Amitadi for very beginning, that she was known to have perfected most of the experimental technique of that time, starting from counting chromosomes to looking for the variation of chromosome number, then phytochemicals, and then to other area. Giving one example of bamboo, she has tried to explain to achieve the ultimate goal with the application of various techniques, right from morphological identification to nucleotide identification, then development of vectors, then expression, all these things, leading to ultimate. This is a demonstration that how a person can do in his own lifetime to move with the pace of the progress of the area. See an example I knew about that. Many of us may not have really understood the whole scene. Those who are expert in one area may know it. I know only certain things, not everything. But I can simply say that this is the spectrum of areas he has covered. And work in an institute like Bose Institute is a very competitive area. You have survived with the colleagues. Perhaps the most prominent institute in the country, biological sciences, where she has used to the label and created a band of students, many of whom I know by name but not by face, because they are either associated with journal or with researchers or all these things. So I think you all agree with me that Amita Di has, by sharing her experience and expertise, she has opened the window how the science can grow, and we are grateful that she has come and accepted our the honor given by the society, and I think you all agree with me that is a well deserved honor. Share with me. Let's give a big hand to her. So we are really thankful to her. Thank you. Next one. Next one. Lunch.
Thank you, Amita Di. Thank you, Dubey sir. Thank you, Dr. Lavania. We are breaking for the lunch, and since all of you know that we are running behind the schedule, I would request everybody to, because this is half an hour lunch is scheduled here, so please do that and come back, because we are having very important lectures. Professor P. Maheshwari Medal Award lecture, Professor V. Puri Medal Award lecture, and Professor Y. S. Murthy Medal Award lecture is there. And I think all the three speakers have already loaded their lectures to the technical persons. And there is one more announcement that we might as well, if the time permits, there are two invited lectures to be held from Malaysia. So Dr. Stephen Manchester and Professor Jennifer N. So their time we have fixed for 6 o'clock. They say this will be too late for them. So this is for Women Botanist Award competition contestants and the Young Botanist Award competitions. If everything is on time, there will be held first, but otherwise we might as well shift it. But होगा आज ही आप लोगों का, but आधा घंटा shift करना पड़ सकता है. All right. Thank you very much. Please join for the lunch.
But the irrigation of the rice crop, which is a water intensive crop, is cannot be checked. So it contaminates the rice. It contaminates the rice as compared to other cereal crops from the wheat and grain, wheat and maize, 10 times more. I'll tell you what are the reasons why it takes 10 times more arsenic than other crops. Next slide. Okay. So if you look into the periodic table, it has atomic weight 33, and it has 3 isotopes, 73, 74, 75. And it occupies a position between metal and non-metal, therefore it is metallite. So there are several metallites like selenium and many else. So he said the two oxygen states which are most toxic to the human beings are the 3 and 5. So they are not only toxic to the humans, they are toxic to the plants and microbes as well. So the arsenic has been used as a poison, as a homicide since the 1st century. And there has been a population poisoning in Japan in 1998, in which 40 people were poisoned. And there are many episodes. And as I told you, in Patna, there is a hospital, and the cancer patients are because of their intake of the groundwater arsenic contamination. But so it causes the diabetes. Sorry, it's not working anyway. It causes many diseases such as diabetes, psoriasis, syphilis, skin diseases, ulcers, and joint diseases, and besides the cancer of kidney and bladder. But it causes different kinds of cancer, but at the same time, it's a very unique drug which is made from arsenite, which is a wonder drug for the blood cancer, leukemia. So that is known as arsenic trioxide, and it is known as Trisonex commercially, or ATO. Is a treatment for an acute myeloma leukemia called acute polymyelitic leukemia. So we know that arsenic is used in different kind of homeopathic drugs, and so it is poison, and it has many kind of medicinal properties also. So if I think the best use of a poison is to make its use in medicine. So as I tell you, it causes the various metabolic disorders, cancer and skin cancer, lungs and bladder. And it has been by WHO. It is known as declared the largest poisoning of the population in the history. Groundwater and rice have identified as the main sources of inorganic arsenic, which is contaminating the food chain. These are the different countries you can see, and India, Argentina, China, Pakistan, USA, Bangladesh, Nepal, Vietnam, Iran, South Africa, Japan, Germany, and France. So all these are contaminated. These are different contamination levels from India, 3.5 to 460 milligram per kg of the soil. So how soil gets contaminated? Because of the continuous irrigation, there is a deposition of the arsenic, and the soil in these areas becomes contaminated. So you can see there is a contamination of the arsenic in the red region shows the contamination of the soil in the West Bengal, Bihar, Uttar Pradesh. Uttar Pradesh, Balia is the most contaminated. It is as contaminated as the Bengal soil. Then Haryana, Delhi, Punjab, and Chhattisgarh. So this is just shows some of the patients suffering with the arsenicosis, and it was coming to in the chicken meat and was served in the McDonald's. And lastly, the US government realized that the chicken which was used in the McDonald's was also arsenic contaminated, so more strict guidelines were issued. And this is in the UP, Balia is the most contaminated. And again, the soil and water, the soil gets contaminated due to water irrigation and through drinking water intake and rice intake and intake of various vegetables grown in that area. It contaminates the food chain. So we had conducted, we wanted to select some rice lines which will be really resistant to arsenic contamination. And you can see that in the West Bengal, we had contained about 300 rice lines, and which are the popular germplasm, some of the traditional germplasm, some were from the Rice Research Institute in West Bengal, some were we got from the International Rice Research Institute in Philippines, and so they were irrigated with. So the idea was to select the rice germplasm which are resistant to uptake by arsenic. Why rice takes 10 times more arsenic than other cereals like wheat? It is because I think every one of us might have experienced the silica content in the paddy grains. If you push it in your fingers, it will start bleeding. It's because of the high silica content. So silicon pathway is highly expressed in rice as compared to other plants. It is 10 times more expressed in rice. So silicon uptake pathway, because this is again an part of the evolutionary pathway in which the rice want to protect. The rice plant wants to protect its survival against many pests and other kind of attacks, abiotic stresses. So again, that is a part of the evolutionary pathway. And you can see that here the red zones, and that shows the different blocks in the West Bengal area which are more contaminated than 50 microgram per liter of arsenic. Then this shade which is red and yellow is 10 to 50 microgram per liter arsenic. And of course there are less green and then more green. So the green areas are the safer. So based on the, so we identified these 7 rice lines: IIT 19226, Nanamoni, CN 16453, 16462, 16455, and CN 1795-2 CSAR NBRI was one of the variety which was based on the high yield, about 5.5 ton per hectare, and very low arsenic content. So how we decide that this variety is low arsenic, having low arsenic level? We can see that the WHO has given a provisional daily intake of 2 microgram of arsenic per kg body weight. You know that most of we take the drugs from the drug stores prescribed by many doctors is based on our body weight. So the 2 microgram per kg body weight is safe for the consumption. Considering that I'm sorry, I don't have a pointer, it's not functional. So considering that the average body weight of a person is 60 kg, women and men, and they eat about 420 gram of rice. So based on that calculation, any rice variety should not have a arsenic level more than 286 microgram per kg of the rice in the grain. So you can see that the green one, that is the variety we developed in collaboration with the rice Chinsura, and it contains those are grown in different arsenic contaminated regions. This Gaighata, Chinsura, Durgapur, Purvasri, Beldanga, and Birnagar. Birnagar is the border of the Bangladesh. So you can see that these are very highly contaminated regions. They have arsenic levels from 10 to 25 milligram per kg of the soil. And this variety doesn't accumulate more arsenic. It accumulates the arsenic level less than the permissible limit. The Prime Minister of the West Bengal, Mamata Banerjee, named this variety as Mukta Sri. Mukta Sri means this is free from the poison. That's not the case actually, but it takes permissible limits as prescribed by WHO. So Mukta Sri was developed by CSAR NBRI and Rice Chinsura, West Bengal, and it is safe for the cultivation, and it was a selection based on the yield and the low arsenic content of the grains. And I'm happy to tell you that variety was released in 2061 by the government of West Bengal as the safe arsenic variety for the cultivation in the arsenic contaminated region of West Bengal. And in 2009, the Central Variety Lease Committee of the ICR Delhi, they also approved as a variety. So recently I was discussing with one of my colleagues working in the West Bengal. So informed me that more than 300 farmers are growing this variety in the arsenic contaminated regions. So it is some kind of basmati type, and you can see how it looks. And this rice variety is suitable for cultivation in arsenic-prone agriculture fields of the country. So there is Muktasri is the name. So a very common feature of the rice is it is consumed by more than 50% of the population globally. So the arsenic accumulation leads to decline of some of the essential nutrients like iron, zinc, copper, and manganese and selenium. So you can see that this is cultivated in the Boro season, the summer season, where at that time we cultivate wheat in UP and many other places, and the other is the Kharif, those are the wetland season. Look at the yellow bars. This is a reference variety. It accumulates very high arsenic level. So in the field trial, we always use a variety which accumulates very high level. So that to see that your experiments are working well. So you can see that variety, it takes very less arsenic. The second number, that is called the Muktasri, the number second, at the different 9 different arsenic contaminated places. So you can see that it accumulates very less arsenic. At the same time, you can see the right hand in the lower panel, and you can see that the variety Muktasri and many other varieties, low arsenic content varieties, they have high level of iron. So you know that in those regions, the children and the women, pregnant women especially, they become anemic due to low iron. So that way, in fact, such a variety will help in getting more iron. So we had done some, try to do some molecular approach also in that, and we found a plant, a arsenic accumulator plant, Ceratophyllum demersum. It grows, it's just like hydrilla. Many of us may have looked it, and it detoxifies arsenic by binding to a kind of peptides known as phytoglycan peptides. They are the polymers of glutathione. So gamma globulin 16 units are repeated n times, and the value of n may be from 2 to 11. So you can see these plants inside lot of phytoglycans to detoxify arsenic. So as Professor Vaidya was mentioning that we need phytoremediated plants also. So we transformed some of the tobacco lines, and you can see that this is the wild type tomato lines and the transgenic lines, and they accumulate a lot of arsenic, and we use the CPMV promoter, so that's a universal promoter. So this is a small gene. It was 1257 base pair long, and it contained about 503 amino acid residues, the protein, phytoglycan synthase protein from Ceratophyllum demersum. So you look right hand, up in the right hand, that is Ceratophyllum demersum plant. It's an angiosperm. So you can see the red spots, they are the flowers. So from that, this phytoglycan synthase gene has been cloned, and the rice transformed, and this species synthase gene was used, and we have identified 7 positive lines which were positive for this enzyme, and you can see that the arsenic level in one of the lines, PCS3, was very low. It's more than, if you see the control, they are accumulating more arsenic, and the PCS3 is accumulating 50% less arsenic. And this will show you that the low arsenic level in the grains was due to high arsenic level in the root. So just 327.3 in the root, and in the grain about 50%, about 60% inhibition. So you can see this PCS3 line was performing well, So one of our students, she works in the Gandhinagar. She did it for her PhD. So there are more number of tillers and more number of panicles, and the weight of the 1,000 seeds was much higher in the transgenic line as compared to wild type lines. Next. So just to see how these some other enzymes like arsenite methyl transferase, which you know, the arsenite has 3 methyl groups, hydroxyl groups, and if you methylate those hydroxyl groups and convert into methyl groups, it is trimethyl arsine. So the trimethyl arsine, this pathway is called Challenger pathway, and trimethyl arsine is a volatile compound, so it has very reduced toxicity or almost insignificant toxicity. So we made this transgenic Arabidopsis, and you can see that these are the small mass residue. They are the non-transgenic lines, and those the big bars, they represent the transgenic lines, and you can see of 50 times more arsenic volatilization potential in the transgenic line. We tried to use this advantage of this Challenger pathway using arsenic methyl transferase, and we transformed the rice plant also, and this work was done by Verma et al. So our student, he was recently working in Israel, but because of the current problem in Israel, he has come back to India. So you can see that these transgenic lines, which were, and this arsenic methyl transferase gene was again 1150 base pair long, and this we had isolated from a arsenic resistant fungus from the West Bengal area, which was arsenic contaminated. The name of the fungus is Westraticella ornatiaca, and from this we could isolate this. We could clone and characterize the arsenic methyl transferase. We transformed the rice plant also, and you can see that the black, the arsenic accumulation, the root and shoot of these arsenic methyl transferase transgenic plants, and you can see that you look here in the right side, arsenic contamination in the roots. You look at the unpolished and polished seeds, and so the upper one is the arsenite, and the lower one is arsenate, and you can see in both the arsenic contamination is reduced more than 50 times. So we use another approach in which we use selenium, which is again which is called an antidote to arsenic toxicity. So selenium is very important, very interesting in the sense that if somebody has got arsenic contamination, if in its blood and tissues, the selenium level you can increase, it will help in excretion of arsenic through the urine. So that way it is very interesting. At the same time, selenium is a trace element. We all require 90 micrograms selenium per day. And you know, in the this is good for hair and skin, and this kind of selenium is essential. You can see in the medical stores, you can have selenium and shampoos. So there's people are using for the good hair growth. So it is an essential element, and many of us you may be knowing that it is a cofactor of some of the enzymes. For example, glutathione peroxidase. So selenium work as a cofactor for that. So selenium prohibits the uptake of arsenic by sharing a arsenic uptake pathway, and you can see that in the root and shoot, and also in the grains, the selenium is reducing the arsenic content. The selenium is reducing arsenic content in the laboratory is also demonstrated under field conditions. See, look at the right hand panel. So the selenium prohibits the arsenic accumulation in the root and shoot. So there are 93 varieties of rice, and you can see this the red peak shows those varieties which are increasing the arsenic accumulation, and you can see the reverse trend in the green graph. So they are accumulating less the high arsenic accumulating lines are have reduced the selenium uptake and vice versa. And we also require selenium, as I told you. So we always require a low arsenic or the safer variety, safer for the human consumption, which has more selenium uptake pathway. So we have done some experiments. One my postdoc is doing these experiments, and we'll see that there are different traits grown in the pots, and the arsenic accumulation in the has been estimated using ICPFS in root and shoot and grain, and gone there 50% inhibition of arsenic uptake in the shoot, 54% in the 50% the root, 54% the shoot, and 40% the grain. So I would like to summarize that skin selection and multi-locational field trials with low-grading arsenic cultivars, more than 300 cultivars. We could select a new rice variety, CS1794-2-CSR as Mukta for arsenic contaminated regions. This variety is being approved by the West Bengal government and the central government also. So, for the growing in the arsenic contaminated environments. And Ceratophyllum demersum and other plants synthesize significant amount of glutathione rich phytokeratin peptides. So while I was showing you the protein sequence of the enzyme phytokeratin synthase, which is an auto-regulated enzyme, when it gets arsenic or cadmium, the enzyme is activated. It synthesizes a lot of phytokeratin or glutathione rich peptides. And if you can see, if you have a speck, a glass, you can look inside the seed, inside the leaf or the root. What happens? You will see that all the arsenic which has been accumulated is made complex with these peptides. So they are not free. So only free arsenic can cause the toxicity, which has been, I told you mentioned that is used since prehistoric times as a homicide. So locally it is called Sankhya. So arsenic is Sankhya. It's a local name as it is known in our states. So the heterologous expression of Ceratophyllum demersum phytokeratin synthase leads to lower arsenic accumulation in the grain, to arsenic sequestration in the root. And transgenic rice plant is because of the volatilization potential. It has more than 50% reduction in the arsenic content in the rice grains. And this WARS-M, Western Digelar-Natka methyl transferase, has been isolated from an arsenic contaminated region of the West Bengal, from which this gene was cloned, characterized, and rice plants and Arabidopsis were transferred. So one thing important I want to tell you that because of this volatilization pathway, the arsenic, it is a microbial pathway, not arsenic methyl transferase is not present in the higher plants or the angiosperms. So we have to take this gene from the rice, and incidentally we took this gene from the fungus or bacteria, and we have to transform these plants. So transgenic rice plants expressing WARS-M exhibit high tolerance, high volatilization potential, and low accumulation in the rice seed, which are safer for the human consumption. At the same time, this pathway can be used for the phytoremediation of arsenic. So I told you some of the demonstration by Arabidopsis, which is a wheat plant, not a food plant. So what you require for phytoremediation of arsenic or any other metal? You require more accumulation in the wheat plant, like tobacco, Arabidopsis, or many others. At the same time, we will require the lowest accumulation of these toxic metalloid like arsenic in the edible plants. So you want a reverse trend. So ultimately, the aim of the phytoremediation is to clean the site so that edible crops can be grown. So that way, it can be advantageous. So selenium supplementation has been found to minimize arsenic accumulation, toxicity, and improve nutritional quality of rice. So as Professor Amita Paul mentioned in the earlier lecture, so this work was not done solely by me, simply I was associated with many workers who are my students, some are my colleagues. So I would like to acknowledge that Devashish Chakravarti, the chief scientist at NBRI, by some had advantage of publishing paper with Professor Lavania and Dr. Devashish. And Prabodh Kumar Trivedi was also associated with this arsenic work, because this arsenic work was started by in the regime of Dr. Tuli, and he, give stimulating help to start this work. Then Dr. Nandita Singh, the Pankaj Kumar Srivastava, Sanjay Dwivedi, Shekhar Malik, Dr. Seema Mishra, again she is my student, she is an associate professor in the Lucknow University. Devesh Shukla working in the CIMAP, Mudal Shukla, Priti Tripathi is a scientist in the Ministry of Environment and Forest. Manju Sri is working in Gandhinagar. Shikha Verma, Pankaj Verma, they work on the arsenate methyl transferase work in my collaboration with Devashish Chakravarti. Amit Kumar Richa Dave is a faculty in the MIT University. Garima Dikshit is a faculty in the Vivekananda Global University in Jaipur. Amit Pal Singh is working in Patanjali, and Pradyumna Kumar Singh, Reshut Chauhan, and Survasti, they are working as postdocs. Poonam Tiwari is working as a postdoc with me in the Nasi project. So we could not have done that, it was not possible to develop a variety without the collaborative work. So I'm grateful to have collaboration with Dr. Bijan Adhikari, Madhav Dhara and Mananjan Mandal, where the scientists with the government of West Bengal in the rice station at Chinchura. So thank you all for your patient hearing. And well, I will be happy to answer any questions. Okay.
Friends, we heard the informative lecture on the topic by Dr. R.D. Tripathi. He delivered his lecture as medal awardee of Professor Panchanan Maheshwari medal awardee. We have, you see, scarcity of time. I thank from core of my heart to Dr. R.D. Tripathi for delivering a nice lecture, and we shall now start the next session. I invite Professor Seshu Lavania, Secretary, Indian Botanical Society, for announcement. Hey, sir.
Now we are moving to for the next award lecture, that is Vipuri Medal Award lecture. Dr. A. Mao, Director Botanical Survey of India, Kolkata, will be delivering it. I would request Dr. Mao to please come to this, and Professor A.K. Pandey sir to kindly chair the session. I would read the citation. Professor Vishwambhar Puri Medal 2023 of the Indian Botanical Society is awarded to Dr. Ashiho Asosi Mao, Director Botanical Survey of India, Kolkata, West Bengal, for his outstanding contributions in floristics, ex situ conservation, plant tissue culture and ethnobotany of northeastern region of India. His practical understanding of declining phytodiversity has aided in the establishment of ex situ gene banks, plant tissue cultures, and botanical gardens, helping to serve rhododendrons, endangered medicinal plants, and orchids, specifically Paphiopedilum species. Further, he has immensely contributed in the documentation of ethnobotanical plants of Naga and Khasi tribal communities. As a Director of Botanical Survey of India, his contributions towards effective realization of its mandate and its outreach program, including support to the Indian Botanical Society and the botanical gardens across the country, are laudable. Congratulations, Doctor. Now I would request Professor Pandey to kindly introduce the speaker.
I am extremely happy to introduce my friend, Dr. A. Mao, whom I know him since 1990 when he joined Botanical Survey of India. And I came in close contact with Dr. Mao when a project was sanctioned by the Department of Biotechnology to me on the molecular taxonomy of genus Panax. And when I visited Shillong, Dr. Mao was heading that circle. And when I requested him about the collection of Panax, he himself accompanied me to Shillong Peak and other areas where Panax species was growing, and we collected the material, and also collectively published a paper on genus Panax in India. Since then, we have been actively involved in different research activities, and after joining as Director Botanical Survey of India, Dr. Mao decided that the BSI should complete the entire Flora of India project, and I am extremely happy to say at this moment that Flora of India project is now almost complete, and the credit goes to, of course, earlier directors, but the final product is coming from Dr. A. Mao. So let us congratulate him. Dr. Mao specializes in two different branches, which are both important. One is tissue culture. He is basically trained in plant tissue culture, and using that technique, he worked on Gymnocladus asamicus and other globally endangered plants and micropropagated those plants, and of course those plants are surviving in natural habitats. And also on the other hand, he has done outstanding work in the field of taxonomy and floristics. He has published over 170 research papers, guided 10 students for PhD program, written over a dozen books, and also he has been president of the East Himalayan Society of Spermatophyte Taxonomy, also Association of Plant Taxonomy and Society of Ethnobotanists, and he has been awarded, of course there is a long list of awards, but I will name a few, that he was awarded Arthur Hosier Award, then Sidge Prize, he was associate in Royal Botanic Gardens, Kew, Dr. S.K. Jain Medal. He was awarded DBT Overseas Fellowship to work in the field of tissue culture, Dr. T.N. Khushu Award of the Orchid Society. And at this juncture, I must say that besides doing excellent work on orchid, different orchid species, his work on rhododendrons of Northeast India, a pictorial guide is also, and when I was in Delhi University and before teaching or going to the field, I used to show the students a CD which was presented to me by Dr. Mao about the rhododendrons of Northeast India. And that was excellent work, and we really appreciate such type of work. Of course, there are many other publications to his credit. And another important thing that he has been supporting all the training programs and workshops. Of course, at Bhopal also I organized training program on traditional and molecular taxonomy, and he supported and not supported, but he himself came to all the way from Kolkata to Bhopal to interact with the participants, and participants were from different parts of the country. So he has also supported all the conferences, including the Indian Botanical Society conference and many other things. So I congratulate Dr. Mao on the honor which is being conferred upon him as Professor V. Puri Medal of the Indian Botanical Society. Thank you. Dr. Mao, please.
Good afternoon, everyone. First of all, I would like to thank Professor Pandey for the kind introduction. I'm not that, but he has said so many things good about me. So anyway, and also I would like to thank, I'm grateful to the IBS for honoring me with this Professor V. Puri's medal. And I'm really honored to be here. You see, maybe you may be wondering why I have chosen this topic. This is not my specialized or my area of work you may be considering, because from the introduction, Professor Pandey has said about tissue culture, floristic study, and all these things. But then, being the director of BSI, I thought maybe, though this is a very general topic, or most of you may be aware, but then I would like to share something. Why I would like to share is that, you see, today we talk about our floristic diversity or the richness of our flora and fauna in our country. We are very proud of that. We claim that we are mega diversity country, or the hotspot country, all those good things we are claiming. But what is the benefit? What is the benefit the common people are getting from this mega biodiversity? That is why I just wanted to just take you all through that for just for a few slides. Then why one reason why I'm taking up this is that, you see, today in Government of India or any funding agency, if you ask for any fund, they'll say, what is the benefit? What will be the societal benefit of your work? And that is what they'll say. You see, you know, whatever we are doing, research or any job, we are paid by the tax of the people, by tax. So that's why the government also expect from us something in return. So that is why I thought maybe let us discuss like this. You see, about me, of course, I joined in 1990 Botanical Survey of India, and I've been working mostly in the Northeast, and I came up only in 2018 to Calcutta. So this is it's actually primarily my work. And why I have shown rhododendron, because this was one of my main research work when I was in Northeast India, especially when I was in Arunachal Pradesh. Because rhododendron is a multi-million-dollar business in the Western country. But the interesting part is that they have hardly 10 species or less. All the species found in the world are from the China, India, or this Southeast Asian countries. But the only thing is, we have not harnessed or we have not utilized this rich horticulture potential or that thing. So that is also there. So, but when I work on this thing, I'm a member of the World Rhododendron Society expert committee. So I've been to 2-3 countries for meeting, and that's why it's a hot cake, like especially in Germany and other countries. So this is one thing. And then orchids, of course. I work on the conservation that lady sleeper orchid, Paphiopedilum species. If you happen to come to Shillong, you'll see we have cultivated the thing on the slope of the wall full. During this time, it will be seen thousands flowering. You will see like that. That is one of the things. So this, of course, this right now, this just wanted to tell about the flora of India. You see, what happened is that, frankly speaking, I'm telling you the thing. BSI has started this Flora of India project long time back ago. It's about in the 80s. But somehow we have not been able to do it. Everybody say, I'm working on this. Then somehow they retired and go. The manuscript is left like that. Then nobody touch it. And it was just like that. Then I thought, this is really bad. And then also, a lot of people were asking us whether BSI has completed or when is the Flora of India going to be completed. Then I felt very bad. So my first thing was that, I said, let us finish all this thing first. So all the scientists, I have engaged more than 100 people in this, the Flora of India to complete. So everyone was, we called for a meeting and everyone was assigned which volume their group will take up. So for each volume, there are 10, 20 people also, some of them. So they took up, and then we have put a target that within this, you have to finish the first draft and submit. Then what I did was that I brought a team of editorial who are among the scientists who are good in writing, who are good in editing from different regional center, and posted them in headquarters. So about 7, 8 of them are there. And their full-time job is just editing of this manuscript. They are checking. Then after that, they send back to the authors to correct it, incorporate. All those things are doing. We are going on that. So here, now today, I wanted to just talk about that bioeconomic aspect of this thing. Yes, you see, because the first part, I'll just talk about the rich floristic diversity of our country, little bit. Just as you all know, I don't have to say much. But in bioeconomics, so what is the potential? Just see the highlight of the rich floristic diversity. This is from Zico Valley, anemone flowering during this time of the year, October, November. If you go full, and that is really beautiful. So here, we say that India ranked as one of the 17 mega biodiversity country in the world. And this is, you see, again, we also say that I have already said all these things. So this is what is there. Everybody talk about it. I think all of you know about it. So I don't have to say that. Again here, you see, our country geographical area is, we are only 2% of the world. But we also contribute about 11% of the world's flora, which is very high. That's why we are considered as mega diversity country in the world. And here, you see, our region is also very rich in primitive plants, families. And for that reason, this Takrajan, when he visited and studied the flora of India, he found, because of these primitive families, he called cradle of flowering plants. The India, our region is like that. Why it's not moving? You see, again, India or our region is home to many wild relative of cultivated plant. And that's why you can see orchid, musa, bamboo, citrus, gingers, all these things you see that. And because of this, we will see, after saying this, he considered the region as Hindustan center of origin of cultivated plant. And that's why India or our region holds great significance from ecological and evolutionary point of view. You can see that. Again here, India is very rich in cultivated plant. About 30% of the world's cultivated plants originated here. And this is why Hindustani Center or the tropical India, we come under that Hindustani Center. You see here, rice, sugarcane, this fruit, all these things, many of these originated from India. No. Again, wild relative crop, I've just see, these are ways to get the best gene out of it to produce resistance to virus or disease or to improve the quality of the hybrid or cultivated plant. And that is why the foreigners in the morning, we have somebody has said about biopiracy. Why foreigners are interested about species? They are interested because they can get something, new genes, which can be utilized for some commercial purpose. And that is what they are doing. But our country, we still lack so much. We need to do it. So that is why what I'm telling to youngsters is that we should see that not just do research, but we should aim that what we are doing, it should become useful and it should be beneficial for the public. You see that? Okay. And also see India, we all know India is very rich in traditional knowledge and especially medicinal plant. Our Indian system of medicine. If you look at, see, like we are rich in Ayurveda, Yoga, Siddha, Unani, homeopathy, Anchi, all these things. And if you see from the chronological of Indian medicinal plant record, you can see it from 3000 to 1000 BC. That time it was, in the Vedic time, it was recorded 289. Then Samhita period, you can see that 650 was recorded. Then Nigundu period, 1814 was recorded. And in recent years, 7,000 plus, some says 8,000. So the number is debatable, or it's plus or minus only that. You can see that. And out of this, 960 plants are commercially traded, you can see in the market, and of that 178 is highly traded of the medicinal plant of that 960. So this is what we can see, that how rich we are. And then of course I just want to come here, BSI. Our government of India, after the reorganization, you see, I mean, what I mean to say is like, BSI was not established by India first. It was established by East India Company or the British, the Crown. So they were visionaries, or they were very far-sighted, because they have come to India not to help India, but they came to India to exploit and extract as much as possible from all parts of India and Far Eastern countries. And here, what they do was that they set up a botanic garden now what we have in Indian botanic garden. They were bringing plants from all parts of this, their territory or the colonies, and experimenting and growing, and once that is successful, they were sent in the different parts of India for cultivation. And also they were sending all the important plants, economic important plants to their colonies in all different parts of the world. And that is what they were doing. And also the main purpose was business or exploitation. And that is how systematically they survey and documented the plans of the country. And that's how in 1890 they set up. Then after the independence, again in 1954, it was a restructure because after independence or just before independence, during the Second World War, the department came to a halt, or there was not much activities. So then, but then the government of India, our first Prime Minister, Jawaharlal Nehru, realized this is very important and it should be restructured. And that's why he appointed Janki Amal, one of the first lady who is a botany in PhD. So she, like that. But our mandate is all these things. You can see that survey documentation of the plant well of the country. That is the, the gist. But then still, what we do is that all this, you can see the CBD articles which we cover under this thing. So now, you see the flora of India. During the British flora, in that, it was recorded 11,585. And then now, but in our revise, we are reaching 21,730. So which is double. And then of course that is what the figure which I put is 2021. So now every year in our plant discovery publication, we are publishing plant discovery. One book which publishes whatever publication of new species coming out from our country, not from BSI alone, all from universities, research institutes, all compiled together we used to put. And it is interesting to see that average around 300 plants are discovered every year. So that's why it's increasing every year. So that is why the figure is, it will be keep on increasing. Okay. So if we see that this is 21 statistic, if you can see the total is, it is coming to 21,730, but that is excluding the cultivated or the taxa or some doubtful ones we have not included. And you can see the dominant species of those families you can see in that. I think I'll go first. And here, if we see the plant discovery in the last 10 years, that is, I have just shown from 2010 to 9, you can see that, average of about 300 plants a year, you can see that, 283 taxa per year. The discovery is like that. That means still a lot of places to be explored, and you are getting that. So you can see that, but I think that I will try to avoid because shortage of time. So this is again, this is what now the figure adjustment here 22,000. That is 23 last year's of data. That is why it has come. So right now, it is expected that in India, where that our expectation is that algae maybe around 12,000 taxa may be there. But we have already reported about 700. Then fungi is one of the subjects where a lot of work is to be done. And then discoveries are made every time, and then a lot of work is to be done, but there's very few people working on fungi. And that is one of the biggest challenges we have. And if we do this, I think a lot of new discovery which are important for commercial purpose, like industries or all this thing. That's why it is one of the important group we need to do a lot. So these are some of the statistics which we are expecting like that. And so of course this again, whatever we have reported, I think that I'm not going to go in detail. Let us go fast. So like by economic potential, I'm just going to give few examples, only 2-3 examples only. This is that quacks, that's I took from Nagaland, just recent visit. Oh, it is not moving. Okay. You see, bioeconomic, the trend in India, if you look at this BIREX report, in 2015, the turnover of our country was just only $35.1 billion. And then you see that within 5 years, it has jumped to $70 billion. That shows that how much that potential, or how much that there's a, prospect for in bioeconomy. But that's why we are talking about plan. You have, in this seminar also, sustainable future. And this is what you can see from here also. You can see that. This is the thing that you can see that. See, of course, you can see our country's economy today is about 4 trillion. We are less than 4 trillion US dollar. Our GDP is. But then here, somebody has evaluated the biodiversity of India, natural services associated with biodiversity provided by India's forest alone is about more or less about 15 trillion US dollar. That is what this is. So that much you can see the and so now why people are focusing on bioeconomy. That I think you all know, so I don't have to tell about that. You see that. I'll not waste my time there. See now the feature that bioeconomy has been identified as a key component in global sustainable transaction. Bioeconomy offers a more productive and prosperous future for those dependent on agriculture system. You know, our country depends on agriculture system, and but yet the transition toward a modern bioeconomy depends on commitment from stakeholders enabling regulatory and trade environment and technology for success. And this is very important. If our country don't do this, we will not be able to achieve our, goal. It is not going. Yeah. Oh, some problem with. देखो. Move now, right? You see that if you look at the share of this bioeconomy today, what is at present? You see the 60% share of its total today is bioeconomy is from pharmaceutical group, biopharmaceutical. So this is what you can see for that is in the 19. That shows that lot of potential is there. Then these are the key segments you can see, but I think let me not. It is not very clear. देखो. Of course, the growth is, if you see, like say now, the projection of our government is that, see, by 2025 or that 2025 financial year, they have projected that 150 billion US dollar. You see, today is 23, and I have shown you that 2020 it was 70 billion US dollar. And now the government has projected by 2030 will reach 150 billion US dollar. That means, see, within 5 years span, it is doubling. So that is what the prospect of this this plant sciences or this animal sciences. So there's a bright future for our country also, if you look at. And these are the area where there's a lot of these entrepreneurs or innovators or this startup hubs there you can see like that. These are all from that. You see, this is what example of bioeconomic potential. This is taken from South Korea when I visited. This is ginseng market. There they sell the ginseng here, bring a wholesale market there like that. You can see how much business they make, $1 billion they make, I mean $1 million they make like that. And then you see here, this is one very simple and very common plan. Terrified or the funds. But here, in our country, I don't think anybody is making money out of this. But if you look at the US, see from fern, cut flower or foliage alone, every year, 150 to 300 million US dollar they utilized by these ferns. But in our country, I don't think we contribute anything of this. So this is why there's a lot of scope. Our people should reorient ourselves to it and make use of the bioresources. Otherwise, it is not going to last. Our biodiversity will not last. I have given here, say, we have so far recorded 1,157 in India, ferns allied. So here we have a lot of potential, but that our scientists need to work on it and make it happen. Okay, then again, orchid. Orchids we are very rich in orchids. And this is, see, in Thailand, they have about 1,129 orchid taxa in Thailand. But they are the largest cut flower exporter in the world. They export about 2.1 billion cut flower worth 63 billion US dollar to 148 countries. Also, life plant worth 17.8 million US dollar. This is a 2019 report. And again, here also we have not harnessed or we have not utilized our resources. That is how we are having sleeping well, or sleeping, what to say, like that, what we call a sleeping beauty. Or we are claiming that we have mega biodiversity, we are rich, all the things. But that is not going to help us by claiming. But we have to do something. And that is what I'm just trying this. This is taken from Thailand when I visited. Beautifully they have arranged. Oh, okay. So here, bamboo again. India is very rich in bamboo resources again. But again, here also, we have not tapped that full potential. If we are to compare China, you see, in China, the global market alone is about 68 billion US dollar in 2019, and China earns about 5 billion per year. And I don't think we earn $100 million or so. This is what you see. That is why India has about 172 bamboo taxas are recorded. So that is why you can see that a lot of potential is there. Here, that's why now, going forward, if we see India's rich biodiversity has tremendous potential, and we need to realize this potential. We should not only talk about it, but we must realize it. The biodiversity and bioeconomic program must benefit the people of this country, explore ways to reduce ecosystem stress caused by economic drivers. India is fairly strong in science and technology. And also we have a lot of manpower or trained manpower. So we have the potential, we can do it, but we need to reorient ourselves. Then it is time that science and biodiversity meet and come to the forefront. They must attract the attention of the society, the economy, the policy makers, and the people of this country. You see, this happened was that when you go to the remote villages or villages, you tell them, you conserve this, this is very important, this is medicinal plant. Immediately they'll ask, why? How can we sell this plant? How do we do this? But you are not able to tell them to do anything. You are not able to give any solution. You only tell that this is medicinal plant, this is medicinal plant, this is very important. But to the local, to the villages, that is of no value. And that is what it happened. So that is why it is very important that we should do. See that. See here, because of this, the biggest challenge we are facing today is that how to conserve our rich biodiversity is that we cannot, see, a hungry man cannot be asked to conserve biodiversity. First is he needs food or he needs something first. Then only he can think of conservation. But our country, we are facing the dilemma is this. The villages where the biodiversity is rich is found, most of the people are poor or they are not having what they need for their livelihood. So that is why, how can we tell them to conserve? So that is why here, we plant scientists should play a big role in making this potential into realize into the potential and make into product or make it to economic benefit for the local people. That is what I just. Oh no, it is gone again. I think I have finished, but last one. So yeah. So our strategies, actually, these are a lot of things. So with the policymakers, bureaucrats, all the things, we need to lobby. We need to work hard. All of this. Who are here, many of you are very influential. You should try your best. That we need to strengthen innovation capacity, human resources, then strengthening both national and international collaboration, increasing policy support, increasing capital investment. These are the things we need to do, prioritize and do that. You see, what is happening is that now our human resources are not good enough. We have a lot of graduates, but they are not skillful or they are not resourceful. That's why we need to improve that. Then another thing is international collaboration. Here, we have a very difficult time to collaborate with foreign agency or this thing. But we need to see that these are make more easier. You see, what has happened is that Chinese, they collaborate with anybody. They send their people everywhere in the world. And they learn and come back and they do everything. That's why you see that when you talk about transgenic plant, China produced the highest or the most in transgenic, anything to say about it. But we are saying that we have a lot of it done, and we cannot, and we lose all the chance, or we don't learn it. Because whether you like it or not, the Western world has a lot to offer to us, technologically or techniques or anything. They are much more advanced than us. So that is why we need to also capitalize that thing. And so that is what only. Thank you so much. Actually, my talk is not related to what Professor V. Puri being, what he has done, and also should have, I think in middle talk, I should have been speaking on my work, but I have not done that because I thought if I give a talk on my particular work, some people may be interested, but may not be interested also. So that's just general, I have gone that. And one thing which I would like to request all the youngsters, the researchers, is that what I have seen is today, many will say, I'm a molecular biologist, and you will not learn anything taxonomy or any other science. He say, I'm only molecular taxonomy, I'm doing this thing, biochemist. He never tried to learn any other thing. But if you see this, our old stalwarts or the professors, Professor V. Puri, Professor Birbal Sahni, they have specialized, you see, morphology, taxonomy, embryology, then molecular, that genetics. They are all rounder. But most of the youngsters today, they don't want to do that. And that is what I'm observing in BSI also. Many will say, I'm a trained ecologist. He will not bother to learn taxonomy, and he will be always try to do that. So, but that should not be the way. What I would like to tell or request all the youngsters is that learn as much possible so that you can fit anywhere. And also you can be, then that is better than just one area. Of course, if you are very good in that area, you may get a niche to get a job. But I think today, that jack of all trades are also very important. That is why we need to know that. Thank you so much.
Thank you, Dr. Mao, for a wonderful presentation. You want? Okay. So, okay. So,
yeah.
Somebody from Calcutta, they have sent me the photographs of your great banyan tree infected with pink fungus and ascomycete. I have identified tentatively as Hypoxylon multiformis. So, if the infection continues, I think there is a danger for the plant. So, as a director of that particular botanical garden and the institute, BSI, I think some care has to be taken. And you ask Dr. Kannadadas to study the life cycle and find out how best they can control this particular fungus.
That is why I told them. They didn't tell me that they have sent to you, but they were saying they have sent to.
But some other person I sent.
Okay, so actually what happened is that I have seen, they have shown me. That's why we say whatever maybe some fungicide also we have to spray. So we have taken up that for spray. Then also there's a mealybug, that white bug.
Yes.
So we have also taken out the precaution that we have. So I told them to buy a spray which can go very far distance. So they have purchased that spray which they can spray from distance. About it can go to 20 meters. Like that kind of thing. We have purchased all those things and we are doing that for both insect and for the thing.
Second thing is, I appreciate your concern for the lower plants, for the fungi, that we need a lot of work. But there are only two people who are working on this particular group. One is Kannadadas and another is Rashmi.
No. We have recruited now also another. Just recently, was scientist C.
But in Hyderabad unit there is
no, not in Hyderabad, but we have posted in Arunachal, because I wanted in Arunachal because there's a very rich in fungi. So then in Shillong, because in Shillong also mushroom is very popular, and every year you will see 1-2% die because of eating wild mushroom. So I told them the first thing is you go and document all the thing and find out, then make all this document properly so that people, it will help the local people. So that we have taken up.
The third one is in recent discoveries. I have seen that fungi have not been added, which are new fungi from reported in different papers in your discovery book.
Thank you.
Okay.
Dr. Mao has some books for you.
We wanted to give for your department.
From Botanical Survey of India, Dr. Mao has brought publications for the Department of Botany.
Very This is the checklist of Flora of India, checklist, 3 volumes, which I was showing the data, 21,730 is in that right there. And then that mangrove group, this year is a year of mangrove, government has given a lot of importance to mangrove ducks. conservation. So we have brought out the book on the mangrove of India. So it is very informative. And
Thank you, Dr. Mao. I think wherever Dr. Mao is going, he is presenting and donating books from Botanical Survey of India. Thank you so much. Now, as far as his award lecture is concerned, Dr. Mao has given a wonderful lecture on the bioeconomy and biodiversity, and citing many examples, starting from flora British India to cradle of angiosperms, especially primitive angiosperm by Takhtajan and Vavilov, and he cited many examples that there are a large number of species which are wild relatives of cultivated plants, and we have to focus our work on those species especially. And he mentioned about that 62% of economy is coming from biopharma, bioagri and biofuel. All those areas are there where there is a future in India. So with this brief my observation about his wonderful lecture, let us give him a big hand. As we are short of time, we have another lectures in line. Thank you. And I thank Professor Seshu Lavania for giving me this opportunity. Thank you.
Thank you, Professor Pandey. Dr. Sahab, aap niche chale gaye. Now we have a last lecture in this session, that is Dr. Neha Pandey, Professor Y.S. Murthy Gold Medal. And I would request Vimala to chair the session. I would like to read the citation. Professor Y.S. Murthy Medal 2023 of the Indian Botanical Society is awarded to Dr. Neha Pandey, CMP Degree College, a constituent PG College of the University of Allahabad, Prayagraj, for her significant contributions on the understanding of the molecular and epigenetic regulation of abiotic stress-induced secondary metabolite biosynthesis in medicinal plants. Her work established DNA methylation dynamics of promoters as a key mechanism behind gene overexpression and greater metabolite biosynthesis in Artemisia annua L. Congratulations, Neha. I would like Professor Vimla to kindly introduce the speaker.
A very good afternoon to the august audience, and thanks to the organizers for giving me the privilege to introduce Dr. Neha Pandey, a very outstanding, meritorious person for this particular award, Professor Y.S. Murthy Award. I was just going through her biodata, and it was so interesting to note that she had an outstanding career starting from Jaunpur to Lucknow to Banaras. She was trained during her graduation to PhD, and she earned UGC's Research Fellowship, Meritorious Research Fellowship Award for doing her research. And not only that, she earned several accolades during that period. From ISCA, she earned the Young Botanist Award. From Indian Botanical Society also, she was awarded as a young botanist. She also, that was Professor M.S. Swaminathan's award, which is turned to be Young Botanist Award. She also got Best Poster Presentation Award and National Postdoctoral Fellowship by DST SERB. She has joined as assistant professor through Higher Education Commission, CMP PG College, which is a constituent college of University of Allahabad. Her outstanding contributions, I will just read out a few, because she has a very high H index to her age, and also very good citations, 574, with the H index standing as 13 at this level. Her contributions are also very many. Out of these, I will just read one line. In one line, I will say that she has made outstanding contributions in the area of plant epigenetics, plant tissue culture, physiology and biochemistry, stress signaling, and secondary metabolism of medicinal plants. She has supervised up to now 20 MSc students for their dissertation, and 2 research scholars are taking up their PhD degree under her. They are being supervised by her. So with this small introduction, I would request Dr. Neha Pandey to present. Thank you.
Thank you, ma'am. A very good evening to all of you. First, I would like to pay my tribute to the great legendary botanist of the country, late Professor Y.S. Murthy sir. And I wish to thank the Indian Botanical Society for recommending me for this prestigious honor. I am greatly thankful to the society. Today, I am going to talk about influence of abiotic stress signals on secondary metabolites of medicinal plants, from transcriptomics to epigenetics. Now, human health has been inextricably linked to the use of herbal medicines for millennia, making natural medicinal resources one of the oldest contributions of nature to human well-being. Now, there is a trend. We all use PubMed for the literature search, and when I used medicinal plants as a keyword, it was observed that only in the recent years there has been a spike in medicinal plant research. Similarly, when I used plant secondary metabolites as a keyword, a similar trend was observed. This shows that in the past decades, despite the disproportionately large investments and research focus on synthetic drug discovery, approximately half of the globally approved pharmaceutical drugs have been derived from or inspired by natural products. The evolving commercial importance of secondary metabolites has in recent years resulted in a great interest in secondary metabolism, particularly in the possibility of altering the production of bioactive plant metabolites by various means. For example, there can be strategies for improved in-planta production of secondary metabolites, or one can make use of any heterologous system for enhanced production of secondary metabolites. Improved in-planta production includes manipulation of biosynthetic pathway like overexpression or downregulation of genes, and various plant tissue culture-based techniques like optimization of culture conditions, somaclonal variant production, and various elicitation strategies. And in heterologous system, secondary metabolite production in heterologous system can be performed via hairy root cultures or plant cell cultures. My focus has been on various plant tissue culture-based techniques and hairy root cultures. Recently, a new strategy has come into existence, and that is exploitation of epigenetic markers. And I have made use of abiotic stress signals in all the strategies adopted. For deciphering role of various abiotic stress signals, I have been using 3 highly valuable medicinal plants: Artemisia annua, which belongs to family Asteraceae and is well known for its antimalarial compound artemisinin. Next is Vidania somnifera and Vidania coagulans. They are commonly called as Ashwagandha and Rashwagandha respectively. And both the species of Vidania, they are known for a specific group of secondary metabolite called as vidinolites. Now today, I am going to talk about 3 major strategies for improved secondary metabolite production. The very first strategy that I am going to talk about is the somaclonal variant related, and is the development of high artemisinin yielding salt-tolerant somaclonal variant in Artemisia annua. Somaclonal variations are the genetic variations that occur in plants regenerated from any type of cell culture that can be exploited to improve the biochemical properties of medicinal plants. These variations appear to be more frequent in callus culture than in other type of cell culture. So what we did is we first generated callus from the leaf of Artemisia annua and taken for callus mutagenesis through gamma rays irradiation, followed by their growth on media containing NaCl for their selection against salt stress. A total of 13 salt-tolerant variants were identified and were characterized based on various morphological and molecular parameters. In addition, the first generation plants were screened up to 3rd generation for confirmation of the mutant character, if any. So this slide shows the wild type. The first one, WT, is the wild type, and the 13 somacolor variants that were identified. We have named them from, ASV1 to ASV13. A stands for Artemisia, S for salt, and V for variant. So 13 salt-tolerant variants were identified, and they were characterized morphologically based on 16 metric traits, majorly yield-related, like root length, plant height, number of branches, fresh weight, dry weight. And the most interesting part is the leaf artemisinin content, which was found to be almost 5 folds higher in one of the variant identified, and the variant was ASV12. Further, the molecular characterization was based on RAPD primers, and similarity matrix was calculated based on the presence and absence of bands. And based on the RAPD scores, a dendrogram was constructed, which showed ASV12 variant, the uppermost one, as a totally different variant. Further, various statistical analysis, including two-way correlation analysis and principal component analysis, showed that all the 13 salt-tolerant variants are better than the wild type in terms of yield. And here we can see the uppermost ASV12 variant has been found to be superior than all types of variants and the wild type in terms of total artemisinin yield. Because ASV12 variant was found to be superior among all, we further exposed them with various salt concentration and checked the expression of biosynthetic pathway genes. And it was found that in ASV12 variant, the expression of artemisinin biosynthetic pathway gene was better as compared to wild type. So there was, while doing the molecular characterization, there was one interesting observation that we used 12 primers for RAPD analysis, and one of them, OPE12, could amplify a specific band of approximately 650 base pair that was present in all high artemisinin yielding plant. Here in the right figure, we can see the 650 base pair band was present in all high artemisinin producing plants, while absent in low yielding plants. So hence this band, the OPE12 650 band, could be used as a marker to predict plants with high artemisinin content through development of sequence characterized amplified region marker, which is a SCAR marker. Though RAPD is one of the widely used techniques for genetic characterization of plant, conversion of RAPD markers into SCAR markers can greatly improve the stability and specificity of this marker technique and make it more accurate and authentic than RAPD, because SCAR markers are able to identify a single gene. Therefore, 650 base pair bands linked with high artemisinin content selected for the first SCAR marker development, and for that, the RAPD polymorphic products were recovered, re-amplified, purified, cloned, transformed, and sequenced. Here is the sequence, which we have submitted on NCBI. So this is how we have successfully developed a SCAR marker, which is 570 base pair in length, subtracting the length of primer that we used to amplify the region. For efficient identification of high artemisinin producing line. The authenticity of this developed SCAR marker was tested on 12 different plants, including parent F1 and F2 lines. We found that high artemisinin linked SCAR marker of about 570 base pair was consistently amplified in ASV12 and all high artemisinin yielding plant. The study was published in PCTOC. Now, talking about the second strategy that we used for high secondary metabolite production is the hairy root culture. Hairy roots are the transformed roots generated from the infection site of the wounded plants with Agrobacterium rhizogenes and are extensively studied as an effective alternative for the in vitro production of plant secondary metabolite. Why? Because they mirror the same phytochemical pattern of the corresponding root of the parent plant with higher growth rate and productivity. Several elicitation methods have been employed to enhance the accumulation of these compounds in the hairy root cultures for both a small and larger scale production. So, keeping these things in mind, we have developed a protocol for hairy root production in Artemisia annua. And with an idea to use some abiotic stress signals, we have exposed hairy roots with the two heavy metals, arsenic and cadmium, separately. And both the heavy metals were found to enhance the artemisinin content in hairy roots of Artemisia annua. Now, once inside the plants, these heavy metals, they are immobilized, chelated, and compartmentalized through metal binding ligands, which include various organic acids and polypeptide. One such polypeptide is phytogelatin. and is formed from glutathione by phytogelatin synthase. So what we did is we took phytogelatin synthase gene from a cyanobacteria, Anabaena PCC7120, and cloned it in the hairy roots of Artemisia annua. Now here we can see that for this study, we have 3 types of tissue: the normal roots, hairy roots, and the transformed hairy roots. It was found that transformed hairy roots expressing cyanobacterial PCS gene, they synthesized maximum amount of phytogelatin and hence accumulated highest amount of heavy metals, both arsenic and cadmium, showing that transformed hairy roots can act as an efficient system for phytoremediation as well. Now, in order to test the hypothesis that abiotic stress signals greatly influence the secondary metabolite production, we have tested artemisinin content in all the tissue samples, and it was found that in both hairy roots and transformed hairy roots, the artemisinin content was higher even in stress plants as compared to the normal roots and as compared to the control, which is attested by the higher expression of biosynthetic pathway genes in transformed hairy roots. So we can say that transformed hairy roots of Artemisia annua, they showed enhanced heavy metal tolerance and enhanced artemisinin production. So transformed hairy roots of Artemisia annua can act as an efficient tool for effective phytoremediation with added advantage of artemisinin extraction from hairy roots used for phytoremediation. The study was published in Plant Biotechnology Reports. Now, talking about the third strategy that we have adopted is abiotic elicitation in vitro raised plants. Abiotic elicitations are considered to be an important strategy towards improved in-planta production of secondary metabolites because plants respond to these stress signals by activating an array of mechanisms similar to the defense responses, affecting the plant metabolism and enhancing the synthesis of phytochemicals. Now, among all the abiotic stress signals, we have chosen ultraviolet B radiation and explored the potential of UVB as elicitor. Since the awareness of increasing UVB radiation due to stratospheric ozone layer depletion has triggered concern, this has been considered as a damaging environmental stress factor. And recent experimental approaches reflect UVB more as a regulatory rather than an impeding environmental factor. Furthermore, either regulatory or detrimental effects of UVB are purely species specific and usually depends on fluence rate and duration of exposure as well as interactions with the surrounding environmental factors. So for UVB elicitation study, we first choose Vidania, both the species of Vidania somnifera and coagulans. So Vidania, which belongs to family Solanaceae, is represented by approximately 61 species distributed around the world. However, only 2 of them, Somnifera and coagulans, they are medicinally and economically significant. There is an interesting observation that among the 2, Somnifera is relatively well distributed and possesses relatively higher germination rate, while coagulans has been declared critically endangered due to its low regeneration rate and higher exploitation rate. So for the for the UVB elicitation studies, we first generated a protocol for in vitro generation of Vidania, both the species, and through seed propagated seedlings, and we standardized the UVB dose based on various parameters, which I am not going to show here because these are preliminary data, and we have calculated LD50, which was found to be 2.8 watt per meter square, and we have provided in vitro raised seedlings with different short-term treatments like 1 hour, 2 hour, 3 hour, 4 hour to the plants. Now, it was found that in Vidania somnifera, 3 hours of UVB treatment can enhance vidaferin A content, which is one of the most important vidanolite found in the both Vidania and Vidania somnifera and coagulans. 35% increased vidaferin A content in Vidania somnifera, and UVB exposure for 3 hours enhanced 41% vidaferin A in Vidania coagulans, which was attested by the expression of their biosynthetic pathway gene SQS and CAS in 3 hour treated plant. UVB is largely known to induce damage. So we tested the oxidative damage in terms of H2O2 and superoxide radical. Here we can see that UVB caused oxidative damage in both the species, Vidania somnifera and coagulans. However, the extent of damage was more in Vidania coagulans, which was critically endangered, and Here we can see in terms of H2O2 and here the superoxide radicals, both are higher in coagulance as compared to somnifera. Further, we have also checked the antioxidative enzyme activities in both the species of Vidania, and it was found that the antioxidative enzymatic activity was better in somnifera as compared to the coagulants. Now, UVB is largely known to induce cell death and DNA damage. So, we have tested the expression of two genes from cell death pathway. This is BAX inhibitor 1, which is a conserved cell death suppressor, and DAD1, which is defender against cell death. Here we can see that the expression of BAX inhibitor 1 was increased up to 3R in somnifera. However, its expression was more or less constant in coagulants. However, the expression of defender against cell death, DAD1, was more in somnifera as compared to coagulants. Similarly, we have also checked the expression of two DNA repair enzyme genes, RUVB-like protein 1 and protein 2. They are known for their ATPase and helicase activity during DNA repair pathway. And here we can see the expression is higher in somnifera and coagulants. These data suggest the better adaptive strategies of somnifera as compared to coagulants. So we can see that short-term UVB exposure for 3R induced vidaffarin A content in both the species, and UVB radiation also caused oxidative damage in Vidania. However, the extent of damage was very low in Vidania somnifera as compared to Vidania coagulants. So Vidania somnifera also showed better defensive strategies against UVB exposure as compared to coagulants, and probably this can be one of the reasons why somnifera is better adapted to the changing environment. Now, talking about my UVB elicitation studies, we have also done it in Artemisia annua. So for this, we have generated a protocol for a in vitro establishment, acclimatization, UVB treatment, and their field transfer. And the protocol was published in PCTOC. Now, similar to the Vidania, UVB acted as elicitor for increased secondary metabolite production in Artemisia annua as well. Here we can see that 3R of treatment has significantly enhanced artemisinin content in Artemisia annua. Additionally, we have tested the total flavonoid contents. The flavonoids of Artemisia annua are also an important group of secondary metabolites. Why? Because there are several studies which have been published recently which shows that flavonoids of Artemisia annua have synergistic effects on artemisinin in both malaria and cancer therapies, and they also play a crucial role in UVB stress mitigation by acting as a sunscreen in plant cell. So we have tested 11 individual flavonoids in addition to the total flavonoids, and all of them were found to be significantly higher in UVB treated plant as compared to the control. Now, to test the authenticity of the enhanced artemisinin and flavonoid content, we have also checked the expression of biosynthetic pathway genes from both artemisinin biosynthetic pathway gene and flavonoid biosynthetic pathway genes, and all of them you can see all the genes, they are better expressed in UVB treated plant as compared to control. Now we again we have transferred the UVB treated plants to field and screened them till maturity, and again checked the artemisinin content at pre-flowering and flowering stage, and it was found that plant retained the induction signals and produced more artemisinin even at maturity. So this part of study shows that UVB radiation as elicitor has the potential to induce the artemisinin biosynthetic pathway genes that ultimately boost up artemisinin accumulation at early stage as well as at maturity. Now, the question is, if UVB is known for its damaging effect, it is important to know that whether short-term UVB radiation is affecting plant health. So we again tested the oxidative stress in terms of superoxide radical formation, and this figure shows that the maximum damage occurred after 4 hours of UV exposure. In addition, we have measured several antioxidative enzymes and protective secondary metabolites. All of them were found to be enhanced in UVB treated plant as compared to control. We have also done the secondary metabolite profiling through GCMS, which shows selective induction of protective metabolite, which may probably contributing to maintain plant health. plant health is largely governed by its efficiency to perform photosynthesis. So we have tested several photosynthetic parameters. Here we can see that although photosynthetic rate is decreased after UVB treatment, one parameter, the Fe by Fm ratio, which gives the maximum quantum yield, and a decline in this ratio is thought to be a very good indicator for oxidative stress. So this ratio was maintained up to 3 hours of UVB treatment. Here we can see in the left, genes encoding for light harvesting complex B1 protein reported constant expression. Genes encoding for Rubisco small and large subunit were upregulated to maintain the photosynthesis, and two genes, PSBO and PSBA, they are also overexpressed. Probably they are maintaining the photosystem 2 health by their overexpression. We have also checked Rubisco activity and did Western blotting of the Rubisco large subunit and D1 protein. So all these data suggest that UVB radiation exposure for up to 3 hours did not cause any direct damage to physiology, but adjusted secondary metabolism and metabolite accumulation as an effective acclimation mechanism to mitigate the adverse effects of radiation. And this study was published in Planta. Now, the data that I have shown, the one interesting observation was that we have provided UVB treatment to 21 days old seedling, and when we let them grow in the field and checked artemisinin content after several months, it was found that even at mature stages, plant retained the induction signals and produced more artemisinin as compared to control. So the question arises that what makes plants to retain the memory of elicitation signals for long? What tells a gene that please overexpress? I found my answer in epigenetic modifications like DNA methylation and histone modifications, which are important players of transcriptional switch and are major causes of retention of stress memory in plants. So I moved forward with DNA methylation, which is most commonly occurring epigenetic event and includes 3 types of methylated bases through post-replication mechanism. Of the 3, C5-methylcytosine is found in eukaryotes, which is much like a regular cytosine. Here we can see the cytosine and 5-methylcytosine. It is much like a regular cytosine, pairs with guanine in double-stranded DNA and is formed by the enzymatic addition of methyl group at carbon 5 position of cytosine residues. We conducted first the global DNA methylation analysis through restriction digestion of genomic DNA using methylation sensitive restriction enzymes. The pair of enzymes that we selected was MSP1 and HAPA2. These are isoshizomers, means they recognize the same restriction site. However, the restriction by HAPA2 is prevented by the presence of methylated cytosine. Here we can see that relatively more digested DNA in UVB treated plant as compared to control suggest UVB radiation mediated DNA demethylation in plants. This was further confirmed with another global DNA methylation analysis through thin layer chromatography based detection of total methylated cytosines in genomic DNA. We calculated a coefficient R, which gives the percentage of total methylated cytosine. Here we can see the lower density of the methylated cytosine spot and lower value of R. It suggests that UVB radiation has the potential to demethylate DNA in Artemisia. Further, we also analyzed site-specific DNA methylation through bisulfite sequencing. What we do in bisulfite sequencing, we take genomic DNA, we treat them with sodium bisulfite, and after treatment with sodium bisulfite, all the cytosines, they get converted to uracil. However, the methylated cytosine, they remain unaffected due to their structure. So here, with an example, we can see that if there is an unmethylated allele with a sequence ATCG CT, this after bisulfite treatment and PCR amplification will be converted to ATTGTT. However, if the cytosine is methylated, the sequence will remain the same. So we selected one gene each from artemisinin biosynthetic pathway and flavonoid biosynthetic pathway for bisulfite sequencing. From artemisinin biosynthetic pathway, we have selected DBR2, and from flavonoid, we have selected phenylalanine ammonia lyase, which is PAL, PAL, and we conducted bisulfite sequencing of their promoters to see the regulation of their expression. Talking about the DBR2, which is a key regulatory gene of artemisinin biosynthetic pathway and was found to be overexpressed under UVB exposure. We did bisulfite sequencing of DBR2 and we amplified the DBR2 promoter. We did bisulfite sequencing and the sequences obtained, they were aligned along with the standard sequence. Here we can see the alignment, sequence alignment. The first one is the standard sequence, the first 3 are the control replicates and the last 3 are the treated replicates. So this sequence alignment showed a total of 9 cytosine sites where UVB caused demethylation. In silico analysis of this sequence revealed that all the demethylated cytosine sites are the transcription factor binding sites, and a total of 7 transcription factor binding site was found to be demethylated in UVB treated plant as compared to control. All of them have roles in different abiotic stress management, including UVB. There is one Virki binding site, which is found to be a positive regulator of artemisinin biosynthesis. Now, Virki are the plant-specific transcription factors and specifically bind to W-box, a cis-acting DNA element found frequently in the promoters of defense-related genes. And role of Virki in the positive regulation of artemisinin biosynthesis has already been established. Now, the in silico studies revealed that this DBR2 promoter contains 2 Virki binding sites. Out of 2, one is active, where C is unmethylated, and the other is inactive, which is where C is methylated. And the UVB treatment has demethylated that methylated Virki sites. And the presence of 2 DNA binding sites or more for a transcription factor result in synergistic stimulation of gene expression. So this study shows that UVB mediated activation of additional Virki binding site in promoter may be the probable reason for overexpression of DBR2. And the study was published in Planta. Now, similar to DBR2, we conducted bisulfite sequencing of PAL1 promoter and found that UVB treatment has demethylated 5 cytosine residues in Artemisia annua PAL1 promoter, which in turn resulted in activation of several transcription factor binding sites, including MIB. MIB are the MIB transcription factors have key involvement in flavonoid biosynthetic pathway, and recently it has been shown to play regulatory roles in flavonoid biosynthesis of Artemisia as well. So, semi-quantitative and real-time PCR analysis showed this higher expression of MIB along with MIG and Virki in UVB treated plant as compared to control. Now further, at protein level expression, we isolated all the protein and conducted Western blot analysis for MIB1 protein, and it shows higher protein expression in UVB treated plant as compared to control. Now the in silico analysis of this MIB sequence revealed that they have DNA binding domains, and when we did molecular modeling, it was found that this MIB has physical interaction with PAL1 promoter, which again tested in gel with the help of electrophoretic mobility shift assay, which confirmed the physical interaction of Artemisia annua MIB with PAL1 promoter. So this further strengthened our observation that UVB mediated overexpression of MIB1 and subsequent cytosine demethylation of its binding site in PAL1 promoter may possibly be involved in overexpression of PAL1 gene and hence greater flavonoid production in Artemisia annua. The study was published again in Planta. Now, coming to the major conclusions of my study, we have adopted 3 strategies. From the very first strategy, we have developed a high artemisinin yielding salt-tolerant somaclonal variant in Artemisia annua. The study provides ample possibility of cultivation of ASV12 variant of Artemisia on saline soil for dual benefit, high artemisinin production and utilization of waste saline land. Further, in second strategy, we have developed high artemisinin producing transformed hairy roots expressing cyanobacterial PCS gene for efficient uptake of heavy metals in Artemisia. The study provides transformed hairy roots of Artemisia as an efficient tool for effective phytoremediation with added advantages of artemisinin extraction from hairy roots used for phytoremediation. From the third strategy, it can be shown that short-term UVB treatment can act as
a potential elicitor for secondary metabolite production in both Vidania and Artemisia. And this study is the first report that provides mechanistic evidence of epigenetic regulation of artemisinin and flavonoid biosynthesis under UVB radiation in Artemisia. The study also provides a platform for exploitation of genes regulating DNA methylation dynamics as a tool for improved secondary metabolite production. Thank you.
So all of you have just enjoyed a very scholarly talk on how UVB radiation can elicit, along with abiotic stresses, the secondary metabolite production, and she has worked it actually through transcriptomics, through epigenetics. She has worked it out from end to end. What she had promised in the beginning in her title, she has done all justice with that. So I'm sure you might have enjoyed it very well. So thank you, Neha Pandey, and congratulations again.
Thank you.
We are running behind the schedule. So, but there are two international speakers who are waiting to present their work. And because of this and the time constraint with them, because we and they do not match the time, we will be postponing right now Women Botanist and Young Botanist Award competitions, and we would be holding them after the two invited lectures. So the lecture time is not much, but still I would request Dr. Mao to kindly chair this session. And I would request him to please call first Jennifer Anne. Jennifer Anne, because she's in Malaysia, she has a problem. So after that, we will be continuing with the Women Botanist and Young Botanist sessions. I do not know, sir, what will happen to EC meeting. So,
Hello, good evening. We are fortunate that we have two foreign speakers. And the first one is Jennifer and Hari Krishna. She is a professor of genetics and molecular biology at the Faculty of Science, University of Malaysia, Kuala Lumpur. And her specialization or interest is transcriptomic, next generation sequencing, molecular biology, cell biology, genetics. Her international experience includes various programs, contribution, and participation in different countries for diverse fields of study. She currently holds 3 research grants and supervises 18 graduate students and is the author of several scientific publications, including 25 international journal papers, 6 books chapters, 3 patents, 5 proceeding papers, and over 50 abstracts. So I think as the time is short, we'll give the time to Miss Jennifer and Hari Krishna. So Jennifer, the time is yours.
Okay, thank you. Can you hear me?
Yeah, yes, we can hear you.
Can you please enable the screen sharing? I'm not able to share my slides. Are you able to see the slides?
Yes, you can go ahead.
Okay, I will move to slideshow. If the slides do not move, let me know. Okay, thank you very much. And especially thank you to the host for this kind invitation. And I will try to move the slides in time and keep going quickly because I know the other speaker has been waiting a while. So just to let you know where I'm from, I'm based in Malaysia, and the university is called University Malaya, and this is always some subject of confusion because why is it not University Malaysia? And that is because the university is in the peninsula of Malaya, and it is older than the country of Malaysia. So the university was formed in 1905, and Malaysia only became a country in 1957. We're based in Kuala Lumpur, and I'm speaking from the Research Center for Biotechnology and Agriculture, which was established in 2005. And we focus on mostly plant molecular biology and genetics, and we do have a group doing aquaculture as well. So my talk basis is the challenge to use fundamental plant research towards producing resilient crops for food security in the face of climate change. I now should be on a slide that shows my outline. Can I just check that you're seeing the slide with the outline? Are you able to view the slide with the outline?
Yes.
Okay, thanks. Just sometimes the slides don't change, so I want to make sure that's working. Okay, so this is the outline of my talk that I will explain why we focused on banana, why we're looking at the microRNA transcriptome, and then how we went on to study these two particular genes that are targets of microRNAs for use in stress tolerance. So this is a photograph that my collaborator from UK, Pat Heslop Harrison, took actually in India. So it's a celebration of the wide variety of bananas that are found across Asia. And Malaysia is actually right in the middle of the banana origin, which spreads all the way from India right across to Japan, and also right down to Northern Australia. And one thing that bananas have in common everywhere is actually a giant herb, but it's very sensitive to all kinds of abiotic stresses as well as many diseases. I'm going to focus on the abiotic stresses of salinity and water stress, and these are the main causes of crop losses in banana other than disease. And so in order to understand the how to maybe mitigate the stress, maybe to improve crops for stress resistance, we're looking at what are the fundamental biology underneath that. And this is because banana is actually a very important food security crop, and it's largely grown by the people who eat it in this Asian region where it's found. So we started with just defining what are the effects of salinity stress on the physiology before we went on to then look at what were the effects on the gene expression, and particularly looking at small RNAs. So this is what happens to bananas in stress. And we're working with the triploid A genome, which is the common dessert banana. And our variety in Malaysia is a Cavendish type called Barangan. And at different levels of seawater, 20%, 50%, up to 100% seawater, you can see obvious and very harmful effect of the seawater. So this is what happens every year in Malaysia in our coastal regions, particularly on our east coast, but now increasingly with climate change, we're getting flooding on our west coast as well. And what that will do is it will kill the plants, and certainly even if they are able to recover, if it's a mild flooding, they will have a very reduced yield. And we can quantify the effects in terms of the chlorophylls and carotenoids, and also in terms of things like the membrane damage by looking at MDA levels, the proline content indicating stress is going up, and also the ROS levels, hydrogen peroxide and superoxide, as markers indicating the stress. And to cut a long story short, what we find is that while they can tolerate very mild flooding for a short while, if they were to undergo severe flooding, that is lethal to bananas. So what effect is that having on the transcriptome, messenger RNA and small RNAs? So for this study, we were really focusing around the regulation by small RNAs, and particularly looking at microRNAs, which are gene regulators acting as post-transcriptional gene silencers. So what this means is that if a microRNA is present, the target that it is complementary to, that messenger RNA, will be reduced, and that protein that messenger RNA encodes is less expressed. And we can get an idea of that by combining a few different kinds of sequencing. So we can compare what happens to the RNA populations in bananas that are exposed to salinity stress with those that are not exposed to salinity stress. And then if we do RNA sequencing, we will be looking at the messenger RNAs, but also the precursors of microRNAs, which are longer and fall into that messenger RNA size. When we do small RNA sequencing, we can look at the mature microRNAs. These are around 20 to 24 nucleotides, mostly in length. They are cleaved from their precursors by RNA3s in the plants. And these short sequences then can be studied for their changes in expression. The third set we look at is parallel analysis of RNA ends, PARE, or also often referred to as degradome, because when a microRNA targets its messenger, it will bind in a complementary manner, and the cleavage will be in the center of the microRNA sequence. So there'll be a very distinct sequence signal at the RNA end, which tells us that it has been cleaved due to the action of a microRNA. So this allows us to map which microRNAs are actually working in the plant at the same time. So we use tissue culture plants. We develop the embryo, we develop callus going through from male flowers, and then we multiply the plants, and we actually treat them in vitro. So they're only looking at plant, not looking at soil RNA. And then we isolate RNA and put it through the different sequencing pipelines. And by comparing the plants that have been treated with salinity to those that have not, we can look at differences in gene expression. We can map the microRNAs to their messenger RNA targets and to the degradome ends, and then we can come up with a smaller set of targets to validate through techniques like PCR. So this was our result, and what is shown around the outside, because at the time we did this, that was the first map of the banana genome. There are 11 chromosomes and one large random piece. Now the version 3 is actually very tight, and we can go back and map them better. But if we look at the yellow, this shows us around 30,000 genes that are expressed in banana. Around in the green, 181 families of microRNAs. This is using other RNAs that have been reported in other plants. And then using our predictive software, we found another 56 microRNAs in banana that had not yet been reported in other plants. But what we observed from this, when we compared the gene expression between salt and non-salt treated plants, that there were almost 3,000 differentially expressed genes. So that's a huge number of targets to go looking at. The next thing we did was we determined which of the microRNAs are differentially expressed. So if you're interested in bioinformatics, I've given you the whole pipeline, but for general audience, if you just look at the pink and the green boxes inside the red circle. So on the right-hand side, we have differentially expressed microRNAs. So these are microRNAs that are either more highly or less expressed under the sodium chloride treatment. And then in the other pink box, we have the targets. So these are the messenger RNAs that they have complementarity to that are also differentially expressed. So we only looked at differentially expressed genes, and we only looked at those where we had both a microRNA and a matching messenger RNA. And then from those, we chose a few to do PCR validation to determine the differential expression at the level of PCR as well as from the sequencing population. So here we have a heat map of the results of those experiments based, first of all, on RNA expression only from the RNA sequencing and the small RNA sequencing. So on the left-hand side, the column that has all the microRNAs is showing you all of the differentially regulated microRNAs in the salt compared to control. A red color in this case indicates that there is less expression of the microRNA under the salt treatment. And what you can notice is that under the salt treatment, a majority of the differentially expressed microRNAs are actually reduced in expression. And the set of columns to the right, these the messenger RNAs that have matching sequences to the microRNAs. So one microRNA may actually target several messenger RNAs. So there could be up to 7 targets for some of them, and some of them just have single targets in our population. The green color of most of these targets indicates that most of these genes, these are the messenger RNAs, are more highly expressed under the stress treatment. So a microRNA by itself is difficult to know its function, but if we look at its target, then we assume that the function is to regulate that target, and that target will then have maybe an enzymic function. So the messenger RNAs that we saw that were differentially expressed and that are regulated by a differentially expressed microRNA were all related to signal transduction, development, stress, defense, and repair. And now we've gone from 3,000 differentially expressed genes to just about 30. So it's a much more manageable group. We then went on to confirm the expression of some selected targets using PCR. So what we are seeing here, that most of the microRNAs that were differentially expressed were actually more highly expressed under the non-stress condition. So I've indicated that with the green box in the top. In the non-stress condition, most of these microRNAs are well expressed. And what that means is they will be silencing their messenger RNA target. Whereas under the increased salinity conditions, they are less expressed. And so we get more expression of the messenger RNA target. So we expect this group of messenger RNAs to be enriched for stress-relieving proteins. And so we were interested in which of these proteins are actually in this group. And from these, we can then go on to say, if these genes are usually overexpressed in the stress condition, and we can see that they are regulated by microRNAs, if we were to artificially overexpress them, does it give any stress protective effect? Now, these genes are from banana, and it would be lovely to do it in banana, but that's very long and difficult experiment. So what we did was we overexpressed some of these genes in Arabidopsis as a model plant system. So first of all, we actually did several of these, but I'm just going to show you 2. At the top, you see RHD3. This is the messenger RNA target, and it is regulated by microRNA 157M. The blue bars indicate the data from the RNA sequencing, and the red bars indicate the data from PCR. So for the messenger RNA, we use RT-PCR, and for the microRNAs, we use stem loop PCR. And this told us that, yes, the PCR confirms the result that we saw from the RNA-seq data. And we're able to do that with more replicates, so we have some standard deviations and confidence in the data when we use PCR. Similarly, for the lower panel, we were looking at ZFP15, and it is regulated by microRNA 19. And the MAC indicates that this was a new microRNA that we predicted in banana that had not at that time been reported in other plants. So RHD3 had been reported as a GTP binding protein related to root hair development. So that's the RHD. And it was found in Arabidopsis poplar, and it was also related to ethylene homeostasis, but it hadn't been reported as a stress-related gene. And the 19 target, ZFP15, is a CCCH zinc finger protein, and it was validated by PCR, and it hadn't been reported to have any particular function. So we isolated the full-length cDNAs from the sodium chloride-treated roots and cloned them into overexpression plant vectors. So the first thing we do with these studies are to look at which part of the plant cell the expression occurs in. So we're using pCambia 1304, and we put our gene into this overexpression cassette, so it will be expressed in every tissue in the plant. And we also get expression of GFP and GUS, which is very useful as the marker when we're looking at our plants. So to start with RHD3, if we look at the onion cell assay, which is just to look at cell localization, we do transient expression in epidermal cells of onions, we can see that the RHD3 is expressed throughout the cytoplasm and not especially in the nucleus. compared to the precambia, where we can see a stronger signal in the nucleus. And this indicates that it's likely a cytoplasmic protein, and that is also consistent with the bioinformatic prediction. Now, for ZFP, this was also put into exactly the same construct, and now we see that ZFP is very strongly expressed in the nucleus compared to even precambia, and we don't see any cytoplasmic or no significant cytoplasmic expression of this ZFP protein. Now I'm going to take you through the results of the overexpression of the banana RHD3 in Arabidopsis. So using the GUS, we could see that our transgenic plants are indeed expressing the construct in reproductive tissues, leaves, and roots. And we selected single gene containing lines and did the back cross to T3. And then we check by PCR that we still have expression now of the RHD3. So this PCR on the right-hand side of the slide, qPCR, indicates using the primers for the RHD3 that the RHD3 is indeed present and being expressed. And the wild type and empty vector, of course, have no expression of this protein. And then we check the protein in terms of the ability to have an effect on the phenotypes of the plants. So the first thing is a simple germination assay of the Arabidopsis containing these genes. So what we saw when we put them in 150 millimolar sodium chloride was when we compare wild type, empty vector, and our 3 transgenic lines, they all grow very well when there is no sodium chloride, and none of them grow when there is sodium chloride. So this RHD3 overexpression is not apparently giving any protection against salinity stress. But when we did the salinity stress selection of these genes, we're actually applying salinity stress. So we're also applying osmotic stress and ion toxicity and various other stresses. So we decided that we better have a look just to check whether this gene might actually have a different way of protecting the plant, not actually against salinity, but maybe against the osmotic stress. We do that by putting polyethylene glycol into the media, which lowers the water activity and mimics a drought condition. And under these conditions, we see that the plants have much better germination rate. In fact, less than 10% of the wild type or empty vector plants can germinate, and almost or well over 90% of the 3 transgenic lines were able to germinate, and they also had significantly better roots. When we look at the plants under control conditions, so that will be the left-hand panel for the photographs of the roots and the left-hand bars in the graph below, we see no significant differences between the control and the overexpressing lines. But when we look at the drought conditions or the right-hand panel of the graph, we can see that there's significantly higher number of roots, root height density, and also root branches in the overexpressing plants. So the ectopic expression enhances lateral root branching and root height density under the drought stress. So this will improve the ability of the plants to withstand the stress. And we can see that also when we put the plants into soil. So in the we take 3-week-old plants and we hold watering for 7 days, and then we water again for 10 days. And 87% of the overexpressing plants recovered, whereas the wild type and empty vector, you see, are still struggling to survive. So what is the mechanism? Certainly we can see better root structure, but when we look at the plants in terms of their water content, even the water content is stronger. So there is less water loss in detached leaves, which is the graph on the left, and there is better water content or higher water content in the leaves when we have the drought condition. And again, you can see so I'm looking at the right-hand side of the slide. The panel on the left is with control conditions. There's no difference in the phenotypes, but when we look under the drought conditions, plants are much healthier. So they have better ability probably to take up water, but also to retain water. So we looked at the stomata, and we see that also the guard cells are visible under the stress conditions. 90% of the stomata are closed in the overexpressing lines. So they have better root structure, and they also have their stomata, more of their stomata closed, so they have less water loss And then we can look at other indicators of plant health. So in all of these next slides, on the control side, which is always going to be on your left-hand side, we see no significant difference between the groups. But when we put the drought stress, we can see that the plants overexpressing the RHD3 are considerably better. So they are higher in terms of chlorophyll level. They are lower in terms of malondialdehyde level, MDA assay, which indicates membrane damage. So lower is better in this case. So less MDA means less membrane damage. In terms of antioxidant, lower again. So lower levels of H2O2, hydrogen peroxide on the top and superoxide on the lower, indicating that they are better able to withstand the stress. And if we look at the stress scavenging enzymes, all of them show no difference under the control conditions and are significantly more highly expressed under the drought stress in the overexpressing lines. What about the ZFP? What happened when we put this one into Arabidopsis? So again, we see good expression in reproductive and non-reproductive tissues by looking at the GUS assay, and we here showing southern blot showing the presence of the ZFP in the transgenic lines. This time we figured we would test both salt and drought at the same time. So here you can see that in fact there was no significant difference under the drought stress using polyethylene glycol as a model. But now we see considerably better growth of the overexpressing lines under salinity stress. So it seems that ZFP is actually able to give salinity stress protection, but in this case, not drought stress protection. And again, we look at the structure of the roots and we look at the water recovery experiment, and we see that root hair density is better in the overexpressing lines and recovery from the salt stress is better in the overexpressing lines. And again, we look at the water content. There is less water loss and higher water content. And when we look at chlorophyll, MDA, and also ROS, again, the assays for hydrogen peroxide and superoxide, we also see that the plants are healthier under the stress condition. And again, this is just showing the antioxidant enzymes. Again, under the stress conditions, all of the 3 transgenic lines are much healthier. So the RHD3 overexpressing plants, when they were challenged by drought stress, had better root structure, but they also had increased stomatal closure, which we know is an ABA-mediated pathway, and increased expression of the ROS scavenging enzymes. And we also see increased expression of ROS scavenging enzymes in the ZFP overexpressing lines. So we want to determine if these overexpression lines have any association with increased levels of ABA pathway genes. So although we're testing the banana genes, we're doing it in the Arabidopsis model. So we look at Arabidopsis genes, an ABA-dependent pathway gene and an independent pathway gene, ABAF3 and RD29A. So here on the top, we have the RHD3 lines under the drought stress. And on the left-hand side, we have ABF3, and we have better expression. So this is showing that it's likely to be ADA dependent, whereas for the RD29, which is on the right-hand side, we see less expression under the stress condition. The bottom 2 graphs are the ZFP lines under the salinity stress, and we see the same pattern. So this indicates that for both of these overexpressing lines, the RHD3 and the ZFP, there is an ABA-dependent pathway likely involved in their mechanisms. So when we put these plants under the drought or the salinity in each case, we can see that there is a reduction in the controls. We will see the reduction in biomass, wilting, water loss, higher ROS levels, and lower activity of ROS scavenging enzymes and stunted roots. Whereas when we have the overexpression, the RHD3 plants are able to recover in drought conditions, and the ZFP plants are able to recover under salinity conditions. And in both cases, these can be associated with ABA responsive pathways. So the ideal would be if we can add both of these genes and overexpress them in banana, will we get a more stress-resistant banana? I don't have any data to show you yet. We are still looking for funding to continue these experiments. So to summarize, we started with a comparative transcriptome approach, which allowed us to identify genes that were targets of microRNAs and responded to the salinity stress, which also by chance also helped us discover a drought stress gene. And there are many genes changing their levels of expression, but by focusing on the microRNA targets, we were able to get a set of genes that were somewhat validated by both being responsive to the microRNA as well as responsive to the stress. Overexpression of RHD3 in Arabidopsis confers drought but not salinity tolerance. And our data are consistent with the role for RHD3 as a cytoplasmic signal transducing G protein that mediates ABA response to water stress, whereas the ZFP confers salinity tolerance and plants are much healthier and is also ABA associated. So it's expected that overexpression of these microRNA regulated genes could be used in protection of crops, and they might be useful for food security in the face of climate change. So I would like to especially thank Prof. Varsha for inviting me, and also the organizing committee for all their hard work to put the meeting together. The work I've shown you has been funded by Ministry of Science, Technology and Innovation Malaysia, as well as the Ministry of Education in Malaysia. The majority of this work was done by 2 PhD students, Wan Sin and Ranga, and we had help from our bioinformatic technician, Wai Shen. The students were co-supervised by Professor Nuzulani and Dr. Rabi, and the RHD3 work was done by Ron, and the confirmation on the ZFP was by Sarah. And these are some of our other publications on banana. We have a big group working on banana and quite a lot of papers. Thank you very much, and I'll be happy to take questions.
Thank you, Jennifer. It was a wonderful presentation, and I'm sure you all have enjoyed listening to it. Even in India, banana is a very important plant, commercial crop, as you all know. And then with the climate change, a lot of challenges are going to take place. And the paper which Jennifer has presented is very relevant in the context of climate change, because salt tolerance or salinity stress, this is one, it's going to be a challenge in the coming year. So thank you very much, Jennifer. So now we'll have the second speaker, that is Dr. Stephen Manchester. He's an adjunct professor, Department of Biology, Florida Museum of Natural History, University of Florida, USA. So major research interests: evolution of extant angiosperm families in the northern hemisphere, multiple organ paleobotanical investigations providing characteristics of flowers, fruit, pollen, leaves, and wood for inclusion in phylogenetic analysis, Paleocene and Eocene flora of Western North America, Central Europe, and Eastern Asia. He has enjoyed collaborative research with paleobotanists in Canada, China, Czech Republic, England, Germany, Hungary, Japan, Russia, and the United States. Then his professional associations: Botanical Society of America, Paleobotanical Section, that is a Paleobotanical Section, American Society of Plant Taxonomists, International Organization of Paleobotany, International Association of Wood Anatomists, American Association of Stratigraphic Palynologists. So he has a publication of over 245 research papers. So it's going to be a privilege for all of us to listen to Stephen. So Stephen, thank you very much for your time. Now the time is yours. You can go ahead. Is it ready? Yeah, Stephen, you can start. You have mute yourself. You on. No, on the mic. You have muted. Okay, okay now.
Can you hear me?
Yes, yes, we can hear you. You can go ahead.
Is it okay?
Yes, yes, okay, it's okay. You can go ahead.
Okay, thank you. I'm sorry about intermittent connection. Good morning, I can say from here, and good evening to you. And I really want to thank you for staying a bit later to hear my presentation, and thanking Professor Nathar for inviting me. This seemed like a good opportunity to share with you some of our recent discoveries in studying these latest Cretaceous early tertiary plants from the Deccan intertrappean beds. The Deccan traps, some of you probably know, are a series of basalt flows, lava flows that covered a huge amount of central India around the time of the end of the dinosaurs. And between those layers of basalt, we find layers of sediment that include chert with fossil plants in them. And that's what I've become so interested in as my co-authors. We could ask, well, why do we want to study these fossils? Well, it's giving us some insights into the past history of India, its biotic history. What was it like when India was still tectonically isolated and had not yet bombarded into Eurasia? What were the local ecological and climatic conditions? What can we say about the relationships of the flora and fauna with those of other land masses? Can we say that some of these fossils provide evidence for radiations of groups into other continents from India? And how did these organisms respond to the Cretaceous Paleogene boundary events? So there's a lot that can be addressed, but of course I've got to keep this quick. And what I want to give you a sense of today is how we're now able to use new methods, including X-ray technology and micro CT scanning, for getting even more information out of these fossils, out of the rocks basically, and how they're giving us new insights on the flora. This work on the Deccan intertrappean beds really goes back to Birbal Sahni. Professor Sahni in the 1940s became quite interested in the cherts from Mogan Kalan near Chhindwara, and you can see there an example of one of the fruits that he described, a fossil fruit called Enigmocarpon, that he inferred to belong to the Lythraceae of the the Bertales order. And you can see that there's beautiful structural information preserved in these plants. And so they're coming out of chert. And here's some examples of this chert that's interbedded between layers of basalt. So it's basically mineralized pond muck with all the plants that might have been falling into these depositional environments. And they have beautifully preserved remains, but these chunks of chert are opaque. You cannot see through them to see what's inside. So different techniques have been used to get the information out of these. One is just fracturing and looking at the surfaces, what you find inside, the broken edges of fruits and seeds and wood. This gentleman is demonstrating that peel technique, where the fresh cut surface of the churd is etched in nasty acid, hydrofluoric acid, to remove a layer of minerals, and then it's painted with plastic, and the plastic is peeled away, and in that way we get serial anatomical sections through these fruits. It's very labor intensive, and it might take many months to get a series of sections going through an entire fruit or seed. These are color images of that same plant that Burbalsani investigated in 1943, and you can see just excellent preservation of seeds within the fruits that can be seen by these traditional optical techniques. I'm hoping that maybe this movie is going to show for you. I'm seeing it on my screen. This is an X-rayed fruit of Enigmocarpon. Basically, it's the same specimen that's shown in the upper left here, collected by Shaya Chidley. And here we see with the CT scan data, we're able to get a three-dimensional view of this thing, basically traveling by transverse section through it. And with the software we have for processing micro CT scan data, we can select any orientation. So we can cut cross sections or longitudinal without damage to the specimen. In the old way we did it, we would cut and peel, and the original specimens were damaged in the process. But they did give a lot of information. So the line drawing on the left here is by Burbalsani in 1943, and he was looking at the detail that could be observed in the seeds under the microscope of that Enigmocarpon fruit, an example you see on the right. And so there's his diagram. Oops, sorry. See if this movie will work. Yeah, his diagram shows what he was able to see optically, and this reconstruction on the right is from the micro CT scan work. So his work was very accurate but painstaking in the amount of time to develop the concept of the internal morphology. And it fits, even today, we agree that Lithraceae, as suggested by Burbalsani, is the proper position for that fossil, although it's extinct. I'm hoping I have time to show you a few examples out of many from different taxa that we've been able to verify from the Deccan Trapian. Here's one of them. It's called a palm. We found that palms were quite abundant and diverse. This was originally thought to be trilocular Euphorbiaceae, but then we found that they have these germination pores down at the base of the fruit, and those pores are diagnostic of the coccoisoid palms. So this panel on the left of this image is some newly recovered micro CT scan of this beautiful detail showing the perianth at the base of the fruit, developing fruit, and even details of the seeds preserved inside the locules. And this section also shows where the germination pores and opercula are. And for comparison, we have closely related examples from South America, Atalaya and Butia. It seems this fossil is extinct, but it is in that coccoid palm group. And we have recognized other palms, also from multiple localities. It's interesting that this one, Hypheniocarpon, is related to something that's now endemic to Madagascar, whereas the previous one I showed you had more similarity to those in South America. Here is some, so we were just looking at light microscopy of Hypheniocarpon, and here is some results from CT scan. And the nice thing about the micro CT scan is we can move through the entire specimen quite easily and discover any unusual features. Like at the very base, this might have been missed in traditional studies, but we can see that there are actually 3 carpels and 2 undeveloped locules at the in addition to the main locule that we typically see. And the specimen on the right shows a germination pore at the top, and then by chance we captured one where it's actually germination is sort of caught in the act with a seedling starting to develop and emerge. We've also found other palms. This one, for quite a long time, even since Sani's time, was known to be a monocot. It has scattered vascular bundles and monocotyledonous embryos, and but whether it was a palm or not has been controversial. Hoping we can get a movie to work here. At my end, I'm seeing a movie, and I hope it is for you. This is a movie moving down through the fruit and showing the three locules, seeds inside each locule, and the wall having cavities surrounding. And we're moving from the base toward the apex, and near the apex you can see the germination area and stigmatic area. So there are a number of features that suggested palms, but something we discovered recently is that they do have germination pores on the distal side of the locules. So that is helping to confirm that they are indeed palms, although they're born in strange infractescences, where you can see them lined up in these tube-like structures where the pedicels of the raceme don't seem to be easily identifiable. So Trichochides seems to be another palm. And this genus that was described as a potential grass, we're now recognizing that it too could be a palm because it has these circular germination pores and has, we've even preserved that the embryo is seated in behind the operculum, and there's a notch on the ventral side that's comparable to what you might remember from looking at date seeds. This, of course, is not so elongate as a phoenix seed, but we think that this is showing an extinct combination of characters. We also have the ginger family, the Zingiberaceae, well represented in this Indian fossil record. Here's a nice complete specimen that was actually first described by Jane to be a banana, a Musa cardiospermum. However, in closer investigation, we've found that yes, it's Zingiberales. He was in the right order, but it doesn't quite fit for Musa, but rather it's an extinct genus which has the name Mordiocarpon in the Zingiberaceae. And here in the lower left panel, we're seeing for the first time what the seeds look like extracted from the chert, because we cannot really see them in 3 dimensions by the traditional method. Although the traditional methods give excellent detail of the anatomy. And another Zingiberaceae we've discovered just by chance, scanning some of the matrix, some of the chert with the CT scanner, we came across this specimen as we're moving through the X-ray data and recognized, well, that is also, it shows this channel, micropilar channel at the top with there's an operculum, and the embryo is seated just below there. Bunch of features of the Zingiberaceae family. So I went ahead and identified where that was in the block and then cut it physically with a diamond blade to get these light microscopy images here. But from that X-ray data, we're also able to reconstruct the 3-dimensionality of the seed, and it's really a strange shaped thing, although it seems to fit nicely. Here's the Wraith. You can see perhaps in here. Good information to say we've got diverse gingers in the latest Cretaceous of India. Same situation here for the Euphorbiaceae. We've found trilocular capsules with a single seed, seeds having subapical axile placentation. Here the seeds have been digitally removed from the fruit that you see in the upper left. And on the right-hand side, I'm showing a micro CT scan survey we did of various modern fruits of Euphorbiaceae, hoping we might be able to link this fossil to a particular clade, but there's a lot of convergence among unrelated clades in the family, so it's a little bit difficult to be sure where this fossil belongs from only fruits. We might like to have the flowers as well. But it does seem to be strikingly similar to the carotenoids. I'm showing you here a picture of three gentlemen. The one in the center is M.T. Sheikh from Nagpur, and he is one of the co-authors of this paper, 1972, with Shahid Chidoli on a plant, an extinct plant that they put in the Malvaceae. Here's a plate from their picture, or from their publication. And on the right, you're seeing some examples of what we've been able to learn from on this plant from the micro CT scan. The upper 1/3 of this, or 2/3 of this right-hand panel are all of a single specimen. So it's the X-ray data used in different orientations and derivations to show how this fruit looked in 3 dimensions. And you can see it has these 10 carpels with 10 awns at the outside. These are schizocarpic fruits, each with a single seed, in each carpel. And there are a number of features that you take these all into consideration. Yes, it belongs to the Malavoideae, but it does not belong to a modern genus. But it clearly indicates that family and subfamily were well represented in India at that time. Here's another genus that Chidoli and Nambudiri described, and we see examples, some color images on the right of some peels we've made more recently, and then here are some CT scan. From the CT scan, we can also see that the seeds are in 2 rows within each of the merocarps, and this too fits for being in the Malavoideae group. So that family was well represented. Until recently, we didn't know that we had, or we hadn't confirmed. There had been some identifications of Burseraceae from petrified wood, but we didn't have fruits or other evidence, flowers. And here we're seeing newly discovered pyrenes that have the morphology of Burseraceae. These golden colored pyrenes here were digitally extracted from the chert that you see in the upper panels. And in the upper left, you might see, well, there's one of those Burseraceae fruits. Also, there is one of those palms that I showed you earlier, Palmocarpon Drypedioides. So there's a pretty good concentration of plant material in this chert from the Gugua National Park area in near Shapura. And here on the right are modern Burseraceae for comparison. So there are good similarities in these triangular fruits being born radially within these fruits, with a placental scar on the middle of the ventral side. And it was a real surprise to us as we looked through the scanning material we came across. So this is a chunk of that chert, and we CT scanned it. And then right here is this little speck that's identified in three orientations here. That turns out to be a flower, which is magnified higher here in the lower panels. And the flower is preserved basically as a space in the rock. The flower has disintegrated or deteriorated, and it's surrounded by the silica. So we can take that inside shadow that you see there, or silhouette, and in 3D we can resurrect this plant. And it turns out to have features of the Burseraceae. Here it is shown for comparison with Proteum and Boswellia, modern Burseraceae, and other Sapindales that it shares a nectary disc, 5 stamens. So good evidence now for Burseraceae. Here's Saniocarpon that was described by Chidoli and Patil. Sorry I don't have the picture of Patil to show you. He might be there. Shahid Chidoli was influential to me too in introducing me to these studies, as well as my main collaborator, Kapgadi, who was her grand student. So this was called Saniocarpon. named after Burbalsani, of course, and its affinities were uncertain. It was thought maybe to be an Ecclusiaceae family, but with the aid of CT scan, we're now able to get more information out of these cherts, and we find that these fruits, these pentalocular fruits, had 5 winged pyrenes. So those are not seeds as they were interpreted before, but pyrenes of the fruit with these lateral wings on them. And you can see similarity with, excuse me, similarity with modern Boswellia, which can have 3 to 5 locules and also winged pyrenes, as you see here, lower panel. So it seems to be good evidence for Bursaceae. There's still other that we haven't worked on yet. These are modern Garuga on the lower left for comparison with something that has also secretory cells in the pericarp. You may know that India is famous for having the oldest known fossil grapes, and here is this Indovitus that was described some years ago. At that time, 2013, we were not using CT scan yet, so that paper was all based on traditional methods. We can now actually use CT scan to supplement that work. If the video is working, you can see that we're looking at an extracted seed from the chert rotating radially. So you can see the ventral infolds and the dorsal chalaza. So, it's in the grape family. It could be in the clade related to Vitis itself, which is here, but it also has similarities to Ampelopsis. There's one other thing I'd like to mention about what we're able to learn from the CT scan. We can find that in different orientations, these fruits will have very different looks. So here's a transverse sections at different, from base to apex, and here are lateral views, sort of sagittal and coronal cuts. These fossils look different, you know, in different orientations, and we don't, until the availability of this CT scan, we weren't able to see the three-dimensional outline of these things. So we've learned, let me get a drink of water here. So we've learned that these 2 panels across the top, they were thought to be 2 different genera. We had these bilocular fruits here and this differently shaped trilocular thing. Those are all, well, you can see they're all, these images on the left are all from a single specimen. So we now know that Plectroniocarpon and Verbenaciocarpon and what was once called perhaps Sparganium, they are all one thing. So this technology is allowing us to, it's reducing the apparent diversity that we had, but we're also discovering more new things this way. Mostly what I've shown you are examples of the rhozoid, monocots and rhozoids. There are some asterids as well. I should say rhosids, but also asterids. And here's an example from the Periophilales Montiaceae, which is really easily recognizable by its free central placentation, the curved embryos, and tricarpellate loculocidal capsules, which are similar to what you see in these modern things. This is related to Portulacaceae, but the Montiaceae has been removed in recent years to its own family. Molecular evidence. And I don't want to give you the sense that we have solved all these taxonomic problems. We still were able to learn a lot about the morphology of these fruits, but it's not clear in some cases what modern group they might belong to. So I realize we have a good number of taxonomists in the group here, and so if anybody has some ideas on the relationships of this Bacatocarpon, this fossil genus, I'd be happy to hear it. There are 2 little seeds that are born in locules of what appears to be an empty third locule dominates the cavity of these fruits. And here again, we have on the right-hand side, we're seeing a single fruit in different planes of section, and you can see that we were misled in the past into thinking that 3 different taxa orientation. So it's really useful to be able to get a sense of the 3-dimensionality of these things and how they look in different. So I can say, yes, many mysteries remain, but by using these new X-ray methods combined with the good information we can get from peels and other older methods with reflected light and transmitted light, we're learning a lot more about this flora from that time. So thank you. I'm sorry that I'm maybe holding up your dinner, but thank you for your attention.
Thank you so much, Stephen. The presentation was so elaborate, and I'm sure you must have seen a lot of Indian plant, and which is very interesting, and also the slides were very beautiful. And that shows that in paleobotany, a lot of work has been done from India also. But I only wonder is that at present, whether universities are offering courses in paleobotany or not. Earlier, this Pune University has a very strong, very good paleobotany section.
So I'm really indebted to some of my co-authors who are, which I list on the first slide.
Anyway,
yeah, Dr. Kapgadi. I showed Dr. Shai.
This time, you should have come to India this time. That would have been very
I apologize. I enjoyed when I was there in 2016. It was a very nice group, and I missed seeing you in person this time.
Because in your presentation, you have shown some from Nagpur, and Nagpur is very close by here from Amravati. That's why it could have been very interesting for you to visit those areas. Anyway, next time you can come again for the next meeting, and you can visit those places. Thank you so much for your time and for the wonderful presentation. Thank you.
Thank you. For your time. Thank you.
We are not asking questions because time is not there, so that's why. Okay, thank you.
Thank you, Dr. Mao. And now we are moving on to our session, which was supposed to held earlier, that is scientific session 3. We have taken up scientific session 5. So now scientific session 3 is Women Botanist Award competition. There are 2 participants, Dr. Aparna Parikh from Jaipur and Dr. Rasmi S from Thrissur. I would request them to kindly restrict their presentation to the limited time, that is 18 minutes, and there will be a buzzer. They have to complete their presentation. The judges are up there. I would request you, Priya. I would request the judges to come up the stage. That's Professor Anita Mukherjee, Professor A.K. Bhatnagar, and Professor Manoharachari sir. Step mind कर लीजिएगा please.
You are most welcome for this Women Botanist Medal Award, and we 3 of us are here, and there are 2 contests.
contending for this particular award. The time that is given is only 18 minutes, followed by questions. You have to be very brief, and you have to be very alert, and you have this being a competition, and you exemplify yourself on the important things in your presentation, and you have to be very clear in your approach. And this is for the information on behalf of the Indian Botanical Society and on behalf of the organizers. I welcome all of you. And now
I call upon Dr. Aparna Panik. Therefore, I'm going to talk on promotion of indigenous knowledge and biotechnological strategies for conservation of endangered plants in dilution of the possible therapeutic activity.
Thank you, ma'am. Very good evening to the galaxy of scientists from all over the country. Today I'm here to present my research on the biotechnological strategies and promotion of indigenous knowledge for conservation of endangered ethnomedicinal plants in the rural regions of Rajasthan with evaluation of their possible therapeutic applications. Traditional knowledge and biotechnology. Is a synthesis possible? This question crossed our minds, and with this hypothesis, the project was sent to the Department of Science and Technology, Government of Rajasthan, and fortunately we got this project and we are working on this project. And this question, it crossed our minds throughout the course of study, but we are very much fortunate that I think this amalgamation of the traditional knowledge and biotechnology has definitely led to some opportunities in the field of drug development and the drug discoveries, both in the accelerated manner. This picture draws our attention that endangered ethnomedicinal plants conservation the need of are in the present scenario. We all are very much aware of the fact that ethnomedicinal plants, they are being targeted in research for the new natural compounds. But at the same time, their overexploitation has led to their need for their conservation. So an effort has been made to conserve the endangered ethnomedicinal plants of Rajasthan. We all are very much aware of the fact that Rajasthan is ranking 6th in the case of tribal population, and these people, they are having tremendous knowledge about the medicinal utility of biodiversity, but at the same time, this traditional knowledge is declining day by day. So there is an urgent requirement for the documentation of this knowledge before it is lost, because these traditional communities, they are losing their socio-cultural characteristic features. So our study is based on the survey in the rural areas of Rajasthan, based on the folk remedies for the cure of health ailments of the indigenous people residing in those areas. A structured questionnaire was based to know about the medicinal importance of the plant, the mode of administration, the occurrence and the popularity of the plants, and mostly the plant part used. So with these objectives in mind, we have done our studies, the floristic survey of the ethnomedicinal plants, the data analysis of different parameters which were based on this survey, the effect and popularity to detect the phytochemicals in the selected ethnomedicinal plants, and the evaluation of the antioxidant and the antimicrobial activities. So mostly the rural areas were being surveyed for the ethnomedicinal plants which are facing the danger of extinction due to the overexploitation by the indigenous people living there. We have surveyed mostly three areas have been surveyed, and because it is an ongoing project, the work has not come to an end, and we are still in the process of doing the experiments with the surveyed endangered ethnomedicinal plants. This is the desert area, the Shekhawati region and the Hadoti region. The structured questionnaire was made to collect the traditional knowledge from the informants. Basically, three sampling techniques were being used: purposive sampling technique, snowball sampling technique, and the random sampling techniques. As a result of this floristic survey, we have made an elaborative chart of about 240 medicinal plants, and from these medicinal plants, we have selected around 30 plants which are facing the danger of extinction. This was the data analysis which was based on the floristic survey. The plant part used, effect and popularity, frequency of occurrence, and the mode of administration. As a result of this survey, the plant which was used maximum by these indigenous people in curing the health ailments was the leaf, followed by the flower, stem, rhizome, root, and the other plant parts. This was the effect and popularity, showing the effect and popularity of these plants. This was the types of, or we can see the plants were formed in this manner: rarely seen, vulnerable, endangered, and some of the plants were presently safe. This is the data analysis of the floristic survey. Then as far as the mode of administration of these medicinally important plants was seen, infusion was the most common part, most common mode of administration, followed by the extract, powder, paste, and the other forms of administrations. This is also showing the mode of administration. Then this is the table showing the endangered plants which are facing the danger of extinction due to overexploitation by these indigenous people. These are some of the endangered plants: Naranji granuluta or the Mahabilb parne. This is called as the future plant or the potent medicinal herb, because each and every part of this plant is used in the cure of the health ailments by the indigenous people. It is having antioxidant property, anti-cancerous property, anti-diabetic property, followed by the commiphora, the endangered plants, Leptadenia, commonly known as Jivanti, one of the most important ingredient of the Chyawanprash. This is the Echorus or Vacha. Commonly, it is the memory enhancer plant. This is Paganum harmala. These are some of the pictures of the endangered plants of Silistris paniculatus, Argyria speciosa or the elephant creeper. It is also an immunomodulator. Curculigo orchoids or the black gold or Kali Musli. It is also known as a memory plant, enhancing the memory. Then Centella asiatica or the Manduk Parni. It is also having the antioxidative properties. Then the plants which were being endangered or facing the danger of exploitation, these plants were chosen for micropropagation. As we had many areas in this survey, so we have chosen one plant from each of the areas surveyed, and these plants were being micropropagated for their multiplication. Two methods were being followed: indirect way by forming the callus, and the direct way without forming the callus. This is a diagrammatic representation. Here we have done the micropropagation of Commiphora mucul from the leaf explants, followed by Paganum harmal commonly known as. This is also done from the leaf explant on MS medium supplemented with various constituents. This is the plant of the mostly Caligonum is known as fog. It is also having medicinal properties. So it was also taken into consideration for the tissue culture studies for its micropropagation. Then in the present scenario where we are facing the drug, drug resistance by the human pathogens, so an alternative had to be screened for the discovery of certain antimicrobials. So plant extracts were also used in this case, and antimicrobial studies were also being carried out to see their effect on the various parameters. So this is the antimicrobial study which is done in the Silistris paniculatus. You can very well see the zone of inhibition. 2 bacterial species, the E. coli and the Bacillus subtilis, were being used, followed by the antimicrobial activity seen in Paganum harmala against the Bacillus subtilis. Then antifungal studies were being also carried out, the root, the stem and the other parts which were having the active constituents. These are some of the pictures showing the antifungal activity of Paganum harmala. Then antibacterial leaves on the E. coli, antibacterial activities of leaves, stems and seeds on E. coli were also seen. These are the results which are showing the antimicrobial activity of the parts which were being examined for this antimicrobial activity. This is also justifying the results which were being seen in the previous tables. Then Bacillus subtilis was the other organism on which it was tested. Then antifungal activities were also being carried out. Then presence of certain phytochemicals was also tested in certain endangered ethnomedicinal plants which were showing the presence of the phytochemicals in various plant parts tested. This is showing the phytochemical screening of Commiphora, just showing all the chemical constituents except the carbohydrates and glycosides which were not seen in the samples. This is Silistris paniculatus. Almost all the active constituents are being seen. This is Argyria speciosa, important ethnomedicinal plants. This is Centella asiatica. Naranji granulata, very important ethnomedicinal plant. Then Jivanti. Then the antioxidant activity was also tested, and the plant extracts, they were having the free radical scavenging activity. The DPPH free radical scavenging activity was also seen in different parts. The leaves, stems, seed, fruits of Justicia adathoda. Then this is also justifying the results. This is in the form of the charts where we can see the different plant parts. Then the anti-diabetic activity was also tested. The plant extracts, they are blocking the amylase activity. So no glucose is being formed, so diabetes is not taking place. It is stopping at the very initial level. Then alpha amylase inhibitory potential was also tested in Andrographis and the Justicia adathoda plant. Here you can see different root, stem, leaf parts. They are showing the activity. Then in the present scenario where we are talking about the nanotechnology or the nanoparticles, we have also tried our work in this direction also. And these are the different synthetic approaches available for the preparation of these nanoparticles. So the plant extracts are now being used for these preparation, and because this green synthesis, it is having many advantages. They can be used on a large scale. These conditions, the experimental conditions like high energy and high pressure, they are not required. The biological component itself acts as a reducing and capping agent, and they are eco-friendly, they are not toxic, they are friendly for the environment. So, from the various part samples, these nanoparticles were also prepared, and they are playing an important role in the antimicrobial area. They are having the anti-cancerous property. The drug delivery is made very easy. We have also tried the antibacterial activity of the synthesized nanoparticles on various microorganisms. Here you can clearly see the positive antibacterial activity, of the synthesized nanoparticles against the standard. Then this is the antifungal activity of these. Then to test the, actually this is an ongoing project, and we have surveyed many plants. So we are doing various parameters, like we are doing the micropropagation, the antimicrobial activities, the other phytochemical analysis. So the study has not come to an end, and it is an ongoing process because we are in the mid of this project, so it will take a long time. So some of the parameters of the plants surveyed we have already finished, and some are on the way to be done. So in the process where we have done the antimicrobial studies or the phytochemical studies, so to detect the presence, we have also carried out the GCMS analysis, and in that series we have taken up this Paganum harmala. So as a result of the GCMS analysis, it was seen that already the phytochemicals which were being present, so they were detected in this analysis, where you can see the hermine, the peak was seen, the hermine, common phytochemical present. These hermeline and hermine, the peaks were seen. So this GCMS analysis also justified the presence of these phytochemicals in the parts which were playing an important role in the studies carried out. So this work, actually this was an amalgamation of the traditional knowledge along with the biotechnological aspects. So this is an area of research which presents a variety of opportunities for conservation, sustainable development and healthcare. Then proper evaluation of ethnomedicinal and ethno-veterinary plants play an important role in preventing various ailments in human beings and the cattle. Then morphogenesis of growth can be understood by performing the biotechnological exercises like the tissue culture. Then phytochemical studies also help to reveal the constituents of the plant extracts for bioactive agents, which are very much helpful in the manufacture of the new herbal drugs. And it may offer immense opportunity for researchers which are engaged in the validation of these traditional claims. The
paper is open for questioning. There are 2 to 3 minutes only. You can ask only relevant questions to the topic concerned.
It was full MS. It is from the plants. No, actually tissue culture is a hit and trial method. So first when we were doing the regeneration, callus was formed. It was not our intention to form the callus, but first callus was formed, and then from the callus the plants regenerated. So there were two ways of micropropagation. One is the from the yes sir. In case of Paganum, we were also able to generate the plants from the callus.
In botanical name, you have not mentioned author's name.
Yes.
Authors are botanical names are not valid until the author's name are not mentioned.
Oh yes, sir, that I will take into consideration. The ethnobotany part, I should have mentioned the authority name there. I will definitely mention.
Number two, you have mentioned endangered plants, 30 endangered plants. So that all are 30 endangered or threatened plants?
From the ethnomedicinal plants surveyed, we made a list of the plants which were threatened, endangered, and we are taking firstly these plants into consideration for doing the studies.
From there you have taken. Because FRLST conducted workshops in 28 states of all over India.
Okay.
They have IUCN authorized FRLST. So in Rajasthan Biodiversity Board, they have also conducted such conference workshop on IUCN criteria 3.1.
Okay.
So that was the authorized literature on threatened plants. Endangered is different or threatened. In threatened category, there are three categories. Critically endangered and vulnerable. So endangered is a part of threatened plants.
Okay, we have mentioned that some plants are threatened, some are endangered. We have. Thank you. Yes.
You've done also also.
Yes.
And did you check the yield of the active?
The yield of the active constituent, we are in the process of doing, ma'am. I told you that this work is not finished. We are still doing, so we have come up to this stage, and next we will see this parameter
Also, doing qualitative analysis with alkaloids and go on with the
quantitative. We are, ma'am, actually we are in the process of doing all these parameters. Yes. Otherwise it will. Okay, ma'am. Similar papers were being complied in this.
Thank you very much, Dr. Aparna Pari. Now I call upon Dr. Rashmi S. from Calicut. Dr. Rashmi, you are given 18 minutes relevant to the topic and follow the norms.
Good evening, everyone. Myself Rashmi from Kerala. I completed my PhD from University of Calicut under the guidance of Professor Santosh Nambi, and very recently I joined as Research Associate in Botanical Survey of India. The topic which I am going to present here is the taxonomy and diversity of the genus Sonorilla, family Melastomataceae in Western Ghats. Sonorilla is the largest genus in the tribe Sonorillae with about 180 species worldwide, and it's showing greatest diversity in tropical Asia. They are mainly distributed in Sri Lanka, India, Indochina and Malay archipelago. Next is why the studies of Sonorilla is so important. India has the largest diversity of Sonorilla in the world, and the Western Ghats harbors a greater percentage of endemic species. But in flora of British India, only 20 species are reported from the present political boundary of India, and there is no real revision after that. In this context, the studies on the taxonomy and diversity of the Sonorilla in Western Ghats is undertaken with the following objectives. One of the major objective is to prepare an updated illustrated taxonomic account of Sonarlas in Western Ghats to document infraspecific diversity and develop keys for the identification of species. Next, moving to the materials and methods, which mainly involves the extensive field exploration to different parts of Western Ghats and to maintain the live collection in Calicut University Herbarium. Herbarium specimens deposited at 21 Indian herbaria and the virtual database of 31 overseas herbaria were consulted. Also, the prestigious herbaria like Royal Botanical Garden Kew and the Royal Botanic Garden Edinburgh and the Natural History Museum were also visited to consult the important collections of Sonarella. Next is about the result, that is systematic treatment. The genus name Sonarella is originally taken from a Malayalam name Sonneri-illa, which has been cited by Van Reed in Hortus malabaricus, and they are generally producing trimerous flowers in uniparous cymes. Similarly, we can also observe the pentamerous and tetramerous condition as an anomaly in different species of Sonarella. And many species exhibit an array of morphological diversity, which makes the genus morphologically very complex. These are the key to the species of Sonarella in Western Ghats based on the morphological characters. There is no sectional classification for Sonarella. So here I try to classify different groups of Sonarella based on the morphological characters. And here I try to classify into 4 groups. The first one is Ecolocene taxa, that is species more or less stemless and tuberous. This group occupies 11 species. Out of these 2 are stoloniferous, including Sonarella janagiana and our recently described species Sonarella stolonifera. The remaining 9 are non-stoloniferous. They are Sonarella vietcampiana, Sonarella rotundifolia, etc. Here the 3 names, Sonarella scapigera, Sonarella vallici and Sonarella rotundifolia are lectotypified during our study. These are the beautiful images of species coming under this group. Only the important findings or observations are discussing here. The first is about the Sonarella stolonifera that we described recently from the Silent Valley National Park, and we published in the journal Britannia, the publishing from the New York Botanical Garden, and it is assessed as critically endangered. This is the images of Sonarella stolonifera and the type specimen of Sonarella stolonifera. Next is about Sonarella vietcampiana. It is a very distinct species with a long peduncle and 10 to 30 flowered cymes and acuminative rostrate anthers, but it is wrongly mentioned as Sonarella rotundifolia in major floras of Kerala. But both species are very distinct. The identity of both are clearly demarcated during our study. Next, we are moving to Sonarella vallici. It is another interesting species which is showing high degree of variation in leaf color, size, shape, and variegation. And this leads to the proliferation of many new taxa recently, like Sonarella ecolis, Sonarella gadgiliana, etc. But those are not tenable. Another interesting findings we noticed here is recently Matthew et al. Published Phyllogathis indica as a newly recorded species of the genus Phyllogathis from India. Actually, Phyllogathis is a distinct species with tetramerous and pentamerous flowers, also with a distinct connective with the dorsal and ventral lobes. So, critical studies shown that this Phyllogathis indica is merely a tetramerous form of Sonarella vallici. So, here we synonymize Sonarella we synonymize Phyllogathis indica under Sonarella vallici. Next, moving to the second group, that is Colocene tuberous species. They are with a delicate stem and a tuberous base, which occupies 7 species. They are Sonarella talboti, Sonarella sahyadrica, Sonarella reedi, etc. These are the beautiful images of the species coming under this group. The first, we are discussing the important findings from this group. Sonarella longipedungulata, we recently described from the Banasura hills of Wayanad. And the epithet longipedungulata refers to the characteristically long peduncle of the inflorescence. These are the images and the distribution of Sonarella longipedungulata. Next is Sonarella congenensis. It is a pretty tuberous species with angular stems and large leaf-like persistent bracts. This Sonarella congenensis we described from the Chandreshwar hills of South Goa. Next is Sonarella lateritica, which we published from the Konkunna hills of Calicut, and the epithet lateritica refers to the precise habitat. This is the illustration and color images of the species. Next, we are moving to Sonarla reedi. It is another interesting species which is showing wide distribution in Kerala. When we are talking about the Sonarla reedi, some points are very important. Like recently, many species are published very recently. I think within 6 months, many species coming, and they are always they are comparing this with the Sonarla reedi. Actually, Sonarla reedi showing high degree of variation. In case of the leaf color, shape and number of flowers and the length of peduncle. So, for example, the Sonarla canaliceriensis and Sonarla carolensis. When we are comparing this Sonarla canaliceriensis and Sonarla carolensis, the characters used to delimit these two species are overlaps with the circumscription of Sonarla reedi. So, it is synonymized under Sonarla reedi. One another important finding is that recently Santosh Kumar and Filimban synonymized Sonarla canaliceriensis under Sonarla sahyadrica without noticing its overlapping characters of Sonarla canaliceriensis with Sonarla reedi. So we corrected here. Actually, Sonarla canaliceriensis is a synonym of Sonarla reedi, not Sonarla sahyadrica. Next is the third group, that is coalescent non-tuberous taxa. They are with a thick or more or less semi-woody stem. This group occupies 24 species and 1 variety, including Sonarla anamudica, axillaris, etc. 8 species are lectotypified here. These are the beautiful images of species coming under this group. Sonarla elegans, Sonarla panmudiana, etc. Next is about the Sonarla gambli. Recently, Singh et al. Synonymized Sonarla gambli under Sonarla speciosa. From these pictures, it is very clear that Sonarla gambli is very distinct from Sonarla speciosa. This is the comparative images of Sonarla gambli and speciosa. The first one is Sonarla gambli, the other one is Sonarla speciosa. Here we can see the distinct inflorescence pattern of Sonarla gambli and Sonarla speciosa. Also the number of flowers, and here it is the anther characteristics are very clear, both are very distinct. So here the original species status of Sonarla gambli is reinstated here. Next we are moving to Sonarla roxburghi, this one we recently described from the Mangalam Reserve Forest of Kerala. The epithet roxburghi honors William Roxburgh for his valuable contributions to the taxonomy of Sonarella. Next we are moving to Sonarla axillaris. Actually, Sonarla axillaris and Sonarla versicolor are described by White in 1846 from the Nilgiris. But after that, many authors treated it as a Sonarla axillaris is treated as a synonym of Sonarla versicolor or a variety of Sonarla versicolor. Here, during our studies, we observed that this Sonarla axillaris and Sonarla versicolor are very distinct. Here we can note the first one is the inflorescence of Sonarla axillaris. It is axillary inflorescence. The other one is Sonarla versicolor, that is terminal. Also, this axillaris possesses lanceolate to elliptic laminae and also elliptic to lanceolate petals. The micromorphological studies also supported our treatment, and thus we reinstated the status of Sonarla axillaris here. The last group which occupies a single species, that is Sonarla lundini, and they are species with prostrate or creeping stem. And this is recently we described from the Western Ghats. Actually, this is the only one species coming under this group from India. The other species like Prostrata, Sonarla reptans and Sonarla reptans all are reported and widely distributed in Indochina and Malay archipelago. Next, going to the discussion. The 49 species and 1 variety are found in India. Out of this, highest species diversity is seen in Western Ghats with 43 species and 1 variety. Out of this 43 species and 1 variety, 42 are endemic to Western Ghats. As I already mentioned, I categorized different groups. The first one is the ecolocent to subecolocent with 11 species. Second group is with 7 species, and the third group is with 24 species and 1 variety. And the last group occupies a single species, that is Sonarla lundini. Next is about the inflorescence architecture. The characteristic scorpioid cyme is more or less consistent within the genus. The other cymose types in the genus are represent a proliferation or reduction based on the cymose unit, and they are incorrectly described as curved raceme or umbel in many species. So these different forms have not been described earlier for any comparative interpretation. So here, first time in Sonarella, we try to classify different types of Sonarella for a taxonomic purpose. The first one is scorpioid, which is very common. The second one is double scorpioid cyme. Many species possess this double scorpioid cyme when the partial inflorescence begin with a dicacial branching. It is very unique in Sonarella, Sonarella elegans and Sonarella speciosa. The last in the first column, the last image. And the another one is pseudo umbel. If the pedicels are very closely set and erect, the inflorescence become umbel shape, which later becomes scorpioid, as in Sonarella scapigera, Sonarella roxburghi, and Sonarella kanyakumariana. And also some species producing Sonarella solitary flower, as in Sonarella arguta. A paper on the inflorescence architecture we recently communicated. And also the characteristic features of anthers are also we can use as a distinguishing feature in Sonarella. Here from these images, it is very clear that this anthers serves an important identifying feature in Sonarella. Then conclusion and summary. Our work is mainly based on the extensive field exploration and herbarium data. 6 new species were discovered. They are Sonarella latratica, Sonarella stolnifera, Sonarella longipedangulata, Sonarella congonensis, Sonarella luntini, and Sonarella roxburghi. Sonarella axillaris and Sonarella gampli were reinstated from the synonymy of Sonarella versicolor and speciosa respectively. 11 names are lectotypified during my study. 30 names are synonymized: Phyllogathis indica, Sonarella gadgiliana, etc. Next is the IUCN conservation assessment. 25 taxa are endangered and 20 as critically endangered, 2 as least concern, 1 as near threatened. And the greater diversity of Sonarella occurs in the Western Ghats, where 43 species and 1 variety found, with 86% of endemics. All collected samples are maintaining in the Calicut University Botanical Garden. These are the diverse habitats of Sonarella in Western Ghats, like sholas, forest margins, grasslands, guard road cuttings, and cliffs and rock crevices of mountains and hills. And they are also showing close association with many lower group of plants. These are the present situation of the type locality of many species. In the first column, that is that I taken when I started my study. But the other side, that is now we exploited these areas that much for guard road widening, plantation activities, and also for mining and tourism related construction activities. And this current availability of large databases of species distributions of Sonarella and the occurrence records from our present study offers outstanding opportunities to identify the conservation gaps within the current protected area network, specifically focus on the narrow endemic Sonarella species. And I think it will serve as a valuable resource for devising measures aimed at filling gaps in forest conservation within Western Ghats. These are some of the major publications from our studies. These are some of the major funding and recognition I received during my study. One is the Royal Botanic Garden Edinburgh Sebald Fellowship. Other one is the research grant of International Association for Plant Taxonomy. The other one is Aspire Scholarship of Kerala government to work on the different aspects of Sonarella. These are some of the images which I taken during my visit to Royal Botanic Garden, Edinburgh, Kew and the Natural History Museum. I also got a chance to get an exposure to study the evolutionary implications of inflorescence development and floral ontogeny in Sonorella as part of Sebald Fellowship. And also we initiated a collaboration with the Neotropical Melastomataceae team. These are some of the newspaper cuttings about our studies on Sonorella. And I already mentioned many of the type localities we are exploiting for the developmental activities, but still these plants are developing some adaptation to survive in this condition. Yes, they are running to standstill. So I am thanking to all funding agencies and also my PhD supervisor, Professor Santosh Nambi, for his unending support, and also for Botanical Survey of India for giving me a chance to continue my studies on Sonorella. And thank you.
So we have sent all the papers for publication already.
5 are published, 1 is awaiting publication.
You are already accepted.
Yeah.
That means already that the literature available.
Yeah.
Many ones already that are available.
Sonorella.
Endemic species and other described earlier.
Yeah.
Other people. Money.
Yeah. Herbarium. In Calicut University Herbarium. Yeah, we deposited that.
So that means sent all the already for publication and
yeah.
One is that already.
Yes.
Now, any questions? Only 2 minutes. Was type
yes, sir.
Yes, sir. Yes.
Because that is a must as per the government of India role at NBA. Many universities, they don't submit. That is what we're doing. Because afterward, you can't find that type specimen. Many universities say we have lost, we don't have all those things. That is very important.
Problem during my work also, sir, because many new species, they are not depositing the specimen. So we are communicating with the authors, but they are not responding properly. Sometimes they are not picking the call also. So continuously we are calling or continuously we are visiting their herbarium. They will say, sometimes they are saying it's lost. So that's the situation.
I would like to ask Dr. Mao, is it molecular analysis mandatory now for the description of new taxon?
It's not necessary. You can publish still many species without molecular studies.
Dr. Yadav sir, is it mandatory?
Yes, in fact, molecular data is a supplementary data. And if on the basis of morphological characters a species can be described as a new species, there is no need of molecular data. But if evolutionary studies are to be done, phylogenetic studies are to be done, molecular data is to be taken into consideration.
I just started the molecular works for the phylogeny, and also it will be useful for the evolution of this inflorescence. Also, we can use molecular studies, so we can predict which group evolved first, like that.
Thank you.
Thank you, sir.
How is it?
Now, I would request Shavandi Kumbhare, Manoshri Mondal, and Roshan Kumar to be ready for Young Botanist contest, and also the judges, Dr. Rajesh Arora, Professor Girdhar, and Professor Shekhawat, GS Shekhawat. 5 minutes, Dr. Manoharachari is preparing result for the women botanists, and then afterwards. बॉटनिस्ट के कॉन्टेस्टेंट्स हैं यहाँ पर? Shawan, Roshan, somewhere Roshan, I'm not able to see if he's in the house. Manashri Mandal, she's here. Where is Roshan? Roshan Singh from NIPGR. I am, must be all around. Phone number. All right, as per the decision of the House, specifically past presidents and the senior members of the Indian Botanical Society, since we are getting too late and the EC members are waiting across the length and breadth of the country for online joining, we are postponing Young Botanist Award Contest for tomorrow morning. Time we will decide and let you know during, I suppose before we will have memorial lectures, and after that we will think about it. Because memorial lectures are already there on the timetable. So we will start at 9 o'clock tomorrow morning for the memorial lectures. two memorial lectures, Professor A.K. Sharma memorial lecture and L.B. Kajale memorial lecture. And after that, during that, we will announce what will happen. Sorry? We will try to manage EC meeting here only. We are trying. Give us 10 minutes time. We will try to manage it here.